Formation of platelet. Platelet plaque formation involves three phases. Platelet adhesion. When the endothelium is injured, platelet adhesion is exposed to subendothelial collagen by von Willebrand factor. Platelet activation. After adhesion, platelet undergo morphologic changes and become activated and then release granules containing ADP, thromboxane A2, secretin, platelet activating factor, and thrombin. Platelet aggregation, ADP, thromboxane A, secretion, and PAF bind with the resting platelets circulating nearby and activate them. Intracellular calcium increases in these activated platelets. Increased cytosolic calcium leads to release of ADP, thromboxane A2, and secrotonin that activates other platelets. Activation of glycoprotein receptors. Glycoprotein receptors bind with fibrinogen. One fibrinogen molecule binds with glycoprotein receptors on two separate platelets, resting in platelet cross-linking and platelet aggregation. Figure: thrombus forming at the site of damaged vascular wall, and the role of platelets and clotting factors. Platelet membrane receptors include the glycoprotein receptor binding to collagen, GPIB receptor binding von Willebrand factor, which binds fibrinogen and other molecules. Antiplatelet prostaglandin is released in the endothelium. Importance of platelet plug. The platelet plug is a crucial step in homeostasis and plays a vital role in preventing excessive blood loss after vascular injury. Immediate response to injury. When a blood vessel is damaged, platelets quickly adhere to the exposed collagen at the injury site, forming a temporary seal. Prevents blood loss. The blood acts as a physical barrier to reduce bleeding until a stable blood clot forms. Activates coagulation cascade. Platelets release clotting factors ADP, thrombin A2 that promote further platelet aggregation and fibrin formation, strengthening the clot. Formation for a stable clot. The platelet plug provides a surface for fibrin mass formation, leading to a durable blood clot. By sealing wounds quickly, it reduces the risk of pathogenic entry. Consequence of platelet plug failure: excessive bleeding, delayed wound healing, increased inflammation risk. Thus, the platelet plug is essential for primary homeostasis and maintenance of vascular integrity. Nice to know: platelet aggregation substances ADP, thrombin A, and serotonin TH. Name the clotting factors chronologically. Clotting factors: fibrinogen, prothrombin, tissue factor, calcium, factor 5, factor 7, factor 8, factor 9, factor 10, 11, 12, 13. Prekallikrein and platelets. Nice to know for VIVA factors. It is not a separate entity but has dropped substances necessarily for clotting are called clotting factors. Clotting factors were named after the scientists who discovered them in their part in the activity, except for 9. Clotting factor 9 or Christmas factor was named after the patient it was whom discovered. Primary factors for blood clotting. These are the factors for present blood clotting: fibrinogen factor 1, prothrombin factor 2, factor 10, and calcium factor 4. Coagulation of blood. The process by which soluble fibrinogen is converted into insoluble fibrin by means of fibrin and a clotting is formed is known as coagulation. Coagulation of blood. Definition: the process by which soluble fibrinogen is converted into insoluble fibrin by means of thrombin is a blood clot is formed by known as coagulations. Basic steps of blood coagulation. Clotting takes place in 3 essential steps: formation of thrombin activator by extrinsic or intrinsic pathways. Extrinsic pathway begins with the trauma of the vessel's wall of extravascular tissue. Intrinsic pathways begins with trauma blood itself. Conversion of prothrombin of thrombin by prothrombin activator. Conversion of soluble fibrinogen into insoluble fibrin by means of thrombin and formation of a clot. Prothrombin, calcium, thrombin, fibrinogen, fibrin monomer, fibrin fibers, cross-linked by fibrin fibers. How Sutter power factor is activated. Extrinsic pathway, intrinsic pathway. Discuss the extrinsic mechanism of blood coagulation. Extrinsic mechanism of blood coagulation. The extrinsic pathway for initiating blood formation of prothrombin activator begins in the traumatized vascular wall of traumatized extravascular tissue that comes in contact with blood tissue platelets. Then write the intrinsic pathway of prothrombin activator formation, showing the use of calcium in it. Give an outline formation of activating factor pathways. Describe the blood coagulation in a test tube. Blood coagulation in a test tube. Blood coagulation takes place in a test tube in an intrinsic pathway because contract with foreign substance such as gas, paraffins, silicon, polythene, silica activates factor 12, which initiates intrinsic pathways. Describe intrinsic pathway from above. Chapter 6. The heart. The heart is hollow, somewhat conical and muscular organ situated in the middle mediastinum, covered by the pericardium. It is about the size of a clenched fist in a normal individual. It consists of 4 chambers, right and left atria, right and left ventricles. The atria are separated from each other by interatrial septum and the ventricles by interventricular septum. Interatrial septum and interventricular septum. Right atrium receives deoxygenated blood via superior and inferior vena cava and by coronary sinus. The blood enters the right ventricle through the right atrioventricular orifice. Atrioventricular orifice. The right atrium receives deoxygenated blood via superior and inferior vena cava. The blood enters the right ventricle, the atrioventricular orifice. And thence reaches into the pulmonary trunk, whereupon the blood reaches the pulmonary capillary plexus for oxygenation. Right atrium receives oxygenated blood via 4 pulmonary veins. From the blood enters the left ventricle through the left atrioventricular orifice and subsequently eject into the ascending aorta for distribution into the entire body. The right atrium deals with the deoxygenated blood for pulmonary circulation. The left atrium deals with oxygenated blood for systemic circulation. Measurements: apex and base. Apex 12 cm, anterior posterior diameter 6 cm. Apex to base 12 cm. Anterior posterior diameter 6 cm. Anterior posterior diameter 6 cm. Anterior posterior diameter 6 cm. Widest transverse diameter 8 to 9 on a radiograph on the end of maximum inspiration in the standing position. The transverse diameter should not be more than 1/2 of the diameter of the thorax. Adult male 180 to 340 grams. Adult female 230 to 280 grams. Presence part. The heart presents apex, base and 3 surfaces. Apex, base and 3 surfaces. Sternocostal, diaphragmatic, and left surface. Sternocostal, diaphragmatic and left surface. 3 borders. Right, left, inferior, right, inferior and left. Right, left and inferior. Right, left and inferior. Apex, the conical area formed by the right ventricle and the directed downwards, forwards and to the left 5th intercostal space. The and 9 centimeters from the middle line, slightly below the median to the left nipple. Apex is separated from the anterior thoracic wall by anterior part of the left lung and pleura. Apex beat is a forward thrust felt on the 5th intercostal space during interventricular systole due to twisting of the heart by the vortex-like disposition of the ventricular muscles. close to the apex. In normal adults, it can be felt in the left fifth intercostal space, just median to the midclavicular line, about 5 7 to 9 centimeters from the mediastinal line. Thus, the position of the nipple is not reliable guide to the apex of the beat. In the newborn, the apex beat is usually felt on the left fourth intercostal space or at lateral to the midclavicular line. About 2 years, it reaches the adult position. Base. Base or posterior surface is most fixed part of the heart and somewhat quadrilateral in outline. It's directed backwards to the right and formed only by the two atria. Borders. Above, bifurcation of the pulmonary trunk separated by bifurcation of the pulmonary trunk. Above, separated by transverse pericardial sinuses. Below, posterior part of the atrioventricular groove containing the coronary sinus and anastomosis in the right and the left coronary arteries. Anterioventricular groove, posterior of the posterior part of the atrioventricular groove containing the coronary sinus and anastomosis of the right and the left coronary arteries. Right side, right border of the right atrium, ascending from the superior to inferior vena cava. Left border, left of the left atrium, extending from the upper to the lower pulmonary veins. Upper to the lower pulmonary veins. Left border, upper from the upper to the lower left pulmonary veins. Features: superior vena cava opens in the upper posterior part of the right atrium. Inferior vena cava opens the lower posterior part of the right atrium. 4 pulmonary veins, 2 on each side, open on the posterior surface of the left atrium. These 6 veins are enclosed in a single tube of serous pericardium, which is arranged in the form of an inverted J. Between the limbs of J, the posterior surface of the left atrium forms the anterior boundary of the oblique pericardial sinus. The shallow ventricular arterial groove may be found between the two atria and indicates the posterior attachment of the atrial septum. Vertebral levels. The recumbent spine position. The base lies opposite to the bodies of the 5th to 8th thoracic vertebra. In standing position, it lies opposite to the thoracic 6 to thoracic 9 vertebra. Relations between the base of the heart and the vertebral column. The following structures are intervened. Clinical importance. Distending of the left atrium or the mitral stenosis may produce difficulty in swallowing due to esophageal compressions. Otter's syndrome. Clinical base of the heart is represented by a hole extent from osculatory areas to the aortic sound of the right second interstitial space, and pulmonary area of the left second interstitial space. Right border. It is a round and convex, formed only by the right atrium. It extends from the 8th side of the opening of the superior vena cava and the inferior vena cava. The border separates the base of the sternocostal surface. A shallow ventricular groove known as sulcus terminalis accompanies the right border of the corresponding with the crista terminalis in the inferior of the right atrium. Sometimes the right border is described in the right surface. Relations. The 3 layers of pericardium, right phrenic nerve and the pericardiophrenic vessels and the mediastinal pleura, and the cardiac impression of the right lung immediately in front of the hilum. Inferior border. It is sharp and separates the sternocostal from diaphragmatic surface. It extends from the opening of the inferior vena cava to the apex of the heart. The border is accompanied by the marginal branch of the right coronary artery. It corresponds in vein close to the apex. It presents a notch known as the incisura apicis cordis and gives passage to the interior interventricular branch of the left coronary artery. Left border. The ill-defined convex border separates the sternocostal from left surface. It extends from the left auricle to the apex of the heart, with the convexity directed upward on the left. The border is accompanied by the marginal branch of the left coronary artery. Marginal branch of the left coronary artery. The left border. Sternocostal surface. It is directed forward and upward, and separated from the base of the right border from the diaphragmatic surface by inferior border and from the left surface by left border. This surface is formed by the following parts of the heart: anterior surface of the right atrium and the auricle, a part of the anterior surface of the left auricle. Anterior surface of the right atrial auricle, a part of the anterior surface of the left auricle. Anterior surface of the right ventricle, anterior surface of the left ventricle. Anterior surface of the right auricle and features sternocostal surface. Anterior part of the atrioventricular groove passes downward and to the right to the right between the right atrium and the right ventricle. Atrioventricular groove downwards to the right between the right atrium and the right ventricle. The groove lodges the trunk, right coronary artery, and is crossed by the anterior cardiac veins. Anterior interventricular groove passes downward parallel to the left border of the heart and meets the inferior border of the incisura apicis cordis. The groove lodges the anterior interventricular branch of the left coronary artery and the great cardiac vein. The groove indicates the anterior attachment of the interventricular septum. Relations covered by the 3 layers of the pericardium. Posterior surface of the body of the sternum, 3rd to 6th costal cartilage both sides. Anterior margin of the lungs. Diaphragmatic or inferior surface. It is flat and faces downward and backwards. Downward and backwards. It faces forward and upwards. It is formed only by 2 ventricles. Diaphragmatic surface. It is separated from the sternocostal surface by inferior border from the base of the posterior part of the atrioventricular groove.
গ্রুপে কি থাকে এগুলো আমি আপনাকে ছবি দিয়েছি, আপনি বুঝতে পারেন নাই যে আপনি ছবিগুলো দেখছেন না। এই যে বাদন সানি গতকাল যেগুলো সেগুলো এখন আপনি দেখানো না, এই যে ফিটাস আপনি সেগুলো দেখানো না, ঠিক আছে, এটা আপনি বুঝে গেছেন আশা করি। এই যে দেখেন, এই যে এই ছবিটা দেখলেই আপনার সব হয়ে যায়। এই ছবিটা আপনাকে একটু বের করে নিয়ে একটু ঘুরাইতে হবে আর কি। এই যে দেখেন গ্রুপ, এই যে গ্রুপের মধ্যে একই সঙ্গে 18 আছে, বেইনো আছে, 18 আছে, বেইনো আছে। এই ছবিটা আপনি দেখছেন কিন্তু আপনি বুঝতে পারেন না যে এটা এর থেকে নেওয়া ছবি, স্নেহার থেকে নেওয়া ছবি। একই ছবিতে 18 আছে, ভেইনো আছে। আপনার বইতে হচ্ছে যে একবার দিয়েছে 18, একবার দিয়েছে ভেইন, একসঙ্গে না দেয় তো আপনি বুঝতে পারেন না। কিন্তু এখানেও তো একসঙ্গে সব দেখা দিচ্ছে না, যে গ্রেট কার্ডিয়াক ভেইন আর
এটা কি এই অ্যানুলাস অফ বিউসেন্স।
অ্যানুলাস অফ বিউসেন্স এগুলো এত লাগে না, আপনাকে গ্রুপ জিসু শিখতে হবে, ঠিক আছে। এই যে এটা হলো সামনে, এটা হলো পিছনের। তো এই আপনি একই সঙ্গে হার্ট এবং বেসাল পড়তেছেন বলে আমি একবার হার্ট বলতেছে বলে ছবিগুলো মিক্স আপ হয়ে গেছে আর কি। এটা হলো আপনার অরিজিনাল হার্টের ছবি না। এই যে আপনি যখন হার্ট ধরবেন, এই যে ফ্যাটের ভিতরে কি থাকে, আপনি হার্ট ধরছেন, লাইফ ধরছেন, কিভাবে ধরছেন? আমরা ওইভাবে ধরি না, আমরা ধরছি এইভাবে, অ্যাপেক্সটা এদিকে মুখের, আমরা ধরছি এইভাবে,
লেফট সাইডে না,
আমরা এইভাবে ধরছি,
আমরা ধরছি এইভাবে। একই কথা হইলো, মানে আমার হার্টের উপরে হার্টের কোন সারফেসটা আছে, ডায়াফ্রাগমেটিক সারফেস। হ্যাঁ, আর যে বেস, পিছন দিকে হলো বেস। তার মানে হার্ট কিন্তু বেজের উপরে বসে না, বসে ডায়াফ্রাগমেটিক সারফেসের উপরে। যাই হোক, এইভাবে ধরে এটা আর কি। যাই হোক, ছবিটা গ্রুপটা খেয়াল করলে হয়ে যাবে, সমস্যা নাই, এগুলো হয়।
মিডল কার্ডিয়াক ভেইন
ভালো আছে, বুঝা আছে যে উনি গুরুত্বপূর্ণ বিষয়গুলো ছাপ দিচ্ছেন না, তাই না? সেটা ভালো।
left surface.
এখন কি করেন সেই হার্ট
Great cardiac pain.
আপনি পড়েন যে কোনো ছবির বই পাশে থাকে না কেন আপনাকে ছবি বলেছি যে পাশে নিয়ে ঘোরার জন্য ট্যাপটা নিয়ে এসে এইভাবে হেলান দিয়ে রাখেন না একবার ট্যাপের ছবি দেখবেন একবার বই দেখবেন তাই না বস অ্যানাটমি তো পাশে ছবি বই লাগবে ছবি ছাড়া অ্যানাটমি কি কিনছেন আমি তো কোনোদিন একটা বই পাশে দেখলাম না দেখাবো না তো আমি তো মোবাইলে ছবি দিচ্ছি আমার কাছে আরো ভালো ভালো একটা ছবি আছে না তো বইগুলো ওকেও দিয়েছি কিন্তু ও তো ওগুলো কোনো দেখেও না
ট্যাপ কি ভালো আছে না দিয়েছে
ভালো আছে
আছে না
ভালো আছে তো
ট্যাব হোক আর মোবাইল হোক আপনি স্ট্যান্ড আছে পাশে বসাইয়া ছবি দেখেন না বইয়ের ছবি ছাড়া আপনার অ্যানাটমি কি
অ্যানাটমি মানে
ছবি
চোখে বাসতে হবে
হ্যাঁ সবকিছু পরে একটা টেকনিক আছে তো
ফিজিওলজি হচ্ছে ঠোঁটস্থ অবস্থা লাইটিং এরিয়া
যেমন তো অনেক ছবি আছে ছবি ছাড়া কেমন
লাইটিং এরিয়া
সায়েন্স কত বলতো তার মুখস্থ এখানে লেফট সাইড এগুলো বুঝাতে হবে এগুলো মুখস্থ করে ফেলতে হবে
বলো অ্যানাটমিক পজিশন অফ হার্ট সবচেয়ে বেশি দরকার
পেরিকার্ডিয়াম তো গাগাটা
আচ্ছা আসো পেরিকার্ডিয়াম
ওরে ডক্টর প্রথম বিষয় যে সাবজেক্ট স্টার্ট হবে
সেটাই তো বলছি
ডক্টর প্রথম বিষয়ের মধ্যে দেখলাম গ্রেট কার্ডিয়াক পেন ওর কার্ডিয়াক পেন
তাহলে দত্ত থেকে বের করো পেরিকার্ডিয়াম বের করো দত্ত
পেরিকার্ডিয়াম তো দত্ত থেকে বললে বুঝতে হবে না এখান থেকে পেরিকার্ডিয়াম পর দত্ত হয়ে হার্টে চলে যাবে তাহলে কি এখানে পেরিকার্ডিয়াম শেষ করে এই Introduction of physiology. What is physiology? Physiology is the science that explains the physical and the chemical mechanisms that are responsible for the origin, development, and process of life. Physiology is the dynamic study of life. Physiology describes the vital functions of living organisms and their organs and molecules. Explains physical and chemical mechanisms that are responsible for the development, origin, and process of life. What are the branches of physiology? The branches, each type of life form a simple various of large tree to the complicated human being has its own functional characteristics. Therefore, a vast physiology can be divided into a viral physiology, bacterial physiology, cellular physiology, plant physiology, human physiology. Define human physiology. The science of human physiology attempts to explain the specific characteristics and mechanism of human body that make the living being. What is the goal of physiology? The goal of physiology is to explain the physical and chemical factors that are responsible for the origin, development, and process of life. Chemical factors responsible for the origin, development, and process of life. Responsible for the origin, development, and process of life. Responsible for the origin, development, and process of life. Automaticity of the body. Automatic of the body refers to the ability to certain physiological process to occur anatomically and involuntarily without conscious thought and effort. This process is regulated by autonomic nerves system ANS, autonomic intrinsic control mechanism. Key examples of autonomicity in the body: heartbeat. As a node generates electrical impulses and spontaneously ensuring the heart beats without conscious control. Breathing. While breathing can be consciously controlled, it continues to autonomically in medulla oblongata in the brainstem. Digestion, parasympathesis and secretions. Peristalsis and secretions. A smooth muscle generate tract and contract rhythmic and digestive enzymes that are released without conscious input. Papillary reflex. The pupil dilates or contracts in response to light levels autonomically. Blood pressure regulation. Blood pressure adjusts the diameter and heart rate changes anatomically to maintain stable blood pressure. Hormonal secretions. The pancreas releases insulin and responds to blood glucose level without conscious innervations. Regulation of automaticity. Nervous system and ANS. Increase heartbeat rate, dilute pupils, parasympathetic divisions, slow heart rate, stimulates digestions. Hormonal and local feedback mechanism. What is autonomicity important? Ensures cervical clinical functions, operate continuously, reduces congestive load, allowing the brain to focus on voluntary actions, maintains homeostasis. Reference. Nice to know the bar. So the hormonal secretions, blood pressure regulation, papillary reflex, and digestion, and then heartbeat and breathing. So what is autonomically important? Ensuring cervical and clinical functions. Operate constantly, reducing the congestive load, following the brain, and focus on voluntary actions. Maintenance of homeostasis. Reduces cognitive load, allowing the brain to focus on voluntary actions, maintains homeostasis, ensures survival critic functions. Nice to know, the body is actually a social order of 300 trillion cells, 100 trillion cells organized into different functional structures called tissue and organs. The importance of physiology in medical science. Physiology is a function of medical science because it explains how the human body functions in health and disease. The bridges the gaps between the basic science like anatomy and biochemistry and clinical medicine, enables doctors to diagnose, treat, and prevent illness effectively. The role of physiology in medicine. Understanding the normal body function, homeostasis. Physiology explains how organisms, systems, and cardiovascular systems, respiratory systems work together to maintain homeostasis. Explain knowing the kidneys regulating blood pressure helps in hypertension. Basics of disease mechanism. By understanding normal function, doctors can identify what goes under disease. For example, diabetes, defect in insulin secretion, and heart failure. Breakdown of cardiac muscle contraction. Development of diagnostic tools. A 34-year-old female came to the output department with complaints of tiredness. Patient complains with tiredness, shortness of breath, and palpitation on exertion. Laboratory tests reveal that hemoglobin is 9, hematocrit is 26%, 36%, and MCV is 30 femtoliter. MCH is 20 picogram. What is the possible clinical conditions occurring in this case? Possible clinical condition. The patient's lab findings suggest microcytic hypochromic anemia, low HB, low MCV, low MCH. Most likely causes: iron deficiency anemia due to chronic bleeding losses and inadequate dietary intake, thalassemia. Genetic defects in hemoglobin chain synthesis, thalassemia. Anemia and chronic thalassemia. Anemia of chronic disease seen in chronic infections, inflammations. Other types of anemia, example normocytic, normochromic, MCV, MCH. Example acute blood loss anemia, chronic disease. Microcytic anemia. Example vitamin B12 deficiency, pernicious anemia, macrocytic anemia. Other types of anemia include normal high MCV. A pregnant woman of low socio-economic status has come to the output department with complaints of vertigo, weakness, and palpitation. An examination doctor found that he is very pale. Investigations reported that hemoglobin is 8, MCV is 60, MCH is 20. Iron deficiency anemia, macrocytic hypochromic anemia. Justify the diagnosis. Clinical symptoms: vertigo, palpitation, and weakness. Signs of anemia: pallor, status of hemoglobin, hemoglobin low, risk factor: low pregnancy, low social economics, poor nutrition, pregnancy, increase in demand for fetal growth. 4 causes of iron deficiency anemia: dietary, chronic blood loss, increased demand, and malabsorption. Next step: oral iron therapy, ferrous and dietary advice. A 30-year-old lady came to the doctor with a history of tiredness and palpitation in examination. Her hemoglobin reported reveals that her corpuscular volume is MCV 50. Mean corpuscular hemoglobin is 20 and corpuscular hemoglobin is 25. In this case, article presents with symptoms: microcytic hypochromic anemia. Types of anemia. The patient has hypochromic anemia. Most likely causes of hypochromic anemia are iron deficiency anemia, inadequate iron, manifestation GIT, bleeding. A 35-year-old female patient came to pallor, weakness of 2 months, and she had weakness for 2 months, and she had a history of prerectal bleeding for 6 months. The common Blood examination for hemoglobin 6, hemoglobin MC V60, MCV 20, MCV 28. Diagnosis classification: iron deficiency anemia. Iron deficiency anemia. Pale skin weakness. Chronic blood loss. Confirmed test: serum ferritin, serum iron, peripheral blood film, stool occult blood, stool, colonoscopy, endoscopy, TIBC. Dietary deficiency, malabsorption. Exam of summary: diagnosis and justification. Take probable reasons: iron deficiency anemia, thalassemia, anemia of chronic disease, and sideroblastic anemia, normocytic, microcytic, macrocytic, normochromic, and hypochromic. Clinical conditions: iron deficiency anemia. Common causes of IDA: low serum ferritin, low serum iron, high TIBC, total binding capacity, high total iron binding capacity, TIBC, total iron binding capacity, low transferrin saturation, peripheral smear findings, pencil cells, anisocytes. Anemia may be defined as a clinical condition characterized by the pale color of the skin, mucous membrane, and due to qualitative or quantitative deficiency of hemoglobin below the lower limit of the peripheral blood with respect to age and sex. Diagnosis: iron deficiency anemia, thalassemia, differential diagnosis. Serum iron profile has been followed bone marrow study. Hypochromic, microcytic, normochromic, normocytic, hemorrhage, hemolytic, or aplastic anemia. Macrocytic, megaloblastic, pernicious, folate deficiency. Important laboratory investigations: serum iron profile, HB electrophoresis. Anemia, electrophoresis. Histological findings: characteristic change, hyperplasia, case of anemia, iron therapy, parental iron therapy, hemoglobinopathies. Define cell and enumerate the number membrane bound organelles of the cell. The cells are the structure function of all living organisms. Membrane bound organelles of typical animal cell below: mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. Mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. What are the functions of lysosome and endoplasmic reticulum? Digestion of macromolecule, phagocytose, microorganisms, foreign particles, cell debris, damaged cell organelles. They act as autophagic vesicles to destroy organelles of cytoplasm before the cells dies due to lack of oxygen, so they are called suicide bug. They act organelles of cytoplasm before a cell dies due to lack of oxygen. Droplets of hormone and other cellular excessive substance are broken down by the lysosomes. Segregation of protein, function of lysosome, function of endoplasmic reticulum. Segregation of protein: initial glycosylation or glycoprotein, glycoprotein, synthesis of phospholipid, assembly of multicultural protein, multi-chain protein, post-transitional modification of newly formed polypeptides. So the membrane bound organelles are mitochondria, endoplasmic reticulum, Golgi apparatus, reticulum, peroxisome, and lysosomes. So mitochondria, endoplasmic reticulum, Golgi apparatus. Mitochondria, endoplasmic reticulum and Golgi apparatus. Mitochondria, endoplasmic reticulum, Golgi apparatus. So basically, the membrane-bound organelles are mitochondria and smooth reticulum, mitochondria, lysosome, peroxisome, and peroxisome, mitochondria, and Golgi complex. So mitochondria, Golgi complex, smooth endoplasmic reticulum, and then peroxisome and lysosomes. Lysosome, the duties or the how the lysosome works. The lysosome function is lysosome breaks down phagocytes, foreign bodies or microbes that are phagocytes, and it also destroys hormonal particles and cell debris, and it also kills a cell before the lack of oxygen, works as a suicide bag. Smooth endoplasmic reticulum synthesizes protein, multipolypeptide and peptide, and glycoprotein, and also phospholipid. So glycoprotein, phospholipids, proteins, and polypeptides. And then write down the difference between organelles and inclusion bodies. Organelles and inclusion bodies. Organelles, permanent. So organelles and inclusions. Inclusions are temporary, non-living, metabolically inactive, composed of various substances including pigments, fat droplets, and glycogen. So inclusions are temporary, organelles are permanent, inclusions are temporary, non-living, living, metabolically active, metabolically inactive, and then composed of protein and other lipids, composed of various substances including pigments, fat droplets, and glycogen. So metabolically active, inactive, permanent, temporary, and then these are composed of pigments and other substances, and these are composed of proteins and lipids. So difference between mitosis and meiosis is that the site is different, the chromosome number is different, the visibility of chromosome is different, and the crossing over is different, the prophase is different, the interphase is different, the phases are different, the daughter cell are different, the site are different, and the genetic crossing over is different. So the result of mitotic division is that zygote formation, 4 haploid, and then gamete. The 4 results of mitotic division: gamete formation, 4 haploids, and zygote formation after fertilization. Lysosome and endoplasmic reticulum. Lysosomes are also called suicide bags as they contain a wide range of hydrolytic enzymes. They are membrane-bound organelles for containing a wide range of hydrolytic enzymes that are responsible for the destruction of foreign bodies and also the phagocytosized microbes by the cell, and also for the cell's own genetic destruction. These are membrane organelles responsible for the cells. These are membrane organelles containing a wide range of hydrolytic enzymes which are responsible for the breakdown of foreign bodies and also the cell's phagocytosized microbes and also the cell's plant death. They contain hydrolytic enzymes which help the cell in various ways. Smooth endoplasmic reticulum. These are small cavities or chambers that are interlinked with one another inside the cell. They contain a different type of substance other than the cell cytoplasm. The total length of the smooth endoplasmic reticulum might be greater than the length of the cell itself, and they have crystal. So the endoplasmic reticulum are of two types, rough and smooth. The rough endoplasmic reticulum contains ribosomes, so they are involved in protein synthesis, multi-chain production, and then also post-transitional modification of polypeptide. They also segregate proteins and also phospholipids and glycoproteins. It's in the endoplasmic reticulum are devoid of ribosomes, but the rough endoplasmic reticulum were in the pancreas, the plasma cell, and the fibroblasts. But the smooth endoplasmic reticulum are in the liver cells and the adrenal gland. They are involved in oxidizing and conjugating substances. They also play an important role in muscle contraction, and they also synthesize glycoproteins. So the cell membrane is a lipid bilayer membrane that separates the external surface from the cell's internal surface. Its thickness is about 7.5 to 10 nanometer. Works. It gives the cell a structure, also maintains size and shape, also maintains pH, and regulates size and shape by maintaining osmotic pressure by the sodium pollution pumps. It is also responsible for the protection of the cell, also the immunoglobulin sites, also the protection of the immunoglobulin sites, also gives immunity to the cell, and also works as a site for stimulus and adheres to receptors. So neighboring cells has say molecules, transport-specific molecules, chemicals, external stimuli, and through cellular contact. Short note: mitochondria. Mitochondria is a membrane-bound cell organelles that generate most of the chemical energy needed to power the cell's biochemical receptors. So the functions are mitochondria is the powerhouse of the cell. Mitochondria is a membrane-bound organelle of the cell, composed of an outer smooth layer and an inner rough layer. Inner membrane composed of a smooth outer membrane and an inner rough membrane. An inner membrane, a smooth outer layer, and an inner highly folded membrane called cristae. Incomplete partitions called cristae, which work as mitochondria are cell organelles. Mitochondria are membrane-bound cell organelles which have a mitochondria are membrane-bound cell organelles consisting of an outer smooth layer and an inner multifolded layer called cristae. The cristae increases the surface area of the mitochondria. Mitochondria is involved in the production of ATP by using oxygen. Production of ATP. It also is involved in the production of proteins and also oxidizing fatty acids. Oxidation of fatty acids and the accumulation of calcium. It is also involved on the Krebs cycle, oxidative phosphorylation, and electron transport system. So Z1 phase, S phase, Z2 phase. It has three phases: Z1 phase, S phase, and Z2 phase. The G1 phase is the gap in between the previous mitosis and the DNA synthesis. The S phase is particularly characterized by the DNA synthesis, and the G2 phase is the gap between the DNA synthesis and the mitotic division, mitotic phase. DNA phase, and once the cell enters the S phase, it is committed to mitosis. And in the M phase, the cell divides into two daughter cells. If some cells enter into G0 phase, it will never mitosis again. It's gone beyond the cell cycle. Cytoskeleton of the cell are needed for the division of cellular movement. So cell division and cell movement. Cell division, for example, spindle helps in cell division and cell movement. Motor proteins like dynein and kinesin. Motor proteins which actin filament is, for example, motor protein. Additionally, actin and motor protein contract with actin filament. This is essential for muscle contraction. Actin, microtubule, and other components of cytoskeleton provide a structural support and serve as a tracks for motor proteins like dynein and kinesin, which transport organelles, vesicles, and cellular cargo as well as dynein and kinesin, microtubules and cytoskeleton. So the basic tissues of the bodies are epithelial tissue, connective tissue, muscular tissue, and the nervous tissue. So they originate from all the three germ layers, They have a unique structure where all the cells rely on a basement membrane for nutrition, and the nutrition comes to them by diffusion, and they have very delicate nerve, they have nerve fibers, and they are they originally from and they are jam packed together with very little intercellular substance, and they are labile in nature. So they rely on a basement membrane for nutrition, and because they don't have their own blood supply, they have delicate nerve fibers. They have polarity with an apical facing outwards, and the basement and a basal and a basement layer, apical and a basal surface resting on the basal lamina. They can origin from all the three germinal layers. They are closely packed together with very little intercellular space, and they have metaplasia. Under certain condition, they can change shape. Functions of epithelial cells are: they work as a covering and a protection. They have secretion, also has absorption. They have contractility and sensation and contractility, and they also work as lubrication. They provide lubrication and prevents water loss. Classify the glands on basis of nature of secretion and the modes of secretion with example. So according to the mode of secretion, according to the nature of secretion, according to the mode of secretion. According to nature of secretion, there are serous, mucus, and mixed. The serous are, for example, they are slimy, watery. For example, the serous include the parotid and the pancreas, and the mucus includes sublingual, submandibular. So the mucus includes the goblet cells, and the serous includes the goblet cells, which is unicellular, but there are also multicellular. For example, the palatine cells, for example, the palatine cells. The mode of secretion, the nature of secretion. According to nature of secretion, they have the mucus, the serous, and the mixed. The mucus, for example, the unicellular goblet and the multicellular palatine, and the mucus includes the serous includes parotid and the pancreatic secretions, and the mixed include the sublingual and the sublingual secretions. And according to mode of secretion, they have apocrine, holocrine, and merocrine. Merocrines are the type of secretions that are done by exocytosis of the individual cells. It is the most common type of secretion. Merocrine includes entire cells to be dispersed on the secretion. The cells accumulate lipid and other substances and completely disperse with their secretion. And on the apocrine, only the bits of pieces of debris and cell parts disperse in the secretion. There are ground substances, fibrous substances, and cells. Cells include adipose tissue, leukocyte, and macrophages and mast cells. And the ground substances include glycosaminoglycans, proteoglycans, and glycoproteins. The fibrous substances include collagen, elastin, and reticulocytes. Write down the name of connective tissue fibers with one location of each. For example, surface modification include microvilli, cilia, and flagella. Microvilli are finger-like projections of cytoplasm that increase the surface area and help in absorption. Cilia are motile hair-like structures. They help in whip-like movement, and flagella are also a kind of cilia that are present on the sperm. Flagella sperm helps in the motility of the sperm. So there are epithelial cells, and then granular epithelial cells have lining epithelium and granular epithelium. On the lining epithelium, they have unilayered and multilayered division. On the unilayered division, they have the simple squamous cells, simple cuboidal, simple columnar. These are simple lining epithelium, and then they also have pseudo-stratified epithelium. And on the multilayered division, it has stratified columnar, stratified cuboidal, stratified cuboidal, columnar epithelium, and they have the transitional epithelium. And on the glandular, there is the pancreas and the stoma, and parenchyma and the stoma. Primary cartilaginous joint and secondary cartilaginous joint, also known as synchondrosis and symphysis. Secondary is symphysis. Only hyaline cartilage forming the joint. Thin phases of hyaline cartilage separated by fibrocartilage. So these are separated by fibrocartilage. They are thin cartilage. It is a temporary joint. It is a permanent joint. Hyaline cartilage is converted into bone. Cartilages do not convert into bone. Do not know definite location. Usually locate on medial plane. Locate on the medial plane. Allows no movement. Allows little movement. For example, manubrosternal joint and sternocostal is a costochondral joint. Primary cartilaginous joint, secondary cartilaginous. Secondary cartilaginous joints allows a little bit of movement, but primary cartilaginous joints no movement. Primary cartilaginous joints calcify. Secondary cartilaginous joints not calcify. It has hyaline cartilage. It is divided by fibrocartilage. Example is manubrosternal joint, and this example is costochondral joint. And also known as symphysis, also known as synchondrosis. So hyaline cartilage, fibrocartilage, and then elastic cartilage. Hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage include the cartilage, the nasal cartilage, the laryngeal cartilage, and the tracheobronchial cartilage. Laryngeal cartilage, the costal cartilage, also the costal cartilage. So the nasal cartilage, the costal cartilage, the laryngeal cartilage, and the costochondral cartilage, and the tracheobronchial cartilage. The elastic cartilage includes ear, eustachian tube, and epiglottis. And the fibrocartilage includes the intervertebral disc and the pubic symphysis. Write the difference between various types of cartilage. Hyaline cartilage, elastic cartilage, fibrocartilage. Homogeneous with type 2 collagen aggregate, darker elastic fibers, type 2 collagen, large areas, dense connective tissue with type 1 cartilage. Major cause: chondrocytes, chondroblasts. Typical arrangement of chondrocytes: isolated in small indigenous groups, usually in small isogenous groups. Isolated in small indigenous groups, usually in small isogenous groups. Isolated in isogenous groups. Presence of perichondrium: yes, no, no. This is a perichondrium: yes, no, no. Main functions: provides smooth low function surface, provides flexibility, provides cushioning, are flexible, smooth. Write the morphological classifications of bone with example: long bone, short bone, long bone, short bone, long bone, short bone, long bone. Types of bones are short bone, long bone, and then irregular bones, and then flat bone, pneumatic bone, and pisiform. Carpal, metacarpal, tibia, fibula, radius ulna, vertebra, hip bone, and then scapula, sternum, and then maxilla, ethmoid, and then patella, and pisiform. Epiphysis is the part of the bone that develops from the secondary center. There are three types: pressure epiphysis, traction epiphysis, and atavistic epiphysis. Atavistic epiphysis are independent, physiologically independent bone. They almost live like parasites, getting nutrition from another host. Traction and pull and pressure epiphysis are the covering of the bones that give protection, and traction epiphysis are bones developed from the muscle pulling. The cells of the bone are osteoblast, osteoclast, and osteocyte. Osteoblast, and osteocyte. The osteoblasts are immature bone cells, and the osteocytes are mature inactive cells, and the osteoclasts are cells that remodel the bone. So the joints include diarthrasis and synarthrasis. Diarthrasis includes the pivot, hinge, and plane, and then ball socket, and condylar, and saddle, and ellipsoid. And these are movable, and immovable or partially movable joints are synarthrosis. Synarthrosis include cartilaginous and fibrous. Cartilages include primary cartilage, secondary cartilages, and fibrous include plain and squamous. The difference between transitional and stratified epithelial can be many layers. There is many layers, and transitional epithelial are made out of 3 layers. Transitional epithelial are meant for stopping reabsorption or preventing reabsorption. So they are found on the uterus. So they are found on the urinary bladder. And transitional epithelium are there for protection and for protection and for a labile structure, which regenerate very quickly. So they can be found on the skin. They can be keratinized or cuticle. They can be keratinized, but transitional not keratinized. Haversian tube, Haversian canal, Hoffman's canal, lamellae, lacunae, and then canaliculi. Characteristic of synovial joint. Articular characteristics of synovial joints. Articular surface of the participating bones are covered by the articular joint, articular capsule, outer fibrous layer, synovial fluid, accessory ligaments. So synovial fluid as a articular surface surrounded by joints, synovial ligaments line the whole interior of the synovial except the articular surfaces. Accessory ligaments as inside the joint cavity. The joint is capable of limited to wide range of movements. Typical include femur, miniature into metacarpal modified into clavicle. Factor maintaining distributed synovial joint. Bony contours, ligament, muscle, and atmospheric pressure. Ligament, muscle, and atmospheric pressure. Hinge joint, according to shape of articular surfaces. So they have the plane, for example, intermetatarsal joint, intermetatarsal joint, then pivot, radioulnar joint, and then hinge joint, for example, humeroulnar joint, and then ball socket joint, for example, shoulder joint, and condylar joint, for example, knee joint, and then ellipsoid saddle joint, for example, radiocarpal joint, and carpometacarpal joint. Articular cartilage, a special type of hyaline cartilage that free space from the bones of the joint. Articular cartilage is not covered by the perichondrium. It's fluid, nerve supply, collagen fiber, sound situation, deepest layer of the articular cartilage, bone. The articular cartilage gets nutrition from vessels of the synovial joint, the bone marrow. Functions of the articular cartilage. Smooth surface between the very little function thus acts as a shock absorber. Growing end of the ends of the bone where the symphysis begins first and the diaphysis begins last. Or the epiphysis appears first. Epiphysis appears first and the diaphysis from the knee I flee to the elbow I go. So the ends of the humerus and the radius ulna, and the upper end of the humerus, the lower end of the radius ulna, the lower end of the femur, and the upper end of the tibia fibula. The ends of the nutrition is against the nutritional flow. Upper end of the humerus, lower end of the radius ulna, lower end of the femur, and ulna. The part of the bone where epiphysis appears first and diaphysis appears the last. Haversian canal, central canal, also known as Bachmann's canal, communicates with each other. Lamellae, these are concentric bony layers. And concentric bony layers, and lacunae, these are spaces in between the lamellae. Osteocytes and canaliculi, these are radiating channels can connect the lacunae with each other and with the central Haversian canal. Periosteum, articular surface. Periosteum layer dense fibrous connective tissue. Periosteum as articular surface. The structure, it has sharpest fibers. Articular surface, it has sharpest fibers attached to the sharpest fibers. And functions. For attachment, provides a media for attachment for muscles. And then metaphysis is important. The importance of metaphysis is metaphysis is the most active growing part of the bone. Metaphysis has profuse blood supply from the nutrient periosteum and juxtapepiphyseal. Nutrient periosteum, the juxtapepiphyseal joint. Hair nutrient arteries from hairpin-like capillary tubes. Microorganisms are present in the blood, may settle the slopes, hence it's more common in metaphysis. Metaphysis is the joint capsule attaches to the close. Spermatogenesis is a series of changes resulting from transformation to spermatids to spermatozoa. Spermatids to spermatozoa. Formation of acrosome, which covers half of the nuclear surface and contains enzymes to assist and penetrate the egg and surrounding of the fertilization. Half of the nuclear surface. Formation of acrosome, which covers half of the nuclear surface. Formation of acrosome, which covers the half of the nuclear surface, contains enzyme to assist the penetration of the egg and surrounds layers of fertilization. Condensation of the nucleus. Formation of neck, middle piece, tail. Shedding of most of the cytoplasm. So spermatogenesis changes into shedding of the cytoplasm, condensation of the nucleus, and development of the acrosome. Different phases of fertilization. Penetration of the corona radiata of 200 to 3 million spermatozoa normally dispose in the female genitalia tract, and only 300 to 500 of reach each site of fertilization. Only one of these fertilized eggs, it is thought to be that others aid to fertilize the sperm. In penetrating the barrier is protecting the female gamete. Capacitated sperms pass freely through the corona cell. Capacitated sperms is a process of maturation of sperm that is characterized by the development of acrosome and the shedding of the cytoplasm. The capacitated sperm pass freely through the corona cell. Penetration of the zona pellucida, release of acrosomal enzymes, acrosin, allows sperm to penetrate zona, thereby coming in contact with the plasma membrane and oocyte. This contact releases the lysosome enzymes from coronal cortical granules, lining of the plasma membrane and oosum of the oocyte. In turn, these enzymes alter properties of zona pellucida to prevent sperm penetration and to prevent sperm inactive specific reactors. Sites for spermatozoa, zona pellucida. Other pairs of spermatozoa have been found embedded in the zona pellucida only to see able to penetrate the oocytes. Then fusion. The steps of fertilization are: in the first step, about 300 to 500 spermatozoa or mature sperm cells. Mature sperms are released in the female genitalia, and they swim up to the ovum through the cervix. And they meet the ovum in the fallopian tube, where they penetrate the corona radiata. On the second phase, only one sperm is capable of fusing with the oocytes, after which the zona pellucida reaction happens, which releases lysosomes and prevents further on sperms to fuse, preventing polyspermy. And then on the third phase, after the fusion happens, it initiates the zona reaction, the metabolic reaction of the ovum, and the secondary mitotic division of the ovum. And then ciliated cells slowly move the ovum towards the wall of the body of the uterus, the anterior or the posterior wall of the uterus, where the implantation happens. Write down the results of fertilization. Restore the number of diploid chromosomes. Determine the sex of new individuals. Initiates the cleavage. Mention the ectopic pregnancy. ectopic pregnancy. Implantation of fibroblasts in abnormal location outside uterus is known as adventitia is characterized by the implantation of the egg, implantation of the blastocyte, implantation of the blastocyte in abnormal location outside the uterus. Clinical importance. Ectopic pregnancy occurs in 2% of all pregnancies and occurred for 9% of all pregnancy-related deaths of the mother. In most ectopic pregnancies, the embryonic dies about the second month of the gestation and may result in severe hemorrhage of the mother. Abnormal blastocytes are common. Hydatidiform mole. Pregnancy is 2% of all pregnancy-related deaths of mother and 9% of pregnancy-related deaths of the mother. Mother in most ectopic pregnancies may result in severe hemorrhage and the entire fallopian tube needs to be removed. Uncommon. Short note: capitation. Capitation is the condition of the sperm. If capitation is a period of condition in the sperm tract to acquire the ability of fertilization. Capitation is the period of the sperm. Conducting is the period of conduction of sperm in the capacitation is the period of conduction. Capacitation is the period of conduction inside the female genitalia to acquire the ability to fertilize the ovum. To acquire the ability to fertilize the ovum. Approximately 7 hours. Uric glycoprotein and seminal plasma proteins are removed from the plasma membrane in acromal region of the other organism. Only capacitation sperm passes through the coronal radiator undergoes acromal reaction. Acromal region, which removes the upper layer of the acromal region. Write down the steps of gastrulation. Gastrulation begins with the formation of primitive stake on the surface of the epiblast. Cells of the epiblast migrate towards the primitive stake. Epiblast, which means the upper region of the stake, it becomes flux-shaped. Dash from epiblast and slip beneath this removal is beneath it. This inward movement is known as invagination. In primitive stake, invagination. Flux-shaped invagination. From epiblast, flux-shaped invagination. Once the cells have invaginated, some displace the hypoblast, creating an embryonic endoderm. Some displace the hypoblast. Others come to lie beneath the epiblast newly revealed endoderm to form mesoderm. It is a phase of embryonic development where the two layers become where the epiblast and the hypoblast. From 2 layers become 3 layers, the endoderm, mesoderm and the ectoderm. By invagination of cells through the primitive stake. Write down the 4 derivatives of neural crest: connective tissue and bone of the skull, cranial nerve and ganglia, C cell of thyroid gland, conostrosal sperm of the heart, chondroblasts, dermis of the face and the neck, spinal ganglia, sympathetic chain and prothetic ganglia, parasympathetic ganglia and gastrointestinal tract, adrenal medulla, Schwann cells, messengers, melanocytes, smooth muscle cells are for brain. Write down the sarcophageal tetramer. Write down the 4 derivatives of neural crest cells. Write down the 4 derivatives of neural crest cells. They are the odontoblast, the dermis of the head and the back, the spine, odontoblast, spine, Schwann cells, ganglia cells. Write about the sacrocegeal teratoma. Sometimes remnants of the primitive stake persist in the sacrococcygeal region. These crusts of Pluripotent cells proliferate and form tumors known as sacrococcygeal tetrachomas, which commonly contain tissue derived from the three germ layers. This is the most common tumor in newborns. Tetrazoma may also arise from perimodal germ cells that fail to migrate to the gonadal ridge. So, what is sacrococcygeal tetrazoma? Sacrococcygeal tetraroma is the most common type of tumor among newborns due to the remnants of primitive strake. Give the process of formation of notochord with diagram. Write down the functions. Formation of notochord. Successive steps in development of notochord. Primitive formation of primitive node. The cephalic end of the primitive stake is thickened to form the primitive node. The center of primitive node presents a depression known as primitive pit. Formation of notochordal process. Pre-notochordal cells invaginate in the primitive pit, move forward cranially. Formation of primitive node. The cephalic end of the primitive stake is thickened to form a primitive node. The cephalic end of the primitive stake is thickened to form the primitive node. The center of the primitive node presents a depression known as primitive pit. Formation of notochordal process. Pre-notochordal cells invaginating in the primitive pit move forward cranially until they reach the precondylar plate. These cells form a tube-like solid process known as notochordal process. Formation of notochordal canal, formation of notochordal plane, and formation of infinitive notochord. The primitive pit extends into the notochordal crest, forming the notochordal canal. The floor of the canal fuses with the underlying endoderm. Forming of the notochordal plane. Gradually, the floor of the notochordal canal disappears. The remaining portion of the notochordal process forms notochordal plane. Formation of definitive notochord. The cells of the notochordal plane proliferate and unfold, form a solid cord, which is called definitive notochord. Functions of notochord. Structural. They serve as the basis of axial skeleton. It forms the foundation upon which vertebral column and induces the formation of neural tube. So, the process of formation notochord is primitive node formation notochordal process. Primitive node notochordal process, formation notochordal canal, notochordal plane, and formation of definitive notochord. So primitive and definitive. Primitive and then definitive. Primitive, definitive. Notochordal process, formation notochordal process and notochordal canal, notochordal plane. So primitive, definitive, canal, plane, primitive, definitive, canal, plate, and then process. Primitive, definitive, canal, plate, and process. Primitive, definitive, canal, plate, process. Primitive, definitive, canal, plate, process. Derivatives of the ectoderm, derivatives on the endoderm, and derivatives of the mesoderm, derivatives of the endoderm. The derivatives of the ectoderm include surface ectoderm and neural ectoderm. Surface ectoderm and then neural ectoderm. Surface ectoderm, neuroectoderm. Surface ectoderm, neuroectoderm. And the primitives of the mesoderm include paraxial mesoderm, and then intermediate mesoderm, and then lateral plate mesoderm. Derivatives of endoderm include gut tube endoderm, cloaca endoderm, and then pharyngeal endoderm, yolk sac, and then allantois endoderm. So there are ectoderm, mesoderm, and endoderm. The ectoderm has surface ectoderm and neuroectoderm. The mesoderm has paraaxial mesoderm, intermediate mesoderm, and lateral plate mesoderm. Lateral plate mesoderm, and then the endoderm has yolk sac, gut tube, foregut, hindgut, and allantois. Surface ectoderm has epithelial lining, glands, and others. Neuroectoderm has neural crest and neural tube. Paraaxial has dermatom, myotom. Intermediate ectoderm, mesoderm has urinary bladder. Lateral plate mesoderm has splagnopleuric and somatopleuric. Yolk sac has a embryonic blood, GIT tract, and thymus, rectum, and urinary bladder. And then the urinary crest has swan cell, meninges, PNS, CNS, etc. And the somatopleuric is splagnopleuric. The somatopleuric has the complexes, gonadal complex, adrenal gonadal complex, and the splagnopleuric has the peritoneum, pleura, and visceral layer of the peritoneum, pleura, and pericardium. Somatopleuric has viscera. Somatopleuric has the visceral, splagnopleuric has the viscera of the peritoneum, viscera of the peritoneum, pericardium, and pleura. Splagnopleuric has the gonadal complex. Second week development is called the week of 2. The second week development is known as the week of 2 because the trophoblast differentiate into two layers. Cytotrophoblast and syncytotrophoblast. Cytotrophoblast and syncytotrophoblast. The embryoblast from two layers. Epiblast and hypoblast. The extraembryonic mesoderm splits into two layers, somatopleuric and splagnopleuric, the extraembryonic layer. So there is the trophoblast dividing into cytotrophoblast and syncytotrophoblast, embryoblast dividing into epiblast and hypoblast, and extraembryonic mesoderm divided into somatopleuric and splagnopleuric. Two cavities, amniotic cavity and the yolk cavity, and two cavities, amniotic cavity and the yolk sac. So basically the second week is known as the week of the 2 because the trophoblast divides into cytotrophoblast and syncytotrophoblast. The embryoblast divides into hypoblast and epiblast, and the two cavities are from yolk sac and amniotic fluid cavity, and then the extra embryonic mesoderm divides into somatopleuric and splagnopleuric layer. Formation of chronic phrognostom development. Briefly discuss on the formation of placenta. It has the formation of decidual basalis and the formation of chorionic frondosome, and formation of decidua basalis. Write the structure of full-term placenta. Structures of full-term placenta. At full term, the placenta is discoid with a diameter of 15 to 25 cm, is approximately 3 cm thick, and weighs about 500 to 600 grams. At birth, it turns from the uterine wall. After birth, when placenta is viewed from the maternal side, 15 to 20 slightly bulging areas known as cytoledons covered by the thin layer of decidua basalis are clearly recognizable. Decidua basalis. Grooves between the cytoledons are formed by decidual septa. The fetal surface of the placenta is covered entirely by the chorionic plane. The fetal surface of the placenta is covered entirely by the chorionic plane. The fetal surface of the chorionic plane. Chorionic frondosome. Chorionic frondosome chorionic frontosome and decidua basalis. chorionic frontosome decidua basalis. Chorionic basalis তাহলে কি chorionic plate. All numbers of large arteries and veins, the chorionic vessels. A number of large arteries and veins, the chorionic vessels, cover between the abdominal cord, the end, and chorion in turn is covered by amnion. Chorion in turn is covered by amnion. Chorion amnion. Chorionic frontosome. Decidua basalis. Villi, secondary stem, villi. Attachment of the umbilical cord usually ancentric and axonally, even marginal. Rarely, however, does it insert into the chorionic membrane inside the placenta. Function of placenta: exchange gases O2 and from the mother to fetus, O2 from mother, and then exchange of nutrition and electrolytes, transmission of maternal antibodies, providing fetus and passive immunity. Production of hormones: human chorionic hormone, human chorionic gonadotropic hormone, somatomamotropin hormone, progesterone, estrogen, and estradiol. Detoxification of some drugs, exchange of metabolic waste products between fetus and mother. Name abnormal placenta. According to implantation: placenta praevia, placenta parietalis, placenta praevia, placenta parietalis. According to attachment of umbilical cord: battle-born placenta, velamentous placenta, battle-born placenta, velamentous placenta. According to shape: lobed placenta, placenta ambrosia, and circumvallate. According to number: bilobe, trilobe, placenta succinata. According to degree of adhesion: placenta acernata, placenta insenata, placenta prasinata. According to the distribution of the umbilical arteries: dispersed type, magistral type. Fetal membrane: certain extraembryonic membranes which provide nutrition and protection and support the embryo of the fetus are collectively called the fetal membrane. Names of the fetus membrane are amnion, chorion, yolk sac, allantois. Umbilical cord, connective tissue, and palatine duct. So, amnion, chorion, yolk sac, and allantois. Amnion, chorion, yolk sac, allantois. Amnion, chorion, yolk sac, allantois. Write the difference between monozygotic and dizygotic twin. Fertilization of one ovum by fertilization from the zygote. Fertilization stimulates shedding and two oocytes and fertilization different spermatozoa. Sex is same. Sex may or may not be same. Dizygotic twin, monodizygotic twin. Placenta amnion and other chronic sac resulting in twin having separate common or placenta separate or common placenta. Normally there is separate placenta, amnion, chorion, and usually are resulting twins. Resemblance between the twins: having string resemblance in blood groups, fingerprints, sex and external appearance, eye color and hair color. Normally there is no similarities. Dizygotic twin: when two ova are discharged at the same time in a single ovarian cycle, either from the Graafian follicle or from two separate follicles, either from one Graafian follicle or two separate follicles, and are fertilized by two different sperms, and the resulting twins are fertile and unlike conveying different genetic materials. They are called zygotic twin. Amnion, chorion, placenta. Characteristics: different sex, no similarity in physico-mental character. Draw and label the boundaries of contents of typical intercostal space. Boundary: above, lower border of the upper rib, below, lower border of the lower rib. Draw and label. Describe the interior of the right atrium. Anterior of the right atrium. The anterior of the right consists of posterior part known as sinus venorum. Posterior smooth part known as sinus venorum. So basically, the heart has the heart can be divided into two parts: anterior rough part and a posterior smooth part. The anterior rough part, also known as atrium proper, which develops from the primitive atrium, and the posterior smooth part is developed from the right horn of the sinus venosus. Sinus venosus, or the sinus venerum. Sinus venerum. Sinus venerum. Sinus venerum. The posterior smooth part has superior vena cava, inferior vena cava, opening of the vena cordis minimi, and then the opening of the coronary sinus and has a intervenous tubercle. Intervenous tubercle. The rough anterior part has musculi pectinati. So the heart basically has an anterior rough part and a posterior smooth part. The anterior rough part is also known as atrium proper, which develops from the primitive atrium. It has the musculi pectinati and the crista terminalis, which corresponds to the interior part of the sulcus terminalis, and then it also has the reticular network of the auricle. And then the smooth anterior part has some major openings: the superior vena cava and the inferior vena cava, the vena cordis minimi, the intervenous tubercle, and the opening of the coronary sinus. And the septal wall has the triangle of Koch, and then fossa ovalis, annulus ovalis, triangle of Koch, and then torus aorticus, and then remains of the foramen. The conducting system of the heart is made out of myocardium, that is specialized for initiating and conduction of a cardiac impulse. The conducting system has the following parts: sinoatrial node or SA node. It is known as the pacemaker of the heart. It generates impulses of rate of about 70 to 100 beats a minute and initiates the heartbeat. It is a horseshoe shape. It is situated on the atriocaval junction in the upper part of the sulcus terminalis. The impulse travels through the atrial wall to reach the AV node, atrioventricular node. It is smaller than the SA node and is situated in the lower and the dorsal part of the atrial septum, just above the opening of the coronary sinus. Atrioventricular node is a smaller node. It is capable of generating impulses of 30 to 40 beats per minute, 70 to 100 beats, 40 to 100 beats per minute. The atrioventricular bundle or the AV bundle or the bundle of His. AV bundle or the bundle of His. It is the only muscular connection between the atrial and the ventricular musculatures. It begins at the atrioventricular AV node, crosses the AV ring and descends along the posterior inferior border of the membranous part of the ventral septum. The atrioventricular bundle or the AV bundle or the bundle of His is the only muscular connection between the atrial and the ventricular musculature. It begins at the atrioventricular node, crosses AV ring and descends along the posterior inferior border of the membranous part of the ventricular septum. At the upper border of the muscular part of the septum, it divides into right and left branches. Right branch of the atrioventricular bundle passes down the right side of the atrioventricular septum. A large part enters into the mandatory band to reach the anterior wall and the right ventricle, where it divides into Purkinje fibers. The left branch of the atrioventricular bundle descends on the left side of the atrioventricular septum and distributed to the left ventricle after divided into Purkinje fiber. The right Purkinje fiber form a subendothelial plexus. They are large pale fibers strained only at their margins. They usually possess double nuclei, Purkinje fibers. This generates impulses at the rate of 20 to 35 beats per minute. Blood supply. Whole of conductive system except the left branch of epibundle is supplied by right coronary artery. In 40% of cases, left coronary artery supplies SNO. The whole conducting system except the left branch of the atrioventricle is supplied by the right coronary artery, except for the left branch of the coronary and left coronary artery. In 40% of cases, left coronary artery supplies SA node. So the sinoatrial node, atrioventricular node, sinoatrial node, bundle of his, right branch of epibundle, left branch epibundle, and then Purkinje fiber. Right branch of epibundle includes the marginal band, and left includes the septum, interventricular septum. Now the interventricular septum large moderator band reaches the anterior wall of the interventricular Purkinje fiber. So sinoatrial node, atrioventricular node, bundle of his, right atrioventricular bundle, and left atrioventricular bundle. Right is moderator bundle and left bundle is septum. Then Purkinje fibers. Draw and label the histological features of cardiac muscle. Cardiac muscle nucleus, cardiac muscle fiber, cardiac muscle anterior internodal branch, middle internodal branch, posterior internodal branch. And then cardiac muscle is made out of the elongated fibers. Within the cardiac muscle is made up of elongated fibers within which numerous microtubules are present. The individual muscle cells join end to end by intercalated discs. Nuclei of the myocytes and placed centrally. Nuclei of the myocytes are placed centrally. Cardiac muscle made out of elongated fibers with numerous fibers present. In the muscle cell join the end of the interventricular disc. Sarcoplasm contains myofibrils. The myofibrils show alternate dark and light bands. This accounts for cross striations of cardiac muscle fibers. Question: draw and label the histological muscle features of muscular artery. The features of muscular arteries consist of three layers: tunica intima, tunica media, and tunica adventitia. Tunica intima, tunica media, and tunica adventitia. Muscular arteries of three layers. Tunica intima, made of endothelium and interstitial elastic lamina, no subendothelium. The interstitial elastic lamina is bright reflectile membrane flow into the field due to contraction of smooth muscle of the media. Tunica media. It consists mainly of the smooth muscle cells arranged circularly, about 40 layers, hence the name 40 layers. It is also contains elastic and few collagen fibers intermixed with smooth muscle. Tunica adventitia. The inner part of the tunica adventitia contains more elastic than collagen fibers, and it includes the external elastic lamina. The medial part contains collagen and elastic fibers running longitudinally. The outer part is made out of loose connective tissue that merges with the surrounding areolar tissue. Consists of endothelium and the internal elastic lamina. Tunica media consists of smooth muscle arranged circularly. And tunica adventitia is the inner part of the adventitia, contains more elastic collagen fiber and includes the external elastic lamina. Middle part of the contains collagen with elastic fibers running longitudinally. The outer part is made out of loose connective tissue, merges with the surrounding areolar tissue. Development of the right atrium, sinus venorum. It is developed from the reabsorbed right horn of the sinus venosus. Atrium proper, rough part, develops from the right part of the primitive atrium. Cister terminalis is a remnant of the upper part of the right venous valve. Upper part of the right venous valve. Remnant of right venous valve. Valves of the inferior vena cava and coronary sinus are derived from the lower part of the right venous valve. Right venous valve. Upper part of the right venous valve. Upper part of the right venous valve. And inferior vena cava and coronary sinus are the lower part of the right venous valve. Upper part of the right venous valve. Upper part of the right venous valve. The floor of the fossa ovalis, right venous valve. Right valve. The floor of the fossa ovalis is formed by the septum primum and the annular fossa. Ovalis fossa annulus fossa is developed from the remnant of lower margin of the septum sagentum. Septum primum and septum sagentum give the boundaries, formation and vertebral relation of the base of the heart. Base of the heart. The posterior surface of the heart is called the base of the heart. It is the most fixed part of the heart and is actually anatomically based. It is actually anatomically based. Boundaries. Above, bifurcation of the pulmonary trunk separated by transverse pericardial sinus. Above, bifurcation of the pulmonary trunk, transverse pericardial sinus. Transverse pericardial sinus. And then below, posterior part of the atrioventricular groove containing coronary sinus and anastomosis of the right and the left coronary arteries. Below, posterior part of the atrioventricular groove. Posterior of the atrioventricular groove containing coronary sinus and anastomosis of the right and the left coronary arteries. Left coronary arteries. Right and the left coronary arteries. Coronary arteries below. Right side. Right border of the extending from the superior and the inferior vena cava orifice. Left side. Left border of the atrium. Left border of the extending from upper to lower pulmonary veins. Formation. This base of the heart is formed by the two atria, 2 to 3 by left atrium, 1 to 3 by right atrium. Vertebral relations. Between the base of the heart, the vertebral column of the following structures intervene. Oblique sinus, posterior part of the fibrous pericardium, right part of the pulmonary veins. So, oblique sinus and then posterior part of the fibrous pericardium. Posterior part of the fibrous pericardium, rough pair of the pulmonary veins. Right and the left bronchi, esophagus with plexus of nerves, ascending thoracic aorta thoracic duct, descending thoracic aorta thoracic duct. Right and left bronchi, esophagus with plexus of nerves, descending thoracic aorta descending thoracic duct, azygous and hemizygous veins, great and the lesser splanchnic nerves, posterior and mediastinal lymph nodes. Vertebral levels. It is the recumbent spine position lies in bodies of the 5th to 8th thoracic vertebra. In the standing position it lies opposite to the 6th to 9th vertebra. Right, the support of atrial support of the heart. Fibrous structures of the heart give support to the heart. Fibrous structures consist of fibrous rings and fibrous septum. Oblique sinus, posterior part of the fibrous pericardium, right pair of the pulmonary veins. Support of the heart. Fibrous skeleton of the heart gives fibrous the heart. Fibrous skeleton of the heart consists of 4 fibrous rings and 3 fibrous septum. Fibrous rings. Pulmonary rings around the pulmonary orifice. Aortic ring around the aortic orifice. The right atrioventricular ring around the tricuspid valve. Left atrioventricular ring on the bicuspid valve. Fibrous septum. Tendon of infundibulum. Fibrosum tendorum infundibulum. Fibrosum tendorum dextrum. Fibrosum tendorum sinistrum. The pulmonary ring of the aortic ring are connected by the fibrous septum known as the tendon infundibulum. Bones of the thorax. The bones of the thorax form major part of the thoracic cage and provide support and protection to viscera, lung and the pleura, heart and the lungs. Present within the thoracic cavity, the thoracic cage is not static in nature but dynamic as it keeps on moving in various joints. The joints of the thorax are sternum, the 12 pairs of ribs, the 12 thoracic vertebra. Sternum. The sternum is an elongated flat bone, lies in the anterior medial part of the chest wall. It is about 17 centimeters long. Parts. The sternum consists of the following three parts. Upper part, the manubrium sterni, middle part, the mesosternum, lower part, the xiphoid process or the metasternum. Upper part, middle part and the lower part. Episternum, mesosternum, metasternum. The sternum resembles a dagger or a small sword in shape in three parts. Manubrium, body and xiphoid represents the handle, blade and the point of the sword respectively. The upper part of the sternum is broad and thick, whereas the lower part of the sternum is thin and pointed. The anterior surface is slightly rough and convex. The anterior surface is slightly rough and convex. Slightly rough and convex. Slightly rough and convex. Slightly rough and convex. The sternum resembles a dagger. Upper part is slightly rough and convex. The posterior surface is smooth and slightly concave. The manubrium and the body of the sternum lie at an angle. The manubrium and the body of the sternum at a 160 of each, which increases the duration of inspiration during an expiration. The angle between the long axis of the manubrium and the long axis of the sternum is about 17 degree. Anatomical position. In anatomical position, the sternum as whole is directed downwards and inclined slightly forward with a rough convex surface facing interiorly. The upper border of the upper end is directed upward and is pointed lower directed downward. Features and attachments. Manubrium or episternum, the rough quadrilateral shape. It lies opposite to the third and the fourth intercostal vertebra. It is the thickest and the strongest part of the sternum and presents the following features: two surfaces, anterior and posterior, four borders, superior, inferior and the lateral. The anterior surface of each side provides attachment to the sternal head of the sternomastoid. The anterior surface of each provides attachment to the sternal head of the sternocleidomastoid and the pectoralis major muscles. The sternocleidomastoid and the pectoralis major muscle. The posterior surface is smooth and forms the anterior boundary of the posterior superior mediastinum. The posterior surface is smooth and forms the anterior boundary of the posterior mediastinum. On each side, it provides attachment to the two muscles: the sternohyoid muscle, the level of the clavicle notch, and the sternothyroid muscle, the level of the facet. The lower half is related to the arch of the aorta. Lower arch of the aorta, sternohyoid and sternothyroid. And the superior surface attaches to the sternocleidomastoid and pectoralis major muscle. Sternocleidomastoid, pectoralis major muscle. Lower half is related to the arch of the aorta. The upper half is related to the three branches of the arch of the aorta, namely branchioocephalic artery, the left common carotid artery, and the left subclavian artery. The left branchioocephalic vein.
The sternum is varies greatly in size and shape. It may be bifid or perforated. Its anterior surface provides the insertion of the medial fiber of the rectus abdominis muscle, and its posterior surface gives origin to the
Diaphragm.
external fibers of the diaphragm. Its tip provides attachment to the upper end of the linea alba. Muscle attached to the posterior and the anterior surface of the sternum are summarized below.
Sternohyoid, sternothyroid, and pectoralis major, sternocostalis. Anterior surface and posterior surface. Sternohyoid, sternothyroid. Sternohyoid, sternothyroid. Sternohyoid, sternothyroid. Sternocostalis, diaphragm. Sternohyoid, sternothyroid. Pectoralis major, rectus abdominis. External angle. External angle can be felt as a transverse ridge on the sternum, about 5 centimeters below the supraesternal notch. The sternal angle is an important surface bony landmark of many external events that occur in this level. These are: the second costal cartilage articulates with this level, which external level, hence this level is for counting the ribs. The second costal cartilage articulates. Second costal cartilage articulates. It lies at the level of the level of the intervertebral disc between the thoracic core to thoracic vertebrae. It lies at the level of the intervertebral disc between the thoracic core to thoracic 5 vertebrae. Horizontal plane passes through this level, separates the superior mediastinum from the inferior mediastinum. Ascending aorta ends at this level. Arch of the aorta begins and ends at this level. Descending aorta begins at this level. Descending aorta. Trachea bifurcation into the right and the left principal bronchi at this level. Trachea bifurcation. Our upper border of the heart lies at this level. Azagus veins arches over the root of the lung. And ends in the superior vena cava. Azagus arches over the root of the lung to end in the superior vena cava. Azagus vein. Clinical correlation. Puncture.
Manubrium strani is the performed site of the bone marrow aspiration because it is subcutaneously and readily acceptable.
The bone marrow sample is required to a hematological examination. A thick needle is inserted into the upper part of the manubrium to avoid injury of the aorta. It lies behind the lower part. External puncture is not advisable for children because in the plates of the backbone and the sternum are very thin. If needle pierces through the manubrium, it will damage the aorta and its branches, leading to fatal hemorrhage. Mid-sternoctomy. The gain access of the mediastinum for surgical operations on heart on the great blood vessels for the sternum is often divided into the medial plane, called the mid-sternotomy. Funnel chest. It is an abnormal shape of the thoracic cage in which the chest is compressed posteriorly and the sternum is pushed backwards by the overgrowth of the ribs, maybe compression of the heart. Pigeon chest. It is an abnormal shape of the thoracic cage in which the chest is compressed from side to side by the sternum, projects forward and downward near of a boat. Sternal fracture. It is a common automobile accident. For example, when the driver's chest hits against the steering wheel, the sternum is often fractured, sternal angle, and backwards. Displacement of the fracture may be damage aorta, heart, and the liver, and cause severe bleeding to prove fatal. Ossification. The sternum develops from the two vertical collagen plates of sternal plates that fuse in the midline. The sternum occupies 6 double centers, namely 1 in the manubrium, 4 for the body, 4 of each sternobry, 4 for the middle of each sternobry, 1 for the xiphoid process. So 6, 4 for the body and 1 for the manubrium. Body. The first 6, the second sternum body. The first sternibra 6 months, the second sternibra 7 months, third sternibra 8 months, the fourth sternibra 9 months, 7, 8, 9. Xiphoid process 3rd year. Manubrium 5th month. Manubrium 5th month. Appearance. The centers appear in descending order to different parts of the sternum mentioned above. Fusion. The fusion occurs as follows. Fusion between the sternal plate takes place below upward. It begins at the puberty and completed at level 25 years. The xiphoid process fuses the body at the age of 40 years. Manubria does not fuse at the body. As a result, the secondary cartilaginous membrane joints usually persist throughout life. About 10% of individual fusion occurs in old age. Does not fuse with the body. As a result, the secondary cartilaginous manubrium usually persists throughout life. Secondary cartilaginous manubrium. also old age, sternal foramen and cleft sternum. The two sternal plates fuse in the caudocranial direction. Sometimes sternal fails to fuse with the midline. As a result, defect occurs in the body of the sternum in the form of sternal foramen of cleft sternum. The cleft sternum is often associated with ectopic cordis. Ribs.
Ribs are flat. ওটা কি লং বোন কিন্তু স্টার্নাম হচ্ছে লং বোন, এটা ফ্ল্যাট বোন। Ribs are flat ribbon-like elastic bony axis that extended from the thoracic vertebra posteriorly to the lateral border of the sternum anteriorly. Their anterior ends are connected to the costal cartilage. The ribs along with its costal cartilage constitutes the costa. The ribs and their costal cartilage.
From the gather of the thoracic skeleton.
But the occurs are accessory cervical and lumbar rib. May increase them to the 13 pairs of the
or the absence of
12 rib may reduce them to 8 pairs. Attachment of the general origin. The ribs are arranged on below the other, and the gap between the adjacent are called the intestinal space. Intestinal space কি?
The length of the ribs increases from the first to the seventh, and generally increases. Hence, seventh costal cartilage, the seventh rib is the longest rib. The transverse diameter of the thoracic increases progressively up to the first to the eighth rib. Hence, the eighth rib is the greatest lateral projection. Therefore, the ribs are arranged obliquely. That is, their anterior end is lies at the lower border and the posterior end. Obliquely, their anterior end is lies at the lower level, the posterior end. The oblique of the ribs increases progressively from the first to the ninth rib. Ninth rib is almost obliquely placed. The right ribs supply দিলে শুয়ে রাখি তোমার gradually reduce.
একটা ইয়ে দিয়ে দিই।
তাড়াতাড়ি শেষ করো এটা। ওরা এটা দেখা নাই তাড়াতাড়ি দাও। The weight of the rib gradually reduces from above the
এখন যা বলবো সব
আচ্ছা তো টাকা এরপরে reduces above from the downwards. এরপরে the anterior end of the seventh apex of the lung. It corresponds to the outline of the cervical pleura. It corresponds to the outline of the cervical pleura. It is identical with the line of the costo-mediastinal reflex of the right pleural sac. The line commences the point which corresponds with the summit of the apex of lung. Therefore, the point of drawing the anterior border as follows: 2.5 centimeters above the middle clavicular at the mid-clavicular point. 2.5 centimeters right clavicle at the midpoint between the medial one-third. Midpoint at the medial one-third. Midpoint at the medial one-third. Midpoint at the medial one-third. Medial one-third end of the clavicle. Sternal end of the clavicle. A point at the right sternoclavicular joint. A point at the right sternoclavicular joint. A point in the middle sternal angle. Middle of the sternal angle. A point on the G1 joint. A point on the G1 joint. 2.5 centimeters above the left. 2.5 centimeters above the left auricle at the middle point between the medial one-third. A point at 2.5 centimeters above the left clavicle. 2.5 centimeters above the left clavicle. 2.5 centimeters above the left clavicle at the midpoint between the middle one-third of the sternal angle. Midpoint and the middle one-third midpoint and the middle one-third of the sternal end and the clavicle. A point at the left sternoclavicular joint. Left sternoclavicular joint at the middle of the sternal angle at the level of the fourth costal cartilage. At the level of the fourth costal cartilage, the midpoint, midplane, at the level of the fourth costal cartilage. A point in the fourth costal cartilage, 3.5 centimeters away from the sternum. 3.5 centimeters from the sternal angle and the fourth costal cartilage. A point in the sixth costal cartilage, 4 centimeters from the medial plane. 4 centimeters from the medial plane. Join the 4 points of the line which corresponds with the left costo-mediastinal reflection of the pleura. Lung. So 4 and 6, 3.5 centimeters and 4 centimeters from the medial plane. Lower border of the lung. The lower border of the lung, the 6th costal cartilage, a point in the mid-xiphoid process. Xiphoid process on the 6th costal cartilage, a point at the xiphoesternal joint. A point at the xiphoesternal joint on the left 6th costal cartilage, 4 centimeters away from the left lung, 4 away from the medial plane for the left lung, 4 centimeters away from the medial plane, xiphoesternal joint, a point at the xiphoesternal joint, the 6th xiphoesternal joint, and for the left lung 6th costal cartilage, 4 centimeters away from the 6th costal cartilage, a point in the midclavicular line, 6 ribs midclavicular line, and then 8 ribs midaxillary line, midclavicular line, midaxillary line, midaxillary 6 ribs, 8 ribs midaxillary line, midclavicular line, midaxillary line, 2 centimeters lateral from the 10th thoracic spine. The represented by a vertebral line drawn from the point 2 centimeters away from the 7th and 10th cervical spine. A point 2 centimeters lateral to the 2nd thoracic spine, 3 centimeters away from the level of the nipple. Transverse tissues of the lung. A point at the medial plane of the 4th costal cartilage. Transverse line along the 4th costal cartilage up to the midaxillary line where it meets the optic fissure. Mark of the 4th, 5th, 6th thoracic spine by root of the lung. A point on the upper border of the right 3rd costal cartilage. A point on the upper border of the right 3rd costal cartilage. And then 3.5 centimeters from the medial plane, 4th intercostal space. 4th intercostal space. And then right 6 intercostal close to the sternum. So 3rd, 4th and 6th. A point in the lower end of the right border of the 6th costal cartilage, close to the sternum, 6th costal cartilage. 5th intercostal space and a line joining the above the point of the representation border. A point 2.5 away from the 2nd costal cartilage. A point in the apex beat. Aortic orifice of the heart. Pulmonary orifice. Represent a horizontal 2 centimeters long along the upper border of the 3rd costal cartilage. Upper border of the 3rd costal cartilage. Left 3rd costal cartilage. On the left 3rd costal cartilage, aortic orifice. 4 centimeters long in the mid plane of the 4th costal cartilage. Third costal cartilage. 4th costal cartilage. Point of the right extremity of the sternal angle, point of the manubrium externi, and then 2nd costal cartilage, and in the 2nd costal cartilage. Descending thoracic aorta. Esophagus. The entire esophagus can be projected, the making a point left of the 7th costal cartilage, a point on the left on the 7th costal on the medial plane, a point on the 7th light 7 2.5 on the medial plane slightly below the sternal angle, cricoid cartilage 7 costal cartilage, thoracic duct 2.5 centimeters above the trapezoid plane and slightly right to the medial plane. Above the trapezoidal plane, slightly above the and slightly right to the medial plane, right to the medial plane, slightly right to the medial plane, center of the understanding. A point 2.5 centimeters above the left clavicle. Define biochemistry. Biochemistry can be defined as the science concerned with the chemical basis of life. GK Bio Life. Biochemistry can be defined as the science concerned with the study of the chemical constitutes of living cells and the reactions and process they undergo. By this definition, biochemistry encompasses large areas of cell biology, molecular biology, and molecular genetics. Aim of biochemistry. The major objectives of biochemistry is the complete understanding at the molecular level of all the chemical process associated with living cells. Knowledge of biochemistry is essential to medical science. Biochemistry is the most rapid developing and most innovative science subject in medicine. Normal biochemistry process the basis of health. Most and perhaps all diseases have a biochemical basis. Most bases are manifestations of abnormalities in biomolecules, biochemical reactions, and biochemical processes. Many biochemical studies illuminate disease mechanisms. Biochemical studies contribute to diagnosis, prognosis, and treatment. Insulin, interferon, growth hormone, etc., obtained from genetic engineering, are used largely in the treatment of different diseases. Biochemical research has impacted on the nutrition of preventive medicine. Physiology, the study of body function, overlaps the biochemistry almost completely. Immunology employs the numerous biochemical techniques, and many immunological approaches have found wide use by biochemists. Pharmacology and pharmacy rest on a sound knowledge of biochemistry and physiology. In particular, most drugs are metabolized in enzyme catalyzed reactions. Poisons act on biochemical reactions or process. This is the subject matter of toxicology. Biochemical approaches are being used increasingly to study basic aspects of pathology, the study of disease, such as inflammation, cell injury, and cancer. Many workers in microbiology employ biochemical approaches almost exclusively. These relationships are not surprising because life as we know it depends on biochemical reactions and process. In fact, the old barriers among the life sciences are broken down, and biochemistry is increasingly becoming their common language. Nucleic acid genetic disease, protein sickle cell anemia, biochemistry medicine, lipid atherosclerosis, carbohydrate diabetes mellitus. Explain the importance of biochemical investigations in part of medicine. Importance of biochemical investigations in the practice of medicine. We believe that most of the diseases are manifestations of abnormalities of molecules, chemical reactions, or biochemical processes. In most of the diseases conditions, biochemical studies contribute to both the diagnosis and treatment. Some major use of biochemical investigations and of laboratory tests in relation to disease are summarized in the table below. Use: confirm diagnosis of a specific disease. Use of plasma cardiac troponin 1 level in diagnosis of myocardial infarction. Plasma cardiac troponin 1 suggest rational treatment of disease. In elevated low density lipoprotein, there is an increase of therapy with lipid lowering drugs, for example, statins, in persons at risk of cardiovascular disease. Use as screening test for early diagnosis of certain diseases. Measurement of thyroid stimulating hormone levels in neonates helps in diagnosis of congenital hypothyroidism. Assist in monitoring the progress of certain diseases. Serum ALT activity is used to monitor the process of viral hepatitis. Help in assessment of response of disease and therapy. In in patients with hypo or hyperthyroidism, measurement of thyroid stimulating hormone level helps in monitoring the response of patients to treat. Reveal the fundamental cause and mechanism of disease. Demonstrate the nature of the genetic defect in cystic fibrosis. Nice to know. Vital force theory. A life comes from previous life. It was believed for a long that the carbon compounds of organisms, hence the name organic. Carbon compound of organisms organic arose from life only. This is referred as the vital force theory. Most common organic components found in the living system: carbohydrates, lipids, proteins, nucleic acids, and vitamins. Define pH. pH is negative logarithm of hydrogen ion concentration in solution to the base of 10, where H concentration is expressed in molecular L. For example, 25-degree hydrogen concentration pure water is 10 to the power minus 7 mole per liter. pH equal to minus log 10 to the power minus 7 biomedical and chemical importance of pH. Maintenance of appropriate pH is necessary for homeostasis. pH controls the biomolecular reactions. It controls the enzyme activity. It controls the cellular viability. Maintenance of ECF pH between 7.35 and 7.45 is essential for health. Between 7.3 and 7.45 is essential for health. Blood pH is lower than 7.35 indicates acidosis, which occurs in diabetic ketoacidosis and lactic acidosis. pH greater than 7.45 indicates alkalosis that may occur in the vomiting of acidic gastric contents. Define acidic pH, alkaline pH, and natural pH. Acidic pH. The pH is less than 7 is called acidic pH. The pH is more than 7 is called alkaline pH. The pH, for example, pH of water is called natural pH. Define pOH. pOH is the negative logarithm of OH ion concentration in solution. It is useful in calculating the pH of alkaline solution. How is pH measured? Principal measurement of pH. The solution of strong electrolyte, strong acid, strong base. Hydrogen concentration is calculated. OH concentration is calculated. Strong acid, strong base. The base of 10 logarithm of hydrogen is calculated. pOH is calculated by following the equation pOH equal to minus log OH. pH is in the negative of the value found in step 2. pH equal to 14 minus pOH. The solution of weak solutes, weak acid and weak base. Hydrogen concentration is measured by the use dissociation constant. OH concentration is measured by the using of dissociation constant. pH is measured by the following question: pH equal to minus log H. pOH is calculated by the following equation: pOH equal to minus log OH. pH equal to minus 14 minus OPOH. For buffer solution, pH is measured by Henderson-Hasselbalch equation: pH equal to pK plus log A by HA. Methods of determination of pH: specific and non-specific. Specific: pH meter, glass electrode, calomere electrode, hydrogen electrode. Non-specific: buffer indicators, pH paper and litmus paper. Methods of determination of pH. What is the normal pH of blood? Mention the normal pH of urine. Blood 7.45 to 7.35. Range 7.35 to 7.45. Urine 6. Saliva 6. Milk 6.6. Tear 7.4. Pancreatic juice 8.3. ICF 7.2. Bile 7.8. Determine the pH of water. Determination of the pH of water. Idea from pH from law of mass action. Pure water dissociates only slightly and produces equal concentration of H and OH. OH H plus OH. According to the law of mass action, the dissociation constant of H for H2O is H equal to H OH by H2O. As H2O dissociates very slightly, it is essentially constant. H equal to H2O equal to HOH equal to constant, or Kw equal to HOH equal to Kw समान ion constant and of H2O. At 25 degrees, Kw 10 to the power minus 14 mole per liter square. So H and OH equal to 10 to the power minus mole per liter square. H square equal to 10 to the power minus 14 mole by liter square. H equal to 10 to the power minus 7 mole per liter. pH equal to minus log H1 minus log 10 to the power minus 7. So pH is water is minus 7. HOH 10 to the power minus 4. H2 10 to the power minus 4. H equal to 10 to the power minus 7. Define Kw. Ion production of water. Self-ionization of water. Ion production of water is equilibrium constant of the reaction where water dissociates into hydrogen and OH. Kw equal to 10 to the power minus 14 mole per liter square. Factors. Kw depends on temperature, pressure, ionic strength. Why water is standard for comparison of pH. Pure water has pH of 7, which is natural. It is taken as a standard for comparison of pH due to the following reasons. Water is natural and found universally. Water is dissociated very slightly. After dissociation, water produces equal amount of hydrogen ions and hydroxyl ions. Water is stable and do not change physically or chemically for a range of variations. pH scale. It is the arbitrary logarithmic scale of pH extending from 0 to 14, which covers the particular range of acidic and alkali in commonly available solutions. Construction. pH scale is constructed on the basis of ion production of water 10 to the power minus 14. pH scale is considered between 1 to 14 as the product of ion concentration of hydrogen and OH must not be exceed 10 to the power minus 14 in any solution. So the highest pH can only be 14. Salient features, properties. Range of pH scale to form 0 to 14. A solution with pH 7 is natural. pH 7 less than 7 is acidic. pH greater than 7 is basic or alkali. pH scale is logarithmic, not a numeric one. 1 unit of pH change is equivalent to 10 times the difference in H plus. For example, for the unit increased of pH will decrease 10 times and vice versa. 1 unit pH change in equivalent of 10 times difference will decrease 10 times and vice versa. 1 unit pH change in the equivalent of 10 times different OH. For example, the unit increased of pH will decrease 10 times and vice versa. Clinical significance. Acid-base balance is measured using the pH. What will be the pH of 0.1 and HCl? HCl is a strong acid. It dissociates completely in solution. So 0.1 NCl produces 0.1 NH. HCl equal to H plus Cl. 0.1 and 0.1 is 0.1 and H equal to 0.1 equal to 0.1 mole. pH equal to minus log H plus minus log 0.1 equal to 1. Find the function of a solution having concentration of 0.045 gram mole per liter. pH equal to minus log H, so minus log 0.00465 minus log 4.57 4.56 10 to the power minus 3 equal to minus log 4.56 plus log 10 to the power minus 3 equal to 0.006589 minus 10 minus plus minus 2.99 2.993. What is the pH of solution having H plus concentration of 3 10 to the power minus 2 mole per liter? If blood pH is 7.4, what will be the hydrogen concentration? NT log 0.6 equal to 3.94. pH equal to minus log H. Log H equal to minus pH. H equal to anti-log pH minus pH. Anti-log minus 7.4. Anti-log 8 minus 8 plus 0.7, antilog minus 8 into antilog 0.6, 10 to the power 8 into 3.98 mole per liter equal to 3.98 10 to the power minus 8 mole per liter. Optimum pH. The pH at which most physiological enzyme reactions occur is known as the optimum pH. It is the pH at which the rate of enzyme catalyzed reaction is maximized. Effects of temperature on pH. Dissociation of temperature equal to dissociation of 1 by pH. So pH equal to 1 by temperature. Temperature dissociation pH. The pH of a solution which is completely ionized will not vary with the temperature. Define buffer. Buffer is a maxi mixture of weak acids and its conjugate base which can resist the marked change of pH or H of a solution when moderated amount of acids and base are added to its solution. Buffer is a mixture of weak and acid salts of acid that tends to maintain constant hydrogen concentration when acid or alkali is added to it up to a certain limit. Conventionally, the buffer pair is symbolized as B conjugate base and HB weak base. Classification of buffer system of the body, plasma buffer, bicarbonate buffer, plasma protein buffer, and phosphate buffer. RBC buffers, bicarbonate buffer system, hydrocarbon hemoglobin buffer system, oxyhemoglobin buffer system, phosphate buffer system. RBC buffer, plasma buffer, RBC buffer, interstitial fluid buffers, bicarbonate buffer, protein buffer, phosphate buffer, intracellular buffer, protein buffer system, phosphate buffer, and bicarbonate buffer. Buffer acids through the respiratory system, HB buffer system, bicarbonate buffer system, buffer acting through the kidney system, phosphate buffer system, ammonia buffer system, and bicarbonate buffer system. Importance of buffer. Buffer regulates the buffers are bicarbonate buffer system, ammonia buffer system. Importance of buffer. Buffer regulates the neutrality of the body within a relatively narrow limit of pH. Compatibility of the life of more cells. Constant pH maintenance of the buffer is essential for optimal activity of enzymes. They are used in determination of pH of a solution with indicators. Control the pH in chemical reaction catalyzed by enzymes. They are used for making standard solution of constant pH. Buffers are used in lab to maintain the pH of culture media for growth of microorganisms and or tissues. Number of buffer system. Buffer system with chemical structure. Buffer contains 3 buffer systems: biocarbonate buffer system, phosphate buffer system, and protein buffer system. Major buffer system of extracellular fluid, active in intracellular fluid and renal tubular lumen, plasma proteins and globulin together with constitute the protein buffer system. Discuss the role of blood buffers in normal acid-base balance. Role of blood buffers in normal acid-base balance. Please see chapter acid-base and electrolyte balance in this book. Nice to know. Physiological buffers. These are the buffers present in the body and maintains the pH of the body fluid as a constant level despite additional or acidic or basic during normal metabolic activities in the body. Physiological buffers include biocarbonate buffer, protein buffer, and phosphate buffer. Name the 3 important body fluids buffer with the conjugate base. Important body fluid buffer with their conjugate base. Buffer system: weak acid, conjugate acid, bicarbonate, phosphate, and then protein, and then hemoglobin. Ammonia, also ammonia. State the basic mechanisms of buffer action. When the moderate amount of a strong acid or base is added in a buffer solution, the buffer prevents change of pH in the following ways. Buffer acts by converting strong acids into weak acids and strong base into weak base, by thereby minimizing the effort of strong acid and strong base on pH of solution. Weak acid or weak base undergo negligible dissociation. So negligible disturbance of hydrogen ion of solution. For example, a strong acid HCl and HOH3 NaCl and weak acid H2CO3. A strong base NaOH plus H2CO3 equal to NaHCO3 weak base and H2O. Buffer act by donating protons into the solution by the buffer acid. When there is proton deficiency, by removing proton from the solution by the buffer base when there is proton excess. Example, H2 H2CO3 and HCO3 equal to H plus HCO3, H plus H2CO3. There are two components of buffer: salt buffer, acid buffer. There is acidosis, acid low in the solution. There is acidosis or acid low in the solution. The salt part of buffer comes forward from action, and if there is alkalysis or alkali left in solution, the acid parts of buffer come forward from the converse of the neutral salt. Give the diagram the mode of action of hemoglobin and plasma protein buffer. How normal blood pH is maintained in a constant level. Maintenance of blood pH. The pH of blood is maintained at 7.4. There are three defense mechanisms by which this relative consistency of blood pH is maintained. Buffer system. It is the first line of defense which combines immediately with the second base or to prevent excessive change of pH. Respiratory system. It is the second line of defense which regulates removal of O2 and therefore H2CO3 from ECF. Buffer system, first line of defense, immediately within acid and base. Renal system. It is the third line of system, works within hours or days. Kidney excretes their acid or alkali urine, thereby readjusting with ECF or was normal during acidosis or alkalysis. Short note: buffer base. The buffer base BB is equal to a total number of buffer ions, principally both HCO3 and HB, that can accept H plus in blood. The normal value of buffer base in an individual within 50 grams of hemoglobin per deciliter of blood is 84 microliter. The normal value of buffer base in individual with 15 grams of hemoglobin per gram per dl. Buffer base. The buffer base dB is equal to total number of buffer ions accepted by the blood. The buffering capacity of a solution. Buffering capacity of a solution is defined as the number of moles of acid or base which must be added to a molar solution in order to change the pH of that by 1 unit. In the amount of a strong acid or base added to 1 liter of buffer system with pH change by 1 unit, the more the amount added, the more is buffering capacity. Factors affecting buffering capacity. Buffering capacity depends on the total quantity of buffering components. The more the components of the more buffering capacity. Base to acid ratio buffer system. Buffering capacity will be more with this ratio close to 1.0. What is the total buffering capacity? Total buffering capacity, the amount of acid or alkali that body can tolerate without significant change of pH, is called the total buffering capacity. Normally in an adult person, the total buffering capacity is 1000 millimole H plus. Open buffer system. CO2 bicarbonate buffer system is an open buffer system because the bicarbonate and H2O components are under physiological control, reacting to change in hydrogen concentration. They cannot be saturated. Closed buffer system. Closed buffer system are those which can be saturated. For example, hemoglobin buffer system and red cells and hydroxylated buffer in bone. Done. Define pK. Short note: pK is the negative logarithm of dissociation of constant K. pK equal to log K. pK is the body buffers: bicarbonate buffer, phosphate buffer, amino buffer, protein buffer, hemoglobin buffer, oxyhemoglobin buffer. Define pK. It is the negative logarithm of dissociation of constant of acid. pK equal to minus 40 log K. It is the negative logarithm of dissociation of constant of basic. pK equal to minus log Kb. It is the negative logarithm of dissociation of constant of acid. What is the role of HCO3 in maintaining the normal blood pH? Bicarbonate buffer. HCO3 buffer. HCO3 minus and H2CO3 is the most prominent buffer system in the extracellular fluid, ECF, particularly in plasma. It is an ideal buffer having wide field of activity. In normal circumstances, the concentration of bicarbonate and carbonic acids determine the pH of blood. Further, the bicarbonate buffer system serves as an index to understand the disturbance in acid-base balance of the body. Component NaHCO3 and H2CO3. pK value 6.1. Action. Bicarbonate HCO3 minus is blood buffer as well as an urinary buffer, directly linked up to the respiratory and kidney. Function. Acidification of urine as well as HCO3 buffer system serves as an index to understand the disturbance in the acid-base balance of the body. Function in normal state. Mild acidosis and alkalosis. Normally, flattered HCO3 secreted H is retained. In acidosis, loss of excess H plus balance acidosis. In alkalosis, loss of excess HCO3 balances alkalosis. Site of acid in kidney. Proximal tubule, thick ascending segment of the loop of Henle. Proximal tubule, site of action of kidney. Loop of Henle. Early distal tube. Intercalated cells in the latent distal and collecting tubules. What is alkali reverse? It is evident that at a pH of 7.4, the ratio of bicarbonate and carbonic acid is 10 to 20 to 1. Thus, the bicarbonate concentration is much greater, 20 times, than the carbonic acids of blood. This is referred as the alkali reverse, and it is responsible for affecting buffer of hydrogen ions generally in the body. Write down the specific specialties of hydrogen HCO3 buffer. HCO3 buffer is the most important buffer of the body because it has higher buffering capacity, 60% of total body buffer cavity. Its salt part is mineralized by kidney and acid part is maintained by lung. Salt part by kidney and acid part is maintained by lung. Its ratio of salt and acid part is 10 to 1. NaHCO3 25 milliequivalent per liter. H2CO3 1.4 milliequivalent per liter. Salt as the acid. Salt equals to NaHCO3, acid equals to H2CO3. NaHCO3 H2CO3 equal to 25 by 1.4, so 20 is to 1. It acts synergistically with the hemoglobin buffer system. It has wide field of buffering capacity. It is open-end system. It is present in plasma in higher concentration than the buffer system. Nice to know criteria of any ideal buffer. pK will be close to pH. Buffering capacity of buffer system will be higher. The salt part will be slightly higher than the acid part. Bicarbonate buffer is an unique buffer. Explained. Not potent, is the most potent, and why? Bicarbonate buffer is the most potent buffer in intracellular fluid. The bicarbonate buffer system is the most potent, powerful ECF buffer in the body. This is due to mainly the fact that the two elements of the bicarbonate system, HCO2 and HCO3, are regulated respectively by lung and kidneys. As a result of this regulation, the pH of the ECF can be precisely controlled by relative rate of removal and addition of HCO3 by the kidneys and the rate of removal of CO2 by the lungs. The other factor is that its presence in plasma at higher concentration than the buffer system. Write down the disadvantages of bicarbonate buffer. Disadvantages of bicarbonate buffer: limited buffer capacity. The bicarbonate buffer system is the most effective within a certain pH range, around 7.35 to 7.45. In case of extreme acid or base, it might not be sufficient to maintain the pH balance without additional mechanisms. Dependency of respiratory functions. The bicarbonate buffering system is closely linked to respiratory function because CO2 level, which affect bicarbonate concentration, are regulated by breathing in respiratory function is impaired. The effectiveness of the buffer system is compromised. Acidosis or alkalosis. In chronic conditions, excessive use or imbalance of bicarbonate can lead to metabolic alkalosis if there is any excess, or metabolic acidosis. Interact with other systems. In bicarbonate buffer system interact with other buffering systems like phosphate buffers and protein buffers, which can complicate pH management in complex conditions. Why HCO3 is usually used to know the acid-base status. HCO3 is usually used to know the acid-base status. All buffering systems of the body work in single pattern and fashion, but it is most difficult to assay the protein of hemoglobin concentration of the body. So by analyzing a particular buffer component, we can decide about the condition of the body's buffer system totally. HCO3 buffer maintains the maximum criteria of a standard buffer system. So ignoring all other buffer systems, HCO3 buffer is used as the standard to assess the acid-base status of the body. Blood is the most easily accessible medium of the buffer system. I write down the specialties of bicarbonate buffer. Specialties of bicarbonate buffer: most concentration in high concentration extracellular fluid, so it is the most potent in plasma. Directly lined up with respiratory, as H2CO3 is the most valiant acid. Why is phosphorylate buffer stronger than bicarbonate buffer? Phosphorylate buffer is stronger than bicarbonate buffer. We know that the buffering capacity is maximum when pH equal to pK. The pH of the ECF is 7.4. So the ECF of the buffer which have pK equal to closer or 7.4 will have the greatest buffering capacity. The pK of bicarbonate buffer is 7.1, which is away from pH or ECF 7.4, but the pK of phosphate buffer is 6.8, which is closer to the pH of ECF 7.8. This is why phosphate buffer is stronger than bicarbonate buffer. Where phosphate buffer is more active than bicarbonate buffer. Phosphate buffer is more active than bicarbonate buffer in ECF, extracellular fluid and renal tubular fluid, due to high concentration in intercellular fluid and renal tubular fluid. Write down the two important differences between bicarbonate and phosphate buffer. Composed of HCO3, H2CO3. Composed of HPO4 2 minus H2PO4 minus. Normal concentration ratio in blood HCO3, H2CO3. Normal concentration ratio in blood HPO4, H2PO4 equal to 4:1, 20:1, 4:1. Presence at high concentration in ECF. Presence in low concentration in ECF. Presence in low concentration in ICF. Presence in high concentration in ICF. Low concentration, high concentration in ECF bicarbonate and low concentration in PCF. তাহলে বাইরে বাইকার্বনেট বেশি, ভেতরে ফসফেট বাফার কম। বাইরে বাইকার্বনেট বাফার বেশি, তাই ভিতরে বাইকার্বনেট বাফার কম। Acts as a first line of defense as it is less efficient. Directly lines up with the respiratory and kidney. Directly lines up with the kidney. Directly lines up with the kidney. Directly lines up with the kidney. It is weaker than phosphate buffer because its pH is 6.1. Is away from the pH of ECF. Is stronger than bicarbonate buffer because its pK is 6.8 is closer to the pH of ECF 7.4. Compare the constant bicarbonate and ammonia buffer systems with diagram. Compare and constant between bicarbonate and ammonia buffer system of the diagram. Traits. HCO3 buffer system, NH3 buffer system. Bicarbonate HCO3 is blood buffer as well as urinary buffer, composed of carbonic acid and bicarbonate salt. Ammonia buffer is urinary buffer, composed of ammonia and other ammonium NH4, directly linking up to the respiratory of the kidney, directly linking up to the kidney. Sites of action of the kidney: proximal tubule, which ascending segment of the loop of the Henle. Early distal tubule, intercalated cells in the late distal and collecting tubules. Proximal tubule, collecting tubule. Site of action in kidney: proximal tubule and collecting tubule. Function: acidification of the urine as well as HCO3. Buffering system serves as an index to understand the disturbance in acid-base balance in the body. Acidification of the urine as well as about half to 2/3 of the body acids it's eliminated in the form of H and H4 ions by the mechanism, and it is the dominant mechanism to eliminate acid in chronic acidosis. Figure: compare the contrast between bicarbonate, phosphate and ammonium buffer. Compare between bicarbonate, phosphate and ammonium buffer traits. Components: bicarbonate buffer, phosphate buffer and ammonium buffer. NaHCO3, Na2PO4, Na3 NH4. pK value 6.1, 6.4 and 9.8. Ammonium buffer. H loss: normally no H loss, loss of H occurs, loss of H plus occurs. HCO3 gain: normally no HCO3 gain of H plus occurs, gain of H plus occurs. Mechanism: normally filtered on H3 prosected to H3, H3 filtered into the tubular lumen by glomerular and then converted to H2 HPO4. NH is synthesized within the tubular cell to glomerulate secrete NH3 combined with H plus in the tubular lumen site. Important in ICF. Short note: base excess. It is the amount of acid or base that would restore 1 liter of blood to normal acid-base composition at PCO4 and 40 mmHg. Base acid: it is the most of acid or base that would restore 1 liter of blood to normal acid-base composition. Short note: base excess. It is a condition where buffer base value of the patient deviates from the normal value by a quantitative which may be positive or negative. It is a condition where buffer base value of the patient derives from a normal value by a quantity for which may be positive or negative. Explanation: the buffer base Bp is equal to the total number of buffer ions. It can accept H plus. The normal value of Bp is 24 mmol per liter. In a patient, it is observed that Bp value is 20. The base excess value becomes minus 28 by the following relationship: base excess equal to observed Bp minus normal Bp equal to 20 minus 48 equal to minus 51. Base excess is minus 28 actually means either a deficient or 28 mM equivalent liter of base or an excess 20 milliequivalent liter of acid. Clinical importance of base excess. Base excess is positive in alkalosis. Base excess is negative in acidosis. Law of mass action. The law states that the rate of which a reversible action takes place at constant temperature and equilibrium is proportional to the product of the concentration and reaction substances. Explain. The law can be explained by a reversible reaction between ethanol and ethanoic acids. C2H5OH, C3COOH, acid 1 reaction, acid 2 reaction. C6COO, C2H5 and H2O. According to law of mass action, the rate of action is proportional to the product concentration of ethanol and ethanoic acid. Similarly, the rate of reaction is proportional to the product of concentration. Now, at equilibrium, the rate of two reactions are equal. The K is equilibrium constant. Importance. The law of mass action is used in determination of hydrogen concentration pH of a solution. Determination of dissociation of equilibrium constant, which is used to determine pK value. Compare of unknown substance on the basis of equilibrium constant. We can divide Hertzsprung-Hasselbalch equation by this law, using the law of mass action found on the equilibrium constant for following reactions. According to the law of mass action, the velocity of the reaction is proportional to the product of the concentration. Similarly, the velocity of reaction is proportional to the product concentration. Now, equilibrium rates of the two constants are equal. Hertzsprung-Hasselbalch equation. Hertzsprung-Hasselbalch equation. The relationship between pK of a solution and pK of the concentration of an acid and a conjugate base can be expressed by a convenient equation called Hertzsprung-Hasselbalch equation. pK equal to pK plus log equal to conjugate base and weak base. The deduction of equilibrium. A weak acid HA ionizes as follows: weak acid weak acid and its conjugate base. According to the law of action, equilibrium constant can be written as A K equal to H plus K H K. Log equal to log K H A and H. Acids are proton donor in aqueous solution. They may be molecule or ion. Acids contain hydrogen and capable of releasing hydrogen ion on the aqueous solution. Acid may be molecular species with positive charge or negative charge without charge. Conjugated base or an acid is the remaining part of the acid removal of proton. Strong acid is the acid which rapidly and completely ionizes into hydrogen and its conjugated base, even in the strongly acidic solution. For example, HCl, H2SO4 and H3PO4. Weak acid is an acid which slowly and particularly ionizes into hydrogen ion and its conjugated base in acidic solution. For example, H2CO3, H2PO4 acidic acid, acetotic acid and B-hydroxybutyric acid, etc. Degree of dissociation of a substance is called dissociation constant or ionization constant. Dissociation constant is symbolized as K. pK value is the negative logarithm of the dissociation of constant P. Strength of an acid is determined by the strength of hydrogen released by the dissociation in the solution, which again depends on the dissociation constant of the acid. The more the dissociation constant, the more the dissociation, the more the hydrogen release, the more the strength. For a strong acid, degree of dissociation and association of constant K is high. For example, dissociation constant HCl equal to 1% or 100. Conversely, the weak acid degree of dissociation of constant is low. Since pK is negative logarithm of dissociation constant K, so strong acid has pK value, weak pK value. So the relative strength of the acid is expressed in terms of pK value of the acid. pK value of acid and the strength of that acid is inversely related. pK value of the acid and the strength of the acid is inversely related. pK value of the acid and strength of that acid is inversely related. A strong acid, the conjugate base of the less affinity or to proton, because of which it can rapidly and completely ionize to release proton. So conjugate base of strong acids are weak in nature. On the other hand, in case of weak acids, the conjugate base allows strong affinity to proton, because of which acid partially ionizes to release proton. To conjugate the base of weak acids are strong in nature. Strength of an acid and the strength of a conjugate because are inversely related. Degree of dissociation depends on the prevailing of hydrogen ion concentration pH of the media. At higher pH dissociation, the more the lower pH. For example, H2PO4 dissociation in urine at pH 1/4 is more dissociated, is less dissociated, is least dissociated. 7.4, 6.1 and 5.8. Difference between the strong and the weak acid. feature: ionization, strong acid, weak acid, complete, partial, free acidity, high, low, dissociation constant, high, low, pK value, low, constant, strong acid, low, pK value are high, conjugated base, weak, strong, buffering activity, weak, absent, present. Base are proton acceptors in aqueous solution. They may be molecule or ion. Bases may be charged particles. Strong bases are those which have greater tendency to accept proton. They bind rapidly and strongly with the proton, so quickly remove proton from the solution. For example, HCO3OH. Weak bases are those which have very low tendency to accept proton. They bind slowly and weakly with proton, so slowly remove proton from the solution. Conjugated acid of a base in the acid form by the base after accepting proton. So conjugate acid is a strong base is weak conjugate, and a weak base and a conjugate acid of a weak base is strong. H2CO4 is weak conjugate acid strength with H3PO4 acid. HCl is strong conjugate acid of weak HCl base. Strength of an acid and the strength of its conjugate base are inversely related. So the relative strength of a base can be expressed in the terms of pK value of its conjugate acid, pK value of its weak acid, but its base is strong. Conjugate value of low pK value is more acidic than more acid, but its base is weak. So relative strength of the base can be expressed in terms of pK value. So its conjugate acid of its high pK value is weak acid, but its base is strong. Conjugate acid of low pK value is strong and acid, but base is weak. Therefore, pK value of an acid and the strength of the conjugate base of that acid are directly related. Base are directly alkali. Alkali are metallic hydroxides of alkali metals. Alkali in solution ionized to hydroxyl ions that can bind to hydrogen ion solution. Weak alkali are those which slowly and partially ionize to hydroxyl ions that slowly remove hydrogen from solution. In fact, all alkalis also regarded as base. But all bases are not alkali. In spite of that, in fact, all alkalis are base, but the base are not alkali. In spite of that, base and alkali are often used simultaneously. Strength of the alkali is determined by the amount of OH related by dissociation of the solution, which again depends on the dissociation constant of the alkali. The more the dissociation constant, the more dissociation, the more OH released, and the more the strength. Then there is acidity solution. It represents the state of OH concentration of a solution. Free acidity, actual acidity, and then treatable acidity, total acidity, treatable acidity. Then there is pH of solution. pH is negative logarithm of hydrogen ion. Concentration of a solution. H concentration is expressed in terms of molarity. Dissociation of water and its pH. Different effect of temperature on pH of water. Temperature, pH and status. Temperature normal. 25 natural pH. pH 7.66, 6.75, 6.71. At 37 degrees Celsius, pH is to 7.65. This is still in neutral pH. Effects of temperature and pH of water. So 7.66, 7, and then 7.65, 7.65, and then 6.71, 6.75, and 6.71. pH scale of a solution. It is a mathematical scale showing the interrelationship among the different values of pH and UH of aqueous solution at 25 degrees Celsius, and the ion production of factor is constant at 10 to the power minus 12. Different values. Information at pH scale: acidity, alkalinity, and then isoelectric pH. It is the pH at which biomolecules exist as zwitter ions possessing same amount of positive and negative charges on their surface with net charge 0. Limiting a pH solution, these ions either proton donor. By donating proton, they express negatively charged surface, and accepting proton, they express positively charged on their surface. So isoelectric molecules exist at electrical natural zwitter ion when the net charge of the surface biomolecules is 0, because positive and negative charges equalize each other. Therefore, at isoelectric pH, biomolecules are electrically neutral and exist as zwitter ions, not capable of short electrophoric mobility, least soluble in water, rapid precipitation of solution. At pH value of the isoelectric pH, biomolecules accept protons from the medium and exist as cations with net positive charge on their surface. At pH above the isoelectric pH, molecules release protons into the media exist as ions with negative charge on their surface. At pH below the isoelectric pH, biomolecules accept protons from the medium. At pH below isoelectric pH, biomolecules accept proton from the medium exist as cations with net positive charge on the surface. Therefore, pH. Biomolecules release proton at medium exist as anions at negative charge on their surface. So zwitter ions and then isoelectric pH. Buffer. Definition of buffer. Buffer is a mix of weak acid and its conjugate, because that tends to prevent the marked change of the pH and H concentration of a solution when the moderate amount of acid or base is added to it. Common buffering system of the body. Mechanism of the buffer action. And then factors affecting the buffer. pK value. The lower the value of pK, the lower is the pK buffer solution. When the ratio between base and salt. Maximum activity of buffer. Therefore, buffer shows a maximum activity at pH is numerically equal to the number of pK value that buffer system. Therefore, a buffer shows maximum activity at the pH which is numerically equal to the close to the pK value of that buffer system, because it is in this condition buffer pair exists with acid concentration equal to conjugate base of concentration. Therefore, and buffer shows maximum activity of the pH which is numerically equal to the close to the pK value of that buffer system. Traditionally, it is regarded as the buffer pair. Buffer with maximum pH at 7.4. Buffer pair shows the maximum activity at half neuralization. Buffer pair shows the maximum activity at half neuralization. Neutralization. Buffering capacity. It is the constant at which buffer system can buffer acid or base with negligible change in the pH. It is the amount of strong acid or base that can be added in 1 liter of buffer system. pH changed by 1 unit, and the more amount added, the more buffering capacity. Theoretically, it is true that the maintenance of pK 7.1 bodily differs need to pK. Role of buffer in our body. And that biomolecular system. Buffer system. bicarbonate buffer system, and then phosphate buffer system. In spite of all limitations, bicarbonate buffer system is the most effective physiological buffer in our body against all base and all acids except H2CO3 because it is an open-end system. Both work both in ECF and ICF. 86% buffering capacity in ECF and 36% buffering capacity in ICF. 86% buffering capacity ECF and 36% buffering capacity ICF. 36% ICF capacity in ICF. Phosphate buffer system. Mechanism of bicarbonate buffer and then phosphate buffer system. It consists of an acid component NH4 and its conjugate base or basic component NH3. It has pK value of 9. Alkali reserve. It is equivalent to plasma bicarbonate concentration. It is equivalent to plasma bicarbonate concentration. Compression of common body buffer systems. Conjugate base with base. pH value. Bicarbonate, protein, hemoglobin and phosphate. Conjugate base. pK value. Buffering of H2O3. Buffering of valve. Role of urinary buffer. Buffering capacity. The highest of bicarbonate, then protein, then hemoglobin, then phosphate. Composition and functions of blood. Define blood. Give the composition of blood. Blood is a specialized fluid from the connective tissue which is composed of intracellular substance, plasma and suspended form elements which circulate within a closed circuit of cardiovascular system. Whole blood. Formed elements, fluid, plasma. Formed elements are RBC, WBC and platelets. Solid plasma are solid 8 to 9% and water 91 to 92%. Solids are organic substances and inorganic substances and gases. Organic substances are proteins, albumin, globulin, fibrinogen, prothrombin. Non-protein substances are urea, uric acid, xanthine, hypoxanthine, creatinine, ammonia, amino acids. Carbohydrates include glucose, fructose. Lipids have phospholipids, cholesterol, neutral fat, etc. Coloring materials: bilirubin, carotene, xanthophyllin, others. Hormones, vitamins, enzymes. Internal secretions and antibodies. Others: hormones, vitamins, enzymes, amylase, protease, lipase. Internal secretions and antibodies. Nice to know. Special features of blood: color, taste, red, taste salty, specific gravity 1.05 to 1.06, pH 7.35 to 7.45, average 7.4, reaction slightly alkaline, volume 5 to 6 liters in male, 4 to 5 liters in female, viscosity 4.5 times more viscous than water, weight 7 to 8% of the body weight, osmolarity 280 to 310 millimole per liter, temperature 36 to 38 degrees Celsius, osmotic pressure average 55 millimeter mercury. Factors affecting blood volume: size of the body, changes in the fluid and electrolyte concentration, amount of fat tissue present in the body, leads the lifespan of blood cells. Lifespan of blood cells: RBC 120 days, WBC granulocytes 2 to 8 hours in circulating blood and another 4 to 5 days in tissue, granulocytes. Hours in circulating blood, monocytes 10 to 20 hours in blood before wandering through the capillary membrane into the tissues, and this form can live for months. Lymphocytes for weeks. Platelets 8 to 12 days. Name the cellular elements of the blood with their values, with their normal values. Give the total RBC, abbreviate platelet and SI units. RBC in male 4.9 to 5.5 10 to the power 12. 4.9 10 to the power 12, and in female 4.4 to 5 10 to the power 12. Unit per blood. Traditional unit SI unit. Traditional unit 10 to the power 12. Traditional unit 4.4 to 5.5 million, and then female 4.4 to 5 million. Million per cubic millimeter. WBC 4 to 11 over 9, or 4,000 to 11,000. Platelets are 150,000 to 400,000. Enumerate the functions of blood. Blood plays an important role in almost every functional activity. These activities include the following: transportation, regulation, and protection. Transportation of respiratory gases. Transportation of nutrients. Transportation of regulatory gases. Transportation functions of blood are transportation, regulation, and protection. Transportation of gases, transportation of nutrients, transportation of metabolic waste products and toxics to be excreted by excretory system, transport of hormones and enzymes. Regulation: temperature regulation, regulation of water balance, hormonal regulation, regulation of blood pressure and colloidal osmotic pressure, acid base regulation. Protection: carries antibody immunity, regulates water balance, regulates acid base regulation, colloidal osmotic pressure regulation, and hormonal regulation. And properties: carries antibody for immunity, coagulation, and prevention of hemorrhage. Carries antibody for immunity and coagulation or prevention of hemorrhage. Define plasma and serum. Plasma is the clear and yellowish fluid portion of the blood. It makes up 55% of the body's total blood volume. The main constituent of blood of water, 55%. Composition of plasma: it is mostly water and contains dissolved proteins, 6 to 8%, albumin, globulin, and fibrinogen, glucose, clotting factor, electrolytes, hormones, and carbon dioxide. Composition of plasma: this is the part of the blood that contains fibrinogen and other clotting factors. Composition of plasma. Separation procedure: blood plasma is separated by staining a tube of fresh blood containing an anticoagulant in a centrifuge until the blood cells fall in the bottom of the tube. The plasma is then poured or drawn off. Serum is the part of blood that remains once the clotting factors like fibrinogen have been removed by clot formation. Blood serum. Blood serum is blood plasma without clotting factors. Blood serum is blood plasma without clotting factors. It is the clot that makes the difference between serum and plasma. Serum is that part of blood which is similar in composition with blood. Blood excludes clotting factors of blood. Composition of serum. The composition of plasma and serum are similar as both contains hormone, glucose, electrolytes, antibodies, antigens, nutrients, and certain other particles except clotting factors, which are present only in plasma. So we can say that plasma minus clotting factors equal to serum. Why serum does not clot? The serum does not clot because serum lacks clotting factors like fibrinogen and some other clotting factors like factor 2, 5, and 8. Lacks clotting factors like fibrinogen. Other factors like 2, 5, and 8. What difference between plasma and serum? Difference between plasma and serum. Both plasma and serum are important parts of blood. The main difference between plasma and serum lies in their clotting factors. A substance called fibrinogen is essential in blood clotting. The blood plasma contains this fibrinogen. Basically, when serum and plasma are separated from the blood, plasma still remains in fibrinogen. A substance called fibrinogen is essential for blood clotting. The blood plasma contains this fibrinogen. Blood plasma. Basically, when serum and plasma are separated from the blood, plasma still remains in the fibrinogen that helps in clotting, while serum is the part of the blood that remains after the fibrinogen is removed. Plasma white blood cells are clot. Red cells in fibrin clot. Serum minus fibrinogen. Plasma and serum definition: the part of blood that contains both serum and clotting factors. Blood plasma without clotting factors. Clotting factors contains all the blood clotting factors, does not contain fibrinogen and some other clotting factors like 2, 5 and 8. Serotonin content low. Serotonin content high due to breakdown of platelets during clotting. Clot can clot because it has all the blood clotting factors, does not clot due to the lack of some of the clotting factors like fibrinogen. Color yellowish, color straw. Usage of medicine. It is often used for transfusion. It is used in numerous diagnostic tests such as well as blood typing. Name the sites of distribution of red bone marrow in adult person. Short note: red bone marrow. Active cellular bone marrow is called red bone marrow. Distribution: at birth, all the bones are filled throughout their length with highly cellular red bone marrow. At birth, all the marrow is red bone marrow. With increasing age, the red bone marrow is changed into yellow marrow. The process starts first in the distal bones and limbs, torsus and carpus. Then the intermediate marrow of long bones is yellow except for the upper ends of the femur and the humerus. Upper end of the humerus. In the adult, red bone marrow persists mainly in vertebra, sternum and ribs, bones of the skull and pelvis. Functions of red bone marrow. Formation of red bone marrow. Functions of red bone marrow. Functions of red bone marrow corpus cells. Formation of granulocytes into a less extent form of monocytes and lymphocytes. Formation of blood platelets. Destruction of red cells by macrophages while constitute part of lining of the blood sinus. Nice to know. Active marrow that is infiltrated with fat is called yellow bone marrow. Inactive bone marrow that is infiltrated with fat is called yellow bone marrow. Formation of red bone marrow corpuscles. Formation of granulocytes to a less extent, macrophages and lymphocytes. Formation of red bone marrow lymphocytes to a less extent of monocyte lymphocytes. Formation of functions of red bone marrow. Formation of red blood platelets. Distribution of red cells and macrophages constitute of lining of blood sinus. Draw the diagram to show the stages and development formed elements from the bone marrow cell. Stages of development formed elements blood from the bone marrow cell. PHSC, pluripotent hematopoietic stem cell. Colony forming unit. Colony forming unit blast. Colony forming unit erythrocyte. Colony forming unit spleen. Colony forming unit blast. Colony forming unit erythrocyte. CFUGM, colony forming unit granulocyte and macrophages. Colony forming granulocytes and macrophages. Neutrophils, eosinophils, basophils. Macrocytes, monocytes to macrocytes, granulocytes. CFUM, colony forming unit megakaryocyte. And then PHSC, pluripotent hematopoietic stem cell. Lymphatic stem cell, pluripotent hematopoietic stem cell, lymphatic stem cell, T lymphocytes and B lymphocytes. Pluripotent hematopoietic stem cell, L lymphocyte stem cell, T lymphocytes and then B lymphocytes. And pluripotent hematopoietic stem cell, colony forming unit spleen and then blast, and to erythrocyte and erythrocyte, colony forming unit B. Colony forming spleen into colony forming unit M, to megakaryocytes to platelets, and colony forming unit GM, granulocytes and monocytes, monocytes and granulocytes. From monocytes we get macromonocytes. Figure: formation of the multiple different blood cells. from the original pluripotent hematopoietic stem cell in red bone marrow. Bone marrow. It is usually the primary site of hematopoiesis in adults. A structure of bone marrow cells are WBC platelets. Connective tissue stroma, macrophages, reticular cells, fat cells, blood vessels, and nerve fibers. Structures of bone marrow. Bone marrow. What is leukopoiesis? It refers to the formation and development of various types of white blood cell. Granulopoiesis. Various blood cells. Granulopoiesis. It refers to the formation of granulocytes, neutrophil, eosinophil, basophil. Shortly narrate the genesis of WBC. Pluripotent hematopoietic stem cell. And then colony forming unit S. So colony forming unit spleen. And then colony lymphoid cell, lymphoid stem cell. Colony forming unit C, colony forming unit S, and lymphoid stem cell. Colony forming unit granulocytes then include myeloblast, promyeloblast, and monocyte. Myeloblast, promyeloblast, and monocyte. Myeloblast, promyeloblast, and monocyte. Monocytes has macrophages, macrophages. So colony forming unit granulocytes, colony forming unit lymphoid stem cells, colony forming unit S, and then colony forming unit GM, myelocytes, promyelocyte, and monocyte. From promyelocyte, myeloblast. From promyelocyte neutrophilic promyelocyte, eosinophil myelocyte, neutrophil myelocyte, basophil myelocyte. And then polymorphonuclear neutrophil, polymorphonuclear eosinophil, polymorphonuclear basophil. Give the genesis of neutrophil from stem cell and red bone marrow. Steps of genesis of neutrophil from stem cell red bone marrow. Pluripotent hematopoietic stem cell of red bone marrow. CFUS, colony forming unit spleen. GFUGM, colony forming unit granulocytes, monocytes. Myeloblast, promyeloblast. Neutrophil myelocyte, young neutrophil metamyelocyte, band neutrophil metamyelocyte, and polymorphonuclear neutrophil. Metamyeloblast, promyelocyte, neutrophil myelocyte, young neutrophil metamyelocyte, band neutrophil metamyelocyte, polymorphonuclear metamyelocyte. Plasma proteins. These are the proteins that are present in plasma and have a wide variety of functions. In adults, albumin, liver, fibrinogen, liver, prothrombin, liver, and then globulin alpha and beta, liver. Globulin alpha beta is liver, but globulin gamma is lymphoid tissue and plasma cells. In embryo, plasma proteins are derived from mesenchyme cells. Albumin is the first to be formed in the body. In embryo, plasma proteins derived from mesenchyme cells. Name the important plasma proteins with their normal values. Plasma proteins with their normal values: 6.4 to 8.3 gram per dl, 6.8 to 8 gram per dl. Albumin 4.8 gram per dl, globulin 2.3 gram per dl, fibrinogen 0.3 gram per dl, and then prothrombin 0.1 gram per dl. By electrophoresis, a number of proteins are isolated from globulin factors. These are plasma thromboplastin, esomhemagglutinin, angiotensinogens, immunoglobulins, anterior pituitary hormones. By electrophoresis, a number of proteins have isolated from globulin factors. These are plasma. By electrophoresis, a number of proteins are isolated from globulin factors. These are plasma thromboplastin, esomhemagglutinin, angiotensinogen, immunoglobulins, anterior pituitary hormones. Enumerate the functions of major plasma proteins. Albumin: maintain colloidal osmotic pressure, maintain viscosity of blood, acts as a buffer and maintains acid-base balance, acts as a carrier of hormones, fatty acid, bilirubin, and drugs, acts as protein reservoir. So protein reservoir and then acid-base balance, viscosity of blood and pressure. Globulin maintains colloidal osmotic pressure 20%, maintains viscosity of blood. Alpha 1 globulin indicates anti-protein protease and transports lipid and steroids. Alpha 2, so transports lipid and steroids. Copper, alpha 2 glucan transports copper in ceruloplasm. Beta globulin transports ferrum, transferrin, and globulin acts as antibodies. So alpha 1, alpha 2, beta, and then gamma. Fibrinogen, essential for blood coagulation. Increase ESR, helps to maintain viscosity of blood. Essential for blood coagulation. Prothrombin, fibrinogen, and then prothrombin. What are the methods of separation of plasma proteins in the blood? How plasma proteins can be separated? Methods for separation of plasma proteins. Precipitation by salt, fractional precipitation, sedimentation by ultracentrifuge, electrophoresis. Methods for separation of plasma proteins. Separates of plasma proteins. Precipitation by salt, functional precipitations, functional precipitation, sedimentation by ultracentrifuge, and electrophoresis. Precipitation by salt. Different plasma proteins are precipitated from solution by addition of different concentration of salts. Albumin is precipitated by full saturation with albumin is separated by full saturation with NH4SO4, NH42SO4. Globulin is precipitated by half saturation with NH4SO4, NH42SO4. Functional precipitation. It isolates the plasma proteins in quantity. Functional precipitation. Fraction is based on low salt concentration, low temperature, varying the pH, and modifying conditions by the addition of alcohol. Sedimentation by ultracentrifuge. Different proteins sedimentation are at different rates where the fluids of them are spun in very high speed. Swedberg ultrafiltration fuse. Thus the proteins can be separated. Electrophoresis, the movement of a charged electrical particle in an external electrical field towards the oppositely charged electrode is called electrophoresis. What is isoelectric point? It is an immediate pH specific of each protein that protein molecules carries equal numbers of positive and negative charges, and hence net charge is 0. It is an intermediate pH. The protein molecules carry equal number of positive and negative charges, and hence net charge is 0. The pH value for electrical neutrality is called isoelectric pH. Immediate pH protein molecules. List the properties of plasma protein. Properties of plasma protein: precipitated by salt, fractional precipitation, sedimentation by ultracentrifuge, isoelectric point, buffer action. And then molecular weight and shape, osmotic effect, viscosity, electrophoric mobility, and then transport action. Buffer action. Plasma proteins act as buffer by virtue of their power of hydrogen ion acceptance, but they account for less than 1/6 of the total buffering capacity of blood. Molecular weight and shape. Plasma protein albumin molecular weight 69,000, fibrinogen 330,000. Osmotic effect. The plasma proteins normally have an osmotic effect of 35 mm mercury and thus influence the exchange of fluid between the blood and tissue spaces. Osmotic effect. The plasma proteins normally have an osmotic effect of 25 mm mercury and thus influence the exchange of fluid between blood and tissue spaces. Viscosity. The resistance of the fluid through a capillary constant bore depends almost entirely on the viscosity of the fluid. The viscosity of the blood is thus a factor that in maintaining the peripheral resistance and thereby the arterial blood pressure. The viscosity of protein depends far more on the shape of protein than its size. The less symmetrical the molecule, the greater is the viscosity. The less symmetrical the molecule, the greater is the viscosity. Electrophoretic mobility. Transport action. Plasma proteins combine loosely with many chemical agents including hormones, for example thyroxine, cortisol, materials, for example, iron and copper, and numerous drugs. State the importance of erythropoiesis. Importance of erythropoiesis. It is an important property of plasma protein. By this property, different forms of plasma proteins can be detected in their concentration and can be measured. In different diseases, characteristic changes are found in different plasma proteins by it. How is plasma colloidal osmotic pressure produced? Why is plasma colloidal osmotic pressure important? Give the importance of plasma colloidal osmotic pressure. Production of plasma colloidal osmotic pressure. Plasma proteins are the colloids of plasma. Plasma proteins are the colloids of plasma. So plasma colloidal osmotic pressure is produced by the plasma proteins, 80% by albumin and 20% by globulin. Importance. Plasma colloidal osmotic pressure tends to cause fluid movement by osmosis from the intestinal space to the blood. This osmotic pressure normally prevents a significant loss of fluid volume from the blood to the intestinal space. What is hypoproteinemia? This is the cause of hyperproteinemia. This decreased protein concentration, lack of protein in diet, prolonged starvation, malabsorption syndrome, neutrophilic syndrome, acute glomerulonephritis. Because large amount of globulin are lost to liver disease, for example, hepatitis, cirrhosis, fibrin changes in liver, congenital deficiency, one or more plasma, fibrinogenemia, affects consequences of hyperproteinemia in the body. Hyperproteinemia, decreased plasma protein soluble albumin, plasma colloidal osmotic pressure, escape of fluid into the intestinal space from the blood vessels, accumulation of large amount of blood in tissue space, edema. What is plasma pheresis? Plasmapheresis is a process in which plasma cells can be removed from the blood without removing the blood's red blood cells. Plasmapheresis is a process in which plasma can be removed from the blood without removing the red blood cells. Blood is drawn from the patient and the plasma separates from it. The red blood cells are returned to the body of the patient such that there is no loss of these cells, yet some plasmas are removed. Clinical significance of plasmapheresis. It is used in blood purification process for an effective temporary treatment of many autoimmune diseases. It is called therapeutic plasma exchange. Plasmapheresis is an effective temporary treatment for the following diseases: myasthenia gravis, thrombocytopenic purpura, myasthenia gravis and thrombocytopenic purpura, chronic demyelinated polyneuropathy, Guillain-Barré syndrome, red blood cell. Red blood cells are biconcave, circular, non-nucleated disc shape from blood elements. Composition: water 65%, solid 35%, hemoglobin 33%, strong mass 2%. They are protein, phospholipids, cholesterol, ester, and natural fats. Red blood cells. Normal red blood cell at adult 4.9 to 5 million per cubic millimeter. Female 4.4 to 5 million per cubic millimeter. Infant 6 to 7 million per cubic millimeter. 700 times more numerous than leukocytes and 17 times more numerous than platelets. Numerous and 17 times numerous than platelets. Define erythropoiesis. The process of formation and maturation of erythrocyte under physiological condition is called erythropoiesis. Erythropoiesis it starts in third week of intrauterine life. Third week of intrauterine life. Sites of development of RBC. Intrauterine age from third to third month. Mesoderm of the agiosperm age from third week to third month. Mesoderm of the yolk sac life from third to month to fifth month. Liver, mainly spleen, from fifth month to birth. Red bone marrow from birth to 20 years. Red bone marrow after 20 years. Red bone marrow is of vertebrae, sternum, ribs, and bones, and the skull and pelvis, and the upper ends of the humerus and the femur after 20 years. So, third week to third month, mesoderm of the yolk sac. Third month to fifth month, liver, and fifth month to birth and bone marrow. So, third month to fifth month, and then third to third month, third week to third month. Types: mesoblastic erythrobiosis, hepatic erythropoiesis, and then myeloid erythropoiesis. From third week to third month in uterine life, primitive nuclear RBC are produced in intravascular from mesoderm yolk sac at the vasculosa. This is called mesoblastic erythropoiesis. Hepatic erythropoiesis from third to month to fifth month of fetal life. Nuclear RBCs are produced extravascularly from the liver and spleen. This is called hepatic erythropoiesis. Myeloid erythropoiesis from fifth month to the pregnancy to the delivery after birth. Non-nucleated RBC are produced extravascularly from the red bone marrow of the bones. This is called myeloid erythropoiesis. What is medullary and extramedullary erythropoiesis? The erythropoiesis in the red bone marrow is called medullary erythropoiesis. Normal erythropoiesis in adult and in the later half of the uterine life from fifth month to birth is medullary erythropoiesis. Extramedullary erythropoiesis. The erythropoiesis in size other than red bone marrow such as liver, spleen, and mesoderm of yolk sac is called extramedullary erythropoiesis. Normally, extramedullary erythropoiesis occurs in intrauterine life in the mesoblast or hepatic stages. In adults, such extramedullary erythropoiesis may occur in the liver and spleen, in which the bone marrow becomes destroyed or fibrosed. Development stages of erythropoiesis: pluripotent hematopoietic stem cell, committed stem cell, pro-erythroblast, basophil erythroblast, polychromatophil erythroblast. Pluripotent erythroblast, committed stem cell, pro-erythroblast, basophil erythroblast, and then polychromatophil erythroblast. PHF, PHU, pro-normoblast, early normoblast, and intermediate normoblast. Orthochromatic erythroblast, reticulocytes, and then erythrocytes, mature RBC. Pluripotent hematopoietic stem cell. So pro-erythroblast, and then basophil erythroblast, and then polychromatophil erythroblast, orthochromatic erythroblast, reticuloblast, reticulocytes. Pluripotent hematopoietic stem cell. Pluripotent hematopoietic stem cell are the red bone marrow that can be differentiated into all types of circulating blood cells. Characteristics of pluripotent hematopoietic stem cell: few in number, has self-renewing capacity, can give rise to all types of blood cells. Upon appropriate stimuli, stem cells can be converted into any type of cell. For example, hepatocytes property is called stem cell platicity. Briefly discuss the different steps of erythropoiesis. Stages of erythropoiesis. Stage 1: pro-erythroblast, cell size 15 to 20, nucleus large, occupies 3/4 of the cell volume. Cytoplasm basophilic, deep blue, basophilic, hemoglobin absent, and mitosis present only in state of stress. We discuss the different steps of erythropoiesis. Stage 2: describe the changes of erythropoiesis. The stage pro-erythroblast and then basophil erythroblast, somewhat smaller. Nuclei disappears in basophil erythroblast, pro-erythroblast, and then basophil erythroblast, and then polychromatophil erythroblast 10 to 14 meter. Further condensation of chromatin, polychromatic, and increases in active protein. Orthochromatic 7 to 10, a small progressive chromatic erythroblast, cartwheel shaped. Orthochromatic has cartwheel shaped. 15 to 20, 10 to 14, 7 to 10. Reticulocyte disappear. Erythrocyte 7.5 to 8.5, mean 7.1, absent. Nucleus disappears. Further condensation, nuclei disappears, but nuclei disappears in basophilic erythroblast, polychromatophil erythroblast. Further condensation of chromatin, orthochromatic. cartwheel-shaped appearance. Reticulocytes disappears. Erythrocytes absent. Reticulocyte disappears and then erythrocyte absent. And cytoplasm basophil, like basophilic, polychromatic, basoeosinophilic, eosinophilic. Hemoglobin absent, begins to appear, increases, synthesize, complete, present, and then mitosis, present, active mitosis, active mitosis, absent. Basic changes during erythropoiesis: a decrease in size of the cell, increase of condensation, and finally biokinesis of nucleus, kinesis of nucleus, accumulation of hemoglobin, a change in the staining reaction of the cytoplasm from basophil via chromatophilic eosinophil, chromatophil to eosinophil, polychromatophil eosinophil. So nucleus disappears and then 10 to 15 somewhat smaller, 10 to 14, 7 to 10, and then 7.5 to 8.8. Nucleus large and then nucleus disappears, further condensation, cartwheel shape disappears. A basophilic, polychromatic, eosinophilic, basophilic, polychromatic, eosinophilic, basophilic, and then polychromatic, eosinophilic, and increases, basophilic, eosinophilic, polychromatic, eosinophilic. What are the basic changes occurred during erythropoiesis? The change in the staining reaction of the cytoplasm from basophilia via polychromatophil to eosinophil. Name the first non-nucleated cell of erythropoietic series. First non-nucleated cell of erythropoietic series, reticulocytes. Mention the properties of reticulocytes. Larger than mature RBC. No nucleus has some ribosome materials. Bluish tint in supravital staining. Properties of reticulocytes. Larger than RBC. Non-nucleus has some ribosome materials. Bluish supravital staining. How much time is required to form mature RBC from reticulocytes? 2 to 3 days. Why reticulocyte is called the immature RBC? Reticulocyte is called immature RBC because it is the first non-nucleated cell of erythropoietic series. Its hemoglobin content is sufficient due to reticular appearance. Its first immature RBC because its hemoglobin is sufficient due to reticular appearance. Reticular appearance gives less space for hemoglobin. It's insufficient. Its contains more cell organelles than erythrocyte. It's capable of mitotic division. It is rounded and not biconcave. Define poikinosis. Poikinosis is a stage in degeneration of nuclei which breaks up and finally disappears, going to extrusion of lysis. A young RBC reticulocyte is thus formed. It occurs in stages of late normoblast. List the factors controlling regulation of erythropoiesis. Nutrients, essential nutrition stimulation, and then hemoglobinization factors. Nutrition factors: essential amino acids, vitamin B12, folic acid, vitamin C. Intrinsic factor of cell. So essential amino acid vitamin B12, amino acid vitamin B12, folic acid vitamin C, and intrinsic factor of cell. Essential amino acids: globin synthesis, vitamin B12, DNA synthesis, folic acid, DNA synthesis, vitamin C, iron absorption. Intrinsic factor of cell: absorption of vitamin B12. Stimulation promoting influence factors. Hypoxia: it enhances erythropoiesis, which occurs at high altitude, congenital heart disease, after hemorrhage and anemia, etc. Hypoxia. Erythropoietin. Erythropoietin is secreted from the kidneys and liver in response to hypoxia. It directly stimulates bone marrow to produce red cells. Hormones: a thyroid stimulating hormone, growth hormone, prolactin, and acetylcholine. Adrenocorticoid stimulating hormone. And then hemoglobinization factors. We have Fe2 plus UMG, CO, Ca, and bile salt. Fe2 plus required for heme synthesis, bile salt. helps to absorb heavy metals, for example, ferrum, copper, and manganese. Trace elements catalyze reactions of hemoglobin synthesis. State the effects of hypoxia and erythropoiesis. Role of hypoxia and acute hemorrhage on erythropoiesis. Hypoxia and acute hemorrhage both stimulates erythropoiesis, but through related mechanisms. Hypoxia, low oxygen levels in tissue trigger the kidneys to release erythropoietin. EPO promotes the production and maturation of red blood cells in the bone marrow to increase the oxygen carrying capacity of the cell of the blood. Hypoxia triggers the kidney to release erythropoietin. Erythropoietin promotes the production and maturation of red blood cells in the bone marrow to increase the oxygen carrying capacity. Hypoxia. Acute hemorrhage, low oxygen levels. Role of hypoxia in acute hemorrhage erythropoiesis. Reduce both blood volume and number of red blood cells, leading to tissue hypoxia. The further stimulates EPO release, enhancing erythropoiesis to replace the lost red cells and restore oxygen delivery. In summary, both hypoxia and acute hemorrhage promote erythropoiesis primarily by increasing erythropoietin secretion, which drives red cells production in response to reduced oxygen availability or blood loss. In summary, state the effects of high altitude on erythropoiesis. At high altitude, where the quantity of oxygen of the air is greatly decreased, the insufficient oxygen is transported to the tissue. Red blood cell is greatly decreased in red bone marrow. Maturation factor, vitamin B12, coenzyme, and folic acid such as catalytic DNA synthesis. Maturation factor RBC. Please write about vitamin B12 and folic acid from the discussed below. Maturation factor of RBC. Short note: erythropoiesis. Discuss the erythropoiesis. Mention definition stages erythropoiesis with diagram with nutrition factors and influencing erythropoiesis. Erythropoiesis. It is a circulating glycoprotein which in higher organisms in the prime regulator of erythropoiesis. It is a circulating glycoprotein which in higher organisms in the prime regulator of erythropoietin. It is a circulatory glycoprotein hormone having a molecular weight of about 34,000. Erythropoietin, which is released in hypoxia condition, stimulates RBC production. It is a glycoprotein which in higher organisms in the prime regulation of erythropoiesis. Molecular weight 34,000. Clinical structure. It is a glycoprotein containing protein 74% and carbohydrates 26%. Source: kidney. From peritubular intestinal fibers like cell of kidneys. Liver 10%. Stimuli erythropoietin release. Hypoxia, anemia, alkalosis, and androgens. 3A's: hypoxia, anemia, alkalosis, and androgens. Erythropoietin release. Sites of action of erythropoietin: bone marrow, RBC precursors. Role of erythropoietin in erythropoiesis. Important factors of erythropoietin is to stimulate the production of pro-erythroblasts from the hematopoietic stem cell in red bone marrow. Once the pro-erythroblasts are formed, the erythropoietin causes these cells to pass more rapidly through the different erythroblastic stages than they normally do. When there is hypoxia, the kidney secretes erythropoietin. Hypoxia, kidney, erythropoietin. High hematopoietic stem cell of red bone marrow. Pro-erythroblast, RBC, hemoglobin and tissue oxygenation. Kidney erythropoietin blast and oxygenation. Importance: when an individual becomes hypoxic, erythropoietin stimulates erythropoiesis and thus increases tissue oxygenation until the hypoxia is relieved. Factors: inhibitors that production of RBC. Prostaglandin, angiotensin, large doses of steroid hormone 5-HT serotonin, estrogen. So prostaglandin, angiotensin, large doses of steroid hormone 5-HT and estrogen. How is the production of RBC adjusted to meet the varying needs of individual? Adjustment: production of RBC to meet the varying needs of the individual. In hormonal circumstances, erythropoiesis is regulated so as to maintain the number of erythropoiesis and hemoglobin content with a normal range. Normal balance between production of destruction of RBC is maintained by daily output of 10^11 erythrocytes from the bone marrow. The cells surviving for 120 days. The balance is upset if there is an excessive loss of blood, for example in hemorrhage, or if there is some defect in production of erythrocytes by some bone marrow. Erythropoietin is the prime regulator of erythropoiesis. Describe the morphology of mature RBC. Morphology of mature RBC: size and shape: circular, non-nucleated, biconcave disc. Diameter 6.5 to 8.8 micrometer. Thickness 2.5 micrometer. The thickest point 1 less center. Surface area 140 micrometer. Volume 90 to 95 micrometer cube. Lifespan 120 days. Hemoglobin: each hemoglobin contains 29 picograms. There are about 900 grams of hemoglobin in circulating blood. 900 grams of hemoglobin. What are the peculiarities of mature RBC? Absence of nuclei, absence of mitochondria, absence of ribosome, DNA and RNA. So synthesis of protein is impossible in RNA in RBC. Presence of hemoglobin biconcave shape. How can you differentiate between an mature and an immature RBC? Difference between mature and immature RBC. Derived from mature RBC, mature RBC, reticulocyte. Orthochromatic erythroblast, mature RBC, mature reticulocytes, and immature orthochromatic erythroblast. Amount 5.5 × 10^12 liter, 0.1 to 2% of red blood cells. Size 7.8 micrometer, less than RBC, biconcave and rounded. Lifespan 120 days, 1 to 2 days. Stain, Leishman stain and supravital stain. So Leishman stain and supravital stain, mature, immature. Leishman stain, supravital stain. What are the functions of RBC? Function of RBC: respiratory function. The major function of RBC is to transport hemoglobin, which in turn carries oxygen from the lungs to the tissues in the form of hemoglobin and carries CO2 from the tissue to lung in the form of carbo-oxygen hemoglobin. Acid-base balance. The hemoglobin of RBC acts as an excellent acid-base buffer and helps to maintain acid-base balance. Respiratory function, acid-base balance. Maintenance of viscosity. RBC helps in maintaining the viscosity of blood. If RBC is increased in plasma, the viscosity of blood will be increased. Maintenance of viscosity. Production of pigments. Various pigments are produced from hemoglobin after the disintegration of the RBC, bilirubin and biliverdin, etc. Determination of blood group. The blood grouping antigens are present on their membrane. For example, AB blood grouping antigens, RH antigens will determine the blood group. Ionic balance. RBC maintains the intracellular ionic balance by a special permeability of the membrane. What are the functions of RBC? The major function of RBC is the transportation of hemoglobin that carries oxygen, acid-base balance, maintenance of viscosity, production of pigments, determination of blood group, and ionic balance. Shape of RBC: normal blood cell, circular, biconcave, significant biconcave shape. And surface area: uptake, release CO2 and O2. It allows the considerable alternation of volume, and hemoglobin remains distributed in very thin layers in gases. It is usually squeezed through pus and capillary pores. What is the advantages of the absence of nuclei in RBC? It gives biconcave shape RBC. It also gives more space for hemoglobin. What is the advantage of the absence of mitochondria in RBC? RBC contains mitochondria. It will use all the O2 and respiratory chain, thus O2 carrying to the tissue will be destroyed. Provide space for hemoglobin. RBC produce ATP in the absence of mitochondria. Production of ATP by RBC in the absence of mitochondria. Anaerobic glycolysis, HMP shunt. Production of ATP by RBC in absence of mitochondria. Production of ATP by RBC in absence of mitochondria, anaerobic glycolysis, and HMP shunt. Define the following terms related to abnormal forms of RBC. Microcytes, when RBC is smaller than normal size. Macrocytes, when RBC is larger than normal size. Hypochromatic, referred as a cell with less color. Define the following terms. RBC when normal in size and shape is called normocyte. Normochromic, when concentrate of hemoglobin per RBC is normal. The RBC are called to be normochromic. Hemolysis. The process of breakdown RBC is called hemolysis. What are the causes and the variations of RBC count? Physiological causes: high altitude, muscular exercise, sex. So high altitude RBC and hypoxia. A muscular exercise when RBC count increases. Muscular exercise due to contraction of spleen. Exercise due to contraction of spleen when RBC increases in RBC. Muscular exercise due to spleen. Sex. Spleen. Muscular contraction of spleen. In male, RBC count is greater than female because no loss of RBC, no menstruation, testosterone stimulates erythropoiesis. Age: infants during first few days of life having high RBC count. Diurnal variations: the RBC count is the lowest in the morning and highest in the evening. Diurnal variations: high environmental temperature in increased RBC count. High environmental temperature, high barometric pressure increased RBC count. High oxygen pressure, it decreases RBC count. Pathological causes: in condition leading to defective oxygenation of blood on RBC count, such as emphysema, pneumothorax, pulmonary fibrosis, pulmonary tuberculosis. Chronic and congenital heart disease: chronic calcium monoxide poisoning, arsenic and phosphorus and lead poisoning. Polycythemia vera, any condition leading to hemoconcentration, for example burn, shock, cholera, severe diarrhea, severe vomiting, etc. increase RBC count. Conversely, hemoglobin will lead to fail RBC count, loss of RBC followed by fluid replacement and other than fluid mediated hemodilution. In spite of having no nucleus, why RBC is called cell? Because RBC defined lifespan is 120 days, it is most highly adaptable by cell body. It is most highly adapted cell in the body. It carries all the metabolic functions and several enzymes. Microsurgery technique proven that nucleus is not essential to survival of the cell. What is the ratio of WBC and RBC in bone marrow and circulating blood? In bone marrow, WBC RBC 50:1 and circulating blood is 1:500 RBC and WBC. Erythrocyte sedimentation rate: the blood containing an anticoagulant is allowed to stand in a narrow vertical tube. The erythrocytes settle to the bottom half of the tube. The rate at which this occurs is called the erythrocyte sedimentation rate. When a volume is mixed with a specific amount of suitable anticoagulant and is allowed to stand in a vertical position in a narrow tube, the blood cells form elements settle down, leaving the clear plasma above. The sedimentation of red cells that takes place in first hour is called ESR. Anticoagulant used: trisodium citrate. Method of ESR estimation: Western grain method and Winthrop's method. Western grain method and Winthrop method. When a volume of blood is mixed with a specific amount of suitable anticoagulant and allowed to stay in a vertical position in a narrow tube, the blood cells settle down, leaving the clear plasma above. The sedimentation of red blood cells takes place in first hour is called ESR. Methods of ESR estimation is Western grain method and Winthrop's method. Western grain method and Winthrop's method. Male 1 to 15 mm in first hour, 1 to 9 mm in first hour. Female 1 to 20 mm in first hour, 0 to 20 mm in first hour, 0 to 20 mm in first hour. 0 to 15 and 0 to 20. Factors influencing the ESR: any factors which increase relief formation, plasma fibrinogen. Method of determination: the length and the diameter of the tube, amount of anticoagulant, except potassium oxalate, amount of anticoagulant use, excess potassium oxalate will increase ESR, and excess sodium oxalate will decrease ESR. Sodium oxalate, potassium oxalate, and sodium oxalate. Delay in the test. Position of ESR. ESR in inclined position. Temperature ESR. Anemia ESR. Mention of clinical importance of ESR. It is a measure of biochemical changes in blood. It has no specific diagnosis value. ESR is non-specific, used as a screening test. Ultra ESR always indicates presence of active disease, but normal ESR excludes the active disease. Also monitors the progression of disease or process. Short note: raised ESR, physiological condition, old age. All over the age of 60, ESR of 30 mm at the first hour may be normal. Pregnancy, vaccination, high altitude. Pathological indications: acute, chronic, or infective tuberculosis, advanced malignancy, systemic lupus erythematosus, rheumatic arthritis, multiple myeloma, aplastic anemia. Mention the causes of high risk: sky rise, mark of ESR, multiple myeloma, tuberculosis, SLE, systemic lupus erythematosus, rheumatic arthritis, advanced malignancy, aplastic anemia. Cause of reduced ESR: polycythemia, sickle cell anemia, xerocytosis, congestive cardiac failure, hepatic failure, and fibrinogenemia. Write the technical procedure of ESR done in the laboratory. Procedure: ESR estimation. Material required: Western green method, Western green tube bag, test tube, reagent, a sterile needle, cotton splint. Winthrop hematocrit method, EDTA, is used as an anticoagulant. Winthrop method. Procedure: take 0.4 ml drop blood at 1.6 ml of hemoglobin blood in the test tube and mix thoroughly. Draw the blood from the test tube in the Western green tube by shaking it and mouth up to the mark 0. Place the test tube in a rank in vertical position with the 0 mark above. Take the reading after 1 hour by noticing down the level of the in plasma. Results can be given as ESR millimeter equal to 1 first hour. Nice to know. Western green method is better. Western green tube is long calibrated. Western green 1 to 200 millimeter can estimate ESR above 100 millimeter, whereas the Winthrop tube is calibrated in ESR 0 to 100 millimeter. Very high ESR above 100 millimeter can be measured. So Western green method is better, more sensitive. Rayleigh formation. Rayleigh formation by RBC aggregation of one over another is called Rayleigh formation. Rayleigh formation. Cause of formation. The cause is unknown. It's believed that condition is displayed the surface tension. Influence factors. The Rayleigh formation increases when there is increase of plasma fibrinogen and gamma globulin. The tendency of RBC to settle down increased when the Rayleigh formation. Thus, Rayleigh formation of ESR. Pronunciation of Rayleigh is Rayleigh. Packed cell volume. The packed cell volume of hematocrit is the volume of red blood corpuscles in relation to the whole blood. The hematocrit of PCV refers to the proportion of volume of red blood cells relative to the total volume of blood. It is expressed in the volume of RBC per liter in whole blood. It is also expressed in percentage such as 45%, i.e. 45% RBC is present in 100 ml of blood. In male, average 45%. In female, average 42%. Volume of packed red cells, volume of whole blood. Method of estimation: Winthrop's method and microhematocrit method. Indications of PCV estimation: screening test for anemia and a rough guide to accuracy of hemoglobin measurements. In conjugation of estimation of hemoglobin RBC count. It can be used in the calculation of RBC indices. Cause of increased PCV: polycythemia due to any cause, hematoconcentration due to any cause. Cause of increased hematopolycythemia. Cause of decreased PCV: anemia due to any cause, hemodulation due to any cause. Name the materials required for Wintroff's method of PCV estimation. Materials required for Wintroff and method PCV estimation: Wintroff's hematocrit tube, Paul Heller's mixture, capillary pipette, Paul Heller's method PCV estimation, Paul Heller's. Cause of increased PCV: polycythemia due to any cause, hematocrit due to any cause. Cause of decreased PCV: anemia due to any cause, hemodulation. Increased due to any cause, hypoxia and hematoconcentration. Screening for anemia: red cell indices. Name the materials required for Wintroff's method of PCV estimation: Wintroff's hematocrit tube, Paul Heller's mixture, and capillary pipette and centrifuge machine. What is buffing coat? Buffing coat is the top layer of the packed cell in the hematocrit on centrifuge. Buffing coat is the top layer of the packed cell in the hematocrit of centrifuge. This layer consists of WBC platelets and immature brainy cells. Short note: red cells indices. The measurement of mean corpuscular volume, mean corpuscular hemoglobin, and corpuscular hemoglobin concentration. Mean corpuscular volume, mean corpuscular hemoglobin, and mean corpuscular hemoglobin concentration is called red cell indices. Normal range 76 to 95 femtoliter, 57 to 32 picogram, and 31 to 35 gram per deciliter. All the calculated measure of hemoglobin. Mean corpuscular volume. The average volume is RBC is packed cell volume into 10. RBC account million. 45 into 10 by 5, 90 micrometer cube or femtoliter. MCH equal to hemoglobin in percentage into 10, and so on 5 into 10 by 5, so 30 picogram. And then mean corpuscular concentration, MCHC, hemoglobin, and PCV, so 15 into 145, 33.3%. Which data are required for measurement of cell indices? Data required for measurement of cell indices measured: RBC concentration, packed cell volume, total RBC. Mention the uses of RBC indices. They are used in morphological classification of anemia, normocytic and normochromic anemia, hypochromic and microcytic anemia, decreased red blood indices, macrocytic anemia, increased in PCV, MCH but normal MCHC, increased in but normal MCHC, normocytic MCV is normal, microcytic MCV is less than 76 femtoliter. Macrocytic MCV is greater than 97 femtoliter. Macrocytic. Normochromic MCH and MCHC are normal. Hypochromic MCH is less than 75 and MCHC is less than 31 gram per dl. Define hemoglobin. Hemoglobin is the pigment chromatoprotein present in erythrocytes and are responsible for the red color and consistent protein globulin unit pigment hem. Normal range 33 gram per dl. Children 12 gram per dl. Adult 14 to 18 gram per dl. Adult female 12 to 16 gram per dl. 14 to 18 and 12 to 16 gram per dl. Function of hemoglobin: transport of O2 from the lungs to the tissue, CO2 from the tissue of the lungs, transport of blood buffer materials, and then pigments and variation. Pigment and globulin, reserve for iron and protein in lungs. Why hemoglobin is more in males than female? Because female testosterone stimulates erythropoiesis. Why is hemoglobin content more in newborn? In newborn, the hemoglobin content is greater due to more erythropoiesis. Inside the uterus, the fetus remains in a state of relative hypoxia. In this state, erythropoietin production increases in the kidney, which results in more erythropoiesis. In a relative hypoxia, erythropoietin production results in more erythropoiesis. Production occurs due to contain RBC. In newborns, hemoglobin content is high, 20 to 23 gram per dl of blood. Give the chemical structure of hemoglobin. Chemical structure. Hemoglobin consists of 2 alpha chains and 2 beta chains. Each chain consists of heme and peptide chain. Heme is an iron-containing porphyrin known as iron protoporphyrin 9. Iron protoporphyrin 9. The porphyrin nucleus consists of a pyrrole ring joined together by 4 mechanical bridges. The polypyrene are thus tetrapyrrolles. The polyhedral rings are numbered 1, 2, 3, 1, 2, 3, and 4. There are 8 side chains attached to 4 pyrrole rings, the position of which the side chains are attached are number 1 to 8, and side chains to 1, 3, 5, 8 are methyl 1, 2, and 4, vinyl 6 and 7 are propionic acid. The ion heme in the ferrous form is attached to N of each pyrrole ring of the N imidazole group. Imidazole group. Globulin is polypeptide consist of amino acids. It has 4 subunits. Each subunit consists of a global chain and heme moiety. Total 4 globular chains, 2 alpha chains and 2 beta chains, 4 ferrous chains. Each hemoglobin chain combines with heme group, whose central ion is site in which oxygen attaches to hemoglobin. Site of steps of synthesis of hemoglobin. Hemoglobin synthesis begins in the proerythroblast stage and is completed in the orthochromatic erythroblast stage. Raw materials: succinyl-CoA, glycine, and iron. Site of synthesis in mitochondria. Steps: acetic acid is converted to succinyl-CoA. 2 succinyl-CoA plus 2 glycine equal to pyrrole compound. 4 pyrrole compound and equal to protoporphyrin 9. Protoporphyrin 9 equal to ferrum equal to heme. Raw materials: amino acids, ribosome of ER, DNA, MR, and then polypeptide, heme and polypeptide, hemoglobin. Alpha 2 chains, alpha beta chains, and hemoglobin A. Enumerate the factors required for hemoglobin synthesis. Factors for hemoglobin synthesis: first class protein, metals, vitamins, endocrine glands. Factors for protein: first class protein, metals, vitamins, and then endocrine gland, and then porphyrins, bile salt, and erythropoietin, and genetic control. Enumerate the factors required for hemoglobin synthesis. First class protein. What are the type of hemoglobin? Physiological hemoglobin and pathological hemoglobin. Physiological hemoglobin includes types of hemoglobin are physiological hemoglobin and then pathological hemoglobin. Composition of normal and abnormal hemoglobin. Hemoglobin A, hemoglobin A2, and hemoglobin F. Hemoglobin alpha 2, hemoglobin beta 2, hemoglobin alpha 2 and delta 2, hemoglobin alpha 2 and gamma 2. Alpha 141 amino acids, 146 amino acids, 146 amino acids, 146 amino acids. Alpha, beta, delta, gamma. Abnormal hemoglobin. Abnormal hemoglobin SCE. Hemoglobin D Punjab. Alpha 1, beta 2, alpha 1, beta 2, alpha 1, beta 2. Number 6. Glutamic acid is replaced by vanillin. Classify the hemoglobin with their function differences. Hemoglobin A, alpha 2, beta 2, hemoglobin B, hemoglobin A2, alpha 2, delta 2, hemoglobin F, alpha 2, gamma 2. Functions: main hemoglobin in adults, transports and oxygen affinity. Minor hemoglobin, adult hemoglobin, similar to hemoglobin A, and then hemoglobin F, alpha 2, gamma 2. Oxygen affinity in exact to method blood. 98% adult, 9% adult, and fetal and newborns. Difference between hemoglobin A and hemoglobin B. Chain sequence: hemoglobin alpha 2, hemoglobin beta 2, hemoglobin alpha 2, hemoglobin gamma 2, hemoglobin F. And then affinity less and more. Affinity of 2,3 BPG, more, less. Alkali resistance less, much more. Importance: it is the main hemoglobin in adults red cell for carrying O2 in the tissue. The greater affinity for O2 facilitates movement of O2 energy material in fetal circulation. Hemoglobin F. Hemoglobin F, fetal hemoglobin present in blood and human fetus, normally replaced by adult hemoglobin as soon as after after birth. In certain individuals, fails to disappear and persists throughout life. A structure. It consists of two chains and two alpha chains and two gamma chains. Hemoglobin F has a greater affinity for O2 than hemoglobin A because the does not bind with 2,3-DPG. This facilitates fetal RBC in pickup O2 at level PO2 available in the placenta. Moreover, it is more resistant to alkali in hemoglobin A. Hemoglobin A 2,3-dihydroxy phosphoglycerate. Hemoglobin F has a greater affinity for O2 than hemoglobin A because it does not bind with 2,3-DPG. Dihydroxyphosphoglycerate. This facilitates fetal RBC in picking up O2 available placenta. How does fetal hemoglobin benefit the fetus? The greater affinity binds less avidly with 2,3-DPG. Dihydroxyphosphoglycerate. Dihydroxyphosphoglycerate 2,3-DPG. This facilitates movement of O2 from the maternal to the fetal circulation. Moreover, it is more resistant to alkali than hemoglobin A. How can a molecule of Hb carry a 4 O2 molecules? The iron in fetus state Fe2+ is attached to an each iron ring and to the end of the imidazole group of the hemoglobin. One bond is available for loose iron with O2. Thus, one ferrous iron can bind only with one molecule. As one molecule hemoglobin has 4 ferrous ions and carry 4 molecules. Describe the hemoglobin regarding its synthesis, concentration, and blood types. Disorders abnormalities of reduced number of hemoglobin, abnormal globin chains, thalassemia, reduced sickle cell disease, and defects in hemoglobin synthesis, or Peyer's sickle cell disease and thalassemia, reduced hemoglobin, abnormal globin chains, and heme synthesis. Abnormal hemoglobin leads to anemia. Abnormal hemoglobin leads to anemia primarily because it impairs the red blood cells' ability to carry oxygen efficiently, causes their temperature destruction, and disrupts normal red blood cell production. This results in reduced number of hemoglobin and red blood cells, decreased oxygen carrying capacity, premature destruction of red blood cells, ineffective hemoglobin production. So decreased hemoglobin carrying capacity, premature destruction of red blood cells, and ineffective hemoglobin production. Thalassemia, etc. Reduced RBC and lifespan. What are the abnormalities of hemoglobin? Two major types of inherited disorder hemoglobin events: thalassemia and hemoglobinopathies, sickle cell disease, etc. For example, hemoglobinopathy. The inherited disorders of hemoglobin in which abnormal polypeptide chains are produced are called hemoglobinopathies. The hemoglobinopathies are characterized by the production of structurally defective hemoglobin due to abnormalities in formation of globin moiety in the molecule. Hemoglobin C, hemoglobin D, hemoglobin E. Define and classify thalassemia. Thalassemia is an inherited disorder or hemoglobin in which the hemoglobin chains are normal in structure but production is decreased amount or absent because of defective or regulatory portion of hemoglobin genes. The thalassemia are a heterogeneous group of disorders with a genetically determined reduction of rate of synthesis in one or more types of abnormal hemoglobin polypeptide chain. Type thalassemia, beta thalassemia, and alpha thalassemia. Beta thalassemia is the inadequate production of hemoglobin chains leads to reduced amount of hemoglobin A in the red cell. Hemoglobin A leads to reduction of hemoglobin A and macrocytic hypochromic anemia results. Type of beta thalassemia on the basis of extensive reduction of beta chain on clinical class beta cells is major and beta thalassemia minor. And alpha thalassemia. Alpha thalassemia are disorders where the division synthesis of chains with resulting depression of production of hemoglobin alpha chains. Types: alpha thalassemia, alpha thalassemia 1, alpha thalassemia 2, hemoglobin H and hemoglobin birth hydrops. 4 rings are detected. Mention the features of thalassemia: pallor, diarrhea and rare and recurrent fever, severe anemia, hepatomegaly, splenomegaly, mongoloid face, slow growth in childhood, epistaxis, nasal bleeding, skin pigmentation, and leg ulcer and gallstone. Pallor, diarrhea, rare fever, severe anemia, mongoloid face, slow growth in childhood. Give the difference between thalassemia and iron deficiency anemia. What are the methods of hemoglobin estimation? Cyan hemoglobin method, Sahil's acid hematocrit method, Sahil's alkali hematocrit method, oxyhemoglobin method. Cyan's main hemoglobin method is more accurate because it detects all forms of hemoglobin, including sulfohemoglobin. Destruction of hemoglobin. How does destruction of hemoglobin takes place? Destruction of hemoglobin takes place in reticuloendothelial system of the spleen. Reticuloendothelial system of the spleen. For example, nice to know. Reticuloendothelial system. Here the term reticuloendothelial system is used to refer to certain phagocytic cells found normally. Reticuloendothelial system. Here the term reticuloendothelial system is used to refer to certain phagocytic cells found mainly in the bone marrow, liver, lymph nodes, spleen, etc. because they ingest large particles. The cells are called macrophages. Reticuloendothelial systems. Function of macrophages: they ingest and destroy RBC and form or release bilirubin. They also destroy leukocytes and platelets. They ingest bacteria and thus concerned with the defense of the body against infection. They ingest and process antigens with the stimulation antibody formation in plasma cell. Formation of macrophage. What is bilirubin? Bilirubin is the greenish yellow pigment which is a major and production of hemoglobin degradation. Excreted in bile and then eliminated in feces. Excreted in bile and eliminated in feces. It is responsible for the yellowish urine and yellowish discoloration of in jaundice. What are the causes of excess serum bilirubin? Metabolism. Normal level of serum bilirubin 0.3 to 1 milligram per dl. Daily production 250 to 150 milligram per day. Daily production. Normal level of serum bilirubin and daily production 250 to 350 gram per dl. Source of bilirubin mainly formed hemoglobin. Ineffective erythropoiesis. Catabolism of other heme containing proteins such as bilirubin and protein. Steps of bilirubin metabolism. Formation of bilirubin in reticuloendothelial cells. Transport of bilirubin within albumin. Hepatic uptake of conjugation. Biliary excretion and events in intestine. Fate of RBC. After passing 24 days in circulation, RBC are destroyed by endothelial systems. HB is liberated and the fate of HB is follows. Fragile RBC. Destruction by reticuloendothelial system. Hemoglobin. Phagocytosis by macrophage. Forms coleoglobin. Fragile RBC. Hemoglobin forms coleoglobin by making by one of the methane bridges, splitting into macrophage globin and then heme into bilivardin reductase, heme oxide, and then bilirubin combines with albumin and albumin gets to circulation. Liver glucuronic acid glucuronic acid GLU bilirubin glucuronyl transferase. Bilirubin D-glucuronide. So liver and bilirubin D-glucuronide, and then bacterial action intestine. Urobilirogen kidney. intestine, and then urobilirogen. Urobilirogen converts into stercobilin. Urobilirogen then into stercobilinogen. Urobilinogen, stercobilinogen, urobilinogen, and then urobilin, and then stercobilin to feces and to urine. What do you mean by fragility of RBC? The tendency of RBC to disintegrate is called the fragility of RBC. Factors increased in the fragility of RBC increased. Chemical agents, bacterial agents, viral agents, specific hemolysis, and then snake venom, venom from stinging insect and spider, and some kind of anemia. In clinical agents, chloroform, either alcohol, bile salt, alkali, poisonous chemicals such as neuroenzine. Bacterial agents, streptococcus, and then tenus bacilli. Viral agents, smallpox, measles. Specific hemolysis, mismatched blood transfusion, and then snake venom such as cobra venom. Clinical agents, bacterial agents, viral agents, specific hemolysis, snake venom. Venom from stinging insects, and some type of anemia. Bacterial, viral, specific hemolysis, snake venom, chemical agents. Some agents, for example, sickle cell anemia, idiopathic hematopoietic anemia, and pernicious anemia. Importance of measurements of red blood cell osmotic fragility. Measurements of red blood cell osmotic fragility provides a useful indication as to whether a patient's red cells are normal or abnormal. Osmotic fragility principle: red cells that are spherocytes for whatever cause take up less water in an photo 6 ionic solution. Principle osmotic fragility before rupturing than do normal red cells. Measurement of red cells osmotic fragility provides a useful indication as to whether a patient's red cells are normal or abnormal. In HCl solution and aggregate, the test is normally carried by room temperature 15 to 25. RBC lysesters at 0.5%, membrane 50% lysed at 0.4 to 0.43%. Membrane is completely lysed is 0.3% HCl. Importance: the test gives an indication on the surface area volume of the ratio of erythrocytosis. The greatest usefulness in this diagnosis of hereditary spherocytosis. The test may be used to screening for thalassemia. Osmotic fragility increased in hereditary spherocytosis, autoimmune hemolytic anemia. Osmotic fragility decreased in thalassemia major and osmotic fragility, thalassemia major and sickle cell anemia. Why do RBC swell and eventually burst when they are placed in a solution 3.9 solution? Osmotic pressure, and so when RBC are placed in solution 0.9 Cl, whether water or RBC swells or bursts. What happens when RBC is suspended in isotonic and water solution? Isotonic solution. Shape of RBC remains intact because there is no gain or loss of water. Hypotonic solution. Water enters into RBC by osmosis and RBC swells, becomes spherical, and eventually bursts. Water enters into RBC. Hypertonic. The RBC shrinks due to loss of water from it by osmosis. Define jaundice. Jaundice is a clinical condition characterized by the yellow discoloration of the skin, sclera, and mucous membrane of the body due to presence of excessive amount of bilirubin in blood. Jaundice is yellow coloration of skin, conjunctive, and other tissue caused by the presence of an excess bilirubin in the plasma tissue fluid. Normal serum bilirubin 0.3 to 1 microgram per deciliter. When bilirubin plasma exceeds 3 gram per deciliter. Latent jaundice. Clinically, there is no jaundice, but serum bilirubin is more than normal, but less than 3 milligram per deciliter. Pathological classification: predominantly conjugated or hyperbilirubinemia and predominantly conjugated hyperbilirubinemia. Predominantly unconjugated and then conjugated. Pathological classification and celiopathological classification. Clinical pathological classification. Pathological classification: predominantly unconjugated hyperbilirubinemia, excess production of bilirubin, hemolytic, reduced hepatic intake, impaired bilirubin conjugation. reduced hepatic intake or impaired bilirubin conjugation, predominantly unconjugated bilirubinemia, intrahepatic or extrahepatic cholestasis. Clinicopathological classification. This is caused by the excessive breakdown RBC with rapid release of bilirubin into blood. Causes hemolytic anemia, conjugation, typical RBC, congenital enzyme defect, incompletable blood transfusion, malaria, certain drugs, snake venom, hemolytic erythroblastoma fetalis. So there are hemolytic anemia, and then sickle cell anemia, and then incomplete blood transfusions, malaria, and certain drugs, and snake venom erythroblastoma fetalis. Hepatocellular hepatic jaundice. Damage in the liver cells can cause unconjugated bilirubin level to increase in the blood as a result of decreased conjugation. Damage in liver cells can cause unconjugated bilirubin level to increase in blood as a result of decreased conjugation. Viral hepatitis, chronic alcoholic hepatitis, liver cirrhosis. Chronic hepatitis, liver cirrhosis. Infections, poisons, and paracetamol poisoning. Cardiac failure. And obstructive includes causes viral hepatitis, drug-induced alcohol, primary bilirubin carcinosis, secondary bilirubin carcinosis, pregnancy, post-operative, extrahepatic cholestasis, gallstone, stone in common bile duct, carcinoma in common bile duct, ampulla of Vater and head of pancreas, inflammation, inflammatory structures of bile duct, cyst in bile ducts, parasitic infection, and paratraumatic biliary constrictions. Viva. Sclera of the eyes. Sites of observation exercise. Ventral surface of the tongue. Ventral surface of the tongue. Tip of the nose. Buccal mucous membrane. Buccal mucous is skin of the palm and the hand. Skin of the sole and the foot. Extravenic cholestasis, gallstone, cystic bile, parasitic infections, traumatic biliary structures. Why jaundice is best seen in these areas? These areas are rich in elastic fiber, and bilirubin deposits in elastic fiber due to affinity elastic fiber. What is physiological jaundice? Physiological jaundice. Physiological jaundice. Physiological jaundice is a common condition of newborns and occurs in most newborns. This mild jaundice is due to immaturity in the baby's liver, which leads to a slow process of bilirubin. It generally appears at 3 to 4 days at the age and disappears 1 to 2 weeks. It's harmless and does not actually cause a problem. Normally, bilirubin passes through the liver and excretes in bile through the intestine. Jaundice occurs when bilirubin binds up faster than newborn's liver can break it down and pass it from the body. Reasons include newborns make more bilirubin than adults since they have more turnover of red cells. A newborn is still developing liver may not be able to adequate bilirubin for the blood. Too large of an amount of bilirubin is reabsorbed from the intestine before baby gets rid of it in the stool of the intestine. Treatment is usually not needed. For most babies, jaundice usually gets better without treatment within 1 to 2 weeks. Sometimes phototherapy is needed for infants whose bilirubin levels are high. Possible complications. Rare but serious complications from high bilirubin levels include cerebral palsy, deafness, and kernicterus. What is echoleutic jaundice? Chronic jaundice. Jaundice with presence of bilirubin in urine is called chronic jaundice. So hepatitis and obstructive of the common bile duct causes chronic jaundice. Acholuronic jaundice. Jaundice without presence of bilirubin in urine is called acholuronic jaundice. Hemolytic jaundice is acholuronic jaundice. Hemolytic jaundice. Jaundice without presence of bilirubin in urine. Jaundice with presence of bilirubin in urine. So hepatitis and obstruction of common bile duct or hemolytic jaundice. So chloronic jaundice and achloronic jaundice. Achloronic jaundice. State the difference between conjugate and unconjugate bilirubin. Unconjugate, conjugate. Presence in a healthy person, unconjugate bilirubin, conjugate bilirubin. Absence in blood in a healthy person. Present in blood in healthy person, absent in blood, conjugate. Soluble, water soluble, water insoluble, water soluble. In a healthy person, present in blood in healthy person, absent in blood in healthy person, conjugate. Water insoluble, unconjugate base. Lipid soluble and thus can cause damage in CNS. Central nervous system. Cannot excrete through urine, unconjugate. Cannot excrete through urine, can pass through urine, conjugate base. Can pass through urine. Tightly bind with plasma albumin, weakly associated with albumin. Indirectly positive, direct or immediate positive. Mostly found in hemolytic jaundice, most found in hepatocellular obstructive jaundice. Name some important tests which can differentiate between prehepatic and posthepatic jaundice. List the investigations used in clinical practice for diagnosis of jaundice patient assessing liver function. Blood biochemistry, serum bilirubin. Hemolytic, everything is normal hemolytic, but serum bilirubin raised, mostly unconjugated. Conjugated. Hepatocellular jaundice, everything is raised. Reduced is serum bilirubin. And then obstructive bilirubin, everything is high and raised. Serum bilirubin is low in hepatocellular. Urine, bilirubin absent, present, present. Hepatocellular obstructive jaundice present. Hemolytic jaundice absent, bilirubin. Bile salts absent. Urobilirubin absent, increased. Van der Brugge reaction indirect positive. Hepatocellular jaundice, bilirubin present, present. And obstructive jaundice present, present. Urobilirubin decreased, normal or increased. Stool, stercobilirubin increased, reduced or absent. Naked eye, dark brown color, normal color, and then pale, normal color, or pale or normal color, or bulky due to high fat content. And feces fat absent, increased. Specific blood tests. Hemolytic jaundice, hepatocellular jaundice, and obstructive jaundice. Viral marks. Not found, not found. Not found. What are the investigations you will do for diagnosis of jaundice? What are the investigations you will do for diagnosis of jaundice? Serum unconjugate bilirubin. Serum conjugate bilirubin, urine bilirubin, liver function test, serum alanine test, and then serum aspartate transamine, serum alkaline, and then serum globulin test. Conjugal coagulation test, prothrombin coagulation test, prothrombin test, ALT test, ALP test, AST test, serum alkaline test, serum conjugate bilirubin test, serum conjugate bilirubin, unconjugate bilirubin, serum conjugate bilirubin. Mention the indications of liver functions. Liver function test. The liver functions tests are the biochemical investigations to assess the capability of the liver to carry out any other function performs. Major liver function tests are liver function tests for excretory functions, tests for enzyme functions, tests for metabolic functions, tests for synthetic functions. Prothrombin time, test for synthesis prothrombin time, and then total plasma protein time total plasma protein as constant serum and bilirubin level, albumin and globulin ratio. Indications of liver function test: to detect hepatic abnormalities, to measure the sensitivity of liver damage, to identify specific causes, to investigate possible complications. Investigations can differ between free bilirubin and conjugate bilirubin. Free bilirubin, conjugate bilirubin. Pan process. Bilirubin reacts to dialysed sulfuric acids. Conjugate bilirubin. A 35-year-old male patient presents with feeling very tired and vomiting. Patients have a history of taking street food 15 days back. On examination, yellow skin and sclera are hepatomegaly. What are the possible diagnoses of this condition justify it? Acute viral hepatitis A or justification, fatigue, anxiety, vomiting, hepatomegaly, inflamed or swollen liver infections. Tests for confirming the diagnosis: liver function test, serum bilirubin ALT, serum bilirubin ALT, AST. Serological test: hemoglobin M and hemoglobin M, hepatitis A, hepatitis E, HAV, hemoglobin M antibodies, HEB antibodies. Complete blood count CBC, to show leukopenia or leukocytosis. Ultrasound and abdomen due to liver size. Cause of hepatomegaly in condition: caused by inflammation or swelling, inflammatory response leads to edema, mutations in immune cells and cell damage. Sites to observe jaundice clinically: sclera of the eyes, ventral surface of the tongue, tip of the nose, buccal mouth membrane, skin and palm of the head, skin of the sole of the foot. Iron metabolism: absorption of iron from GIT, breakdown of RBC, transport of iron, utilization of iron, storage of iron, loss of iron from body. Iron is mostly found in foods of Fe3+ form, reduced to Fe2+ by gastric HCl, gastric vitamin C, reduced to H2 by vitamin C and HCl. Fe2+ is soluble in readily absorbed erythrocytes and breakdown of WBC. Transport of iron, breakdown of iron, utilization of iron, storage of iron, liver, spleen and bone marrow. Loss of iron: about 0.3 mg iron is lost per day by bile salt. A woman during menstruation loses 1.3 grams of iron per day. Parameters for assessing the iron stored in the body: serum ferritin, serum iron, total iron by iron capacity, percentage of saturation of iron binding protein. Mucus block theory: the iron absorption requires apotransferrin secreted with the bile. So in a person take large amount of iron, all irons cannot be absorbed through the intestinal mucosa, because after saturation all apotransferrin, there will be no more apotransferrin to bind with iron. This is called mucus block theory. Iron absorption requires apotransferrin secreted with bile, apotransferrin. Large amount of iron due to this is called mucosal block theory. So if a person takes large amount of iron, all iron cannot be absorbed through intestinal mucosa, bile, because saturation of all apotransferrin, there will be no more apotransferrin to bind with iron. Storage and iron is stored mostly in liver as ferritin or hemosiderin. Iron is stored in liver, spleen and bone marrow, ferritin, and protein apoferrin. And when it is supplied, where the supply of iron is excess and body demands iron and soluble called hemosiderin of the spleen. When the supply of iron is in excess, the body demands iron to is stored in insoluble called from hemosiderin of the liver. So spleen and bone marrow, the ferritin and then hemosiderin. some iron content food in the rural area. Drinking water, tubal, kochu shak, green banana, green leaf vegetables, egg, liver, etc. Total amount of iron. Hemoglobin 65%, 4%, 1%, 0.1%. What is apotransferrin and apoferritin? The apotransferrin is a beta globulin of blood plasma which is combines with form transferrin. Apotransferrin is a cytoplasm protein which iron combines with this ferritin. Explain the feedback mechanism for regulation of iron absorption. When body stores of iron is increased, intestinal iron absorption is decreased. When body store of iron is increased, body store of absorption is decreased. When the body store iron is decreased, the intestinal iron absorption is increased. White blood cell classification of white blood cell on the basis of presence of granules of cytoplasm. WBC are of two types: granules having granules of cytoplasm, neutrophil, eosinophil, and basophil; and granules having no granules in cytoplasm, large lymphocytes and small lymphocytes, monocytes. What do you mean by different count of WBC? State the different count of WBC and WBC. Count of WBC: 4,000 to 11,000 cells per cubic millimeter of blood. Different count of WBC. Determination of number of each variety of WBC in 1 liter of blood is called differential count of WBC. 4,000 to 11,000 cubic millimeter of blood. Determination of blood of each variety of WBC is 1 liter of blood is called differential count of WBC. So eosinophil different counts: 50 to 70% neutrophil, 3,000 to 6,000. Lymphocyte 20 to 40%, 150 1500 to 2700. Neutrophil, lymphocyte, and then monocyte 2 to 8%, 300 to 600. Eosinophil 1 to 4%, 150 to 300. Basophil 0.1%, 1 to 100. Duration of normal count RBC. Diurnal. Total count rises day by day to day. The morning or after rest, the count is lowest. After midday, the count increases and highest in the evening. Muscular exercise. Any condition leading to hypoxia increases total count. Relation with meal. After meal, lysolysis occurs. Leukocytes occur. Adrenal injection increase the count. Adrenal injection. Emotional stage. Fear, pain, and increase of adrenal secretion, thus hemoglobin increase. WBC increase. Age. The newborn baby has much WBC count. Duration pregnancy. In pregnancy, the count is higher. What is leukocytosis? Leukocytosis is a clinical condition characterized by the increase of WBC in circulation above normal age. Is a clinical characterized by increased number of WBC in circulation above normal range, more than 11,000 cells per cubic millimeter of blood. Neutrophilia is the increase in total number of neutrophils above 6,000 millimeter cube. Neutrophilia is the increase in total number of neutrophils 6,000. 11,000 is leukocytes. Physiological causes. Physical exercise after having meal, pregnancy and parturition, emotional stress, cold bath, diurnal variation. Pathological causes. Neutropathic leukocytosis, acute bacterial infection, tissue injury due to burn, surgery, etc. Hemorrhage, neoplasia, inflammatory disorders such as certain collagen disorders, gout, rheumatic fever. Eosinophilic leukocytosis, allergic reactions, basophilic cytosis, polycythemia, monocytes, chronic bacterial infections, lymphocyte, bacterial disease, viral disease, and then protozoal disease, and Epstein-Barr disease, bacterial disease, syphilis, etc., viral disease, viral infection, etc., and protozoal disease, Kala-Azar, etc. Define leukopenia and neutropenia. Leukopenia, a clinical condition characterized by decreased number of WBC in circulation, below normal range, less than 4,000 cells per cubic millimeter of blood. Leukopenia, a clinical condition characterized by decreased number of WBC circulation, less than 4,000, more than 11,000, less than 4,000. Neutropenia, it is a decreased neutropenia, it is decreased number of neutrophils below the lower limit of absolute 1500 cells millimeter cube of circulating in the blood. So more than 6,000 and more than 11,000, less than 4,000, less than 1500. Cause of leukopenia: viral infections, protozoal infection, aplastic anemia, megaloblastic anemia, hypersplenism, drugs, and excessive irritation, and miscellaneous antimalarial toxic. Give the morphology of different types of leukocytosis. Neutrophil size 5 to cell size 9 to 5, eosinophil 10 to 14, basophil 10 to 9 to 8, monocyte 5 to 8, and then lymphocytes large 15 to 18, 12 to 15, and 6 to 10. Round, reddish brown, reddish, purple blue, pale blue. So pale blue, purple blue, and then reddish brown, reddish, purple blue, pale blue. Multi-lobed, connected by chromine filament, bilobed or trilobed. Bilobed, horseshoe shaped, rounded, multinucleated. Cytoplasm filled with fine reddish brown granules, uniformly distributed, filled with coarse red granules, uniformly distributed, filled with coarse purple blue granules, granescent. Periphery situated, bluish cytoplasm. And then enumerate the function of WBC. Phagocytosis, antibody formation, secretion of heparin, formation of fibroblast, antihistamine, and then chemotaxis acts as a scavenger. In function, each type of WBC: neutrophil. Phagocytosis, acute inflammation, screen platelet factors, PAF, liberates chronic factors for macrophage, have unique enzyme system, eosinophil, defense against parasitic infection, immediate hypersensitivity reaction, inactivation of histamines, release of fibrolysis which dissolves fibrin clot. Neutrophil, eosinophil, and basophil produces heparin in which prevents intravascular clotting. Lymphocytes, T lymphocytes are identify and destroy foreign particles and responsible for cell-mediated immunity. Monocytes, they enter the blood and bone marrow and articulate for 32 hours. They then migrate to tissue and become macrophages, Kupffer cells, antigens, monocytes, and presenting cells, lymphocytes. So function of WBC is neutrophil phagocytosis, acute inflammation, defense mechanism, and then hypersensitivity using basophil, heparin, lymphocytes and humoral immunity, T lymphocytes and B lymphocytes, and then can identify sort of foreign particles. Cells mediated immunity, monocytes. They enter the blood from bone marrow and circulate about 32 hours. They migrate to tissue and become macrophages, Kupffer cells, and antigen presenting cells. Macrophages, Kupffer cell, antigen presenting cells, and continue to proliferate many years. Acute inflammatory response secretes PGE, clot formation, lysis of tumor cells. Briefly describe the properties of WBC. The properties of WBC. Margination. Speaking to WBC vascular wall, margination. Diaperesis. The process by which granuleucocytes come out of lumen of blood vessels into the tissue space is called diaperesis. Mechanism: neutrophils of the monocytes adhere to the endothelium and insinuate themselves through the gap between the adjacent endothelial cells, finally squeezing out of the vessel. Amboid movement: a special type of movement by which neutrophils and macrophages move towards the damaged tissue by producing pseudopod. Maximum rate is 40 micrometer per minute. Mechanism: protrusion of pseudopod from one end to cell. Pseudopod projects far away from cell body. The remainder of the cell moves towards the pseudopod. Chemotaxis: a number of different chemical substances liberated by the damaged cells cause leukocytes, neutrophil and macrophages to move either towards or away from the source of the chemical substance. A special movement 40 microns per minute, and chemical substances damage tissue leukocytes to move either forward or away from the source of the chemical substance. It's called chemotaxis. So the chemical substances are some of the bacterial and viral toxins, degenerative products of inflammatory tissue, components of the complementary system and complementary C5a. So complementary system, complementary system, particularly C5a, and then cytokinase. For example, IL-8, cytokinase, products of blood clotting. Cytokinase, IL-8. Phagocytosis, the process by which phagocytic cells, mainly neutrophil and macrophages, recognize and then engulf, dispose of foreign particles, is called phagocytosis. Steps of phagocytosis has three steps. Recognition and attachment. Neutrophil and macrophages first recognize the particle engulfed, attached by surface. The efficient phagocytosis is greatly enhanced when the microbes opsonized by specific opsonin proteins. Major opsonins are immunoglobulin G antibodies, complement proteins, and then certain plasma lectins. Engulf: after attaching phagocytic projects pseudopods around the foreign body and then completely enclosing it. This creates an enclosed membrane-bound cytoplasm called phagocytosome. Phagocytosomes fuse with lysosome to form phagolysosome, killing the degradation. After engulfing, killing occurs in two ways: either oxygen-dependent free radical mediated killing and oxygen-dependent lysosome enzyme mediated killing. Oxygen-dependent free radical mediated dependent and lysosome enzyme mediated. So what role of lysosome in the phagocytosis? After recognition of attachment, phagocytes and macrophages project pseudopods in all directions around the foreign body. Pseudopods meet another on the opposite side. Used together, complete enclose it. It creates an enclosed membrane and free phagocytic vesicles. Phagosome fuses with lysosome to form phagolysosome, thereby dumping away lysosomal digestive enzymes and bacterial agents into phagocytic vesicles. Thus, phagocytic vesicles now become digestive vesicles, and digestion of the phagocytosis particles begins immediately by the intercellular enzymes. Antibodies coat the bacteria, making them more susceptible to phagocytosis by macrophages that engulf the bacterium, bringing it into the cell and forming phagosome. Lysosomes then attach to the phagosome to form a phagolysosome, which digests the invading pathogen. The phagocytic cell then releases the digestive products of exocytosis. Nice to know. Both neutrophils and macrophages contain an abundance of lysosomes filled with proteolytic enzymes, especially geared for digestive and bacterial and other foreign protein matter. The lysosomes of macrophages, not neutrophils, are also contain some large amount of lipase, which digest the thick lipid membrane processed by some bacteria, such as tuberculosis bacteria. Vesicles. Lysosomes to form phagolysosomes. Thus, phagolysosomes digest particular in intracellular enzymes. Define opsonization and opsoning. Opsonization refers to the attachment of antibody molecules to the surface of phagocytes by the help of some special molecules called opsonins. Phagocytosis of bacteria aided by the presence of substances in plasma are called opsonins. The phagocytic bacteria aided by a presence of substance in plasma called opsonins. Opsonins are the substances which attach antibodies to phagocytes. Examples: immunological and particular class immunoglobulin G, C6C3b, C5a, and then C3b component of complement, plasma lactins, C-reactive protein, and fibrinogen. These are all opsonins. Name 3 killing agents of bacteria which are found inside the bacteria invaded neutrophil. 3 killing a part of bacteria which are formed inside the bacteria invade neutrophil. Superoxide hydrogen peroxide hydrogen ion. Nice to know. A single neutrophil can easily phagocyte 3 to 10 bacteria before the neutrophil becomes inactive and dies. Macrophages are much more powerful phagocytes than neutrophils are, often capable of phagolysis in many 100 bacteria, and also have ability to engulf large particles, even whole WBCs, occasionally malaria parasites. Whereas neutrophils are not capable of phagocytosis. Particles much larger than bacteria. Also, after digestion, bacteria macrophages can exclude the residual products and often survive and function for many more months. Macrophages are much more powerful than neutrophils. Short note: neutrophils are the commonest cells of WBC. Pluripotent hematopoietic stem cell in bone marrow. Half-life. Average half-life of the neutrophil in circulation is 6 hours. Differential count 50 to 70%. 611,000. 9 to 12. Round shape, reddish brown. Multi-lobe, filled with reddish brown granules. Uniformly distributed. Functions of neutrophils: phagolysosome, acute inflammation, separate plates, activation factor, liberates chromatic factors in macrophage, having unique enzyme system. What is macrophage? Macrophages are the large white blood cells whose function to the phagocyte cellular debris and pathogens. Macrophages are the large white blood cells whose function is to phagocyte cellular debris and pathogens. Diameter 10 to 30 micrometer, oval and kidney shape. Macrophages arise from blood monocytes. During macrophages and necrotic cell in the body. skin, Langerhans cell, liver, Kupffer cell, brain, microglia, lung, alveolar macrophages, bone, osteoclast, kidney, mesangial cells, lymph node, dendritic cells, kidney mesangial cells, dendritic cells, spleen macrophages, and red pulp macrophages. The skin, liver, brain, and then lung, and then bone, and then kidney. Functions: phagocytosis, ingestion and killing of microorganisms damage that cells or cellular decrease. Secretion of various substances, for example, IL-1, IL-8, T and F. These are all, and lymphocytes activation via antigen presentation. Regulation of hematopoiesis, killing of tumor cells, tissue repair and remodeling by collagen and elastase. Mononuclear phagocytic system, the combination of monocytes, mobile macrophages, fixed tissue macrophages, and a few specialized endothelial cells in the bone marrow, spleen, and lymph node is called mononuclear phagocytic system. It is sometimes called reticuloendothelial system. Mononuclear phagocytic system, reticuloendothelial system, reticular reticuloendothelial system, or monocytophage system, all of them having the same capability of phagocytic bacteria, virus, and neutrophil. Thus, tissues of the foreign tissues. Mononuclear phagocytic color is from common precursor of bone marrow, which give rise to the bone monocytes. Some monocytes migrate to various tissues and differentiate macrophages. The system includes skin, Langerhans cells, spleen and lymph node, sinus, histiocytes, lungs, alveolar macrophages, liver, Kupffer cell, CNS microglia, bone marrow, osteoclast. Skin, spleen, lung, liver, CNS, and then bone marrow. Sinus, histiocyte, spleen, Langerhans cell, sinus, histiocyte, lung, alveolar macrophage, microglia, and osteoclast. Kidney mesangial cells joined synoviocyte. Name some tissue macrophages. Tissue macrophages are specialized cells that reside in various tissues throughout the body and play key roles in immune defenses, tissue repair, hemostasis. They are derived from monocytes in the bloodstream and differentiate into macrophages when they enter tissue. Below it is a line of some of the major tissue macrophages. Tissue macrophages location: microglia, brain and spinal cord, Kupffer cell, liver, alveolar macrophages, lungs, osteoclast, bone, Langerhans cells, skin, tissue histiocyte, lymph node and spleen, brain and cord, liver, and then joints, peritoneal macrophages, abnormal cavity, intestinal macrophages, gut, mesangial cells, glomerulus, cardiac macrophages, heart, adipose tissue macrophages, fat tissue, ciliary macrophages, eye, meningeal macrophages, meninges. Mast cells are heavily granulated cells that connective tissue that derive from bone marrow precursors and are characterized by abundant basophilic cytoplasmic granules. Shape: oval and round, 20 to 30 micrometer. Filled with cytoplasmic granules. Nucleoplasm is small spherical nuclei entirely situated. Consists of mast cell granules and their functions. Heparin. They can locally act as anticoagulant. Heparin. Histamine. Can promote increased vascular permeability, smooth muscle contraction. Bradykinin, sarcoptenin, and serine protease and leukotrienes. C4, D4, and E4, slow reacting substance of anaphylaxis, which are also triggered by smooth muscle contraction. Mast cell granules when allergens bind to hemoglobin E molecules directed against that previously coat of mast cell surface. Mast cell degeneration produces clinical manifestations of allergy up to this, including anaphylaxis. Distribution of mast cell, dermis of the skin, mucosal limit, digestion, the respiratory tract, liver capsule, and then also in the blood vessels. Immunity. Vast network of molecules of cells which primarily protect the vertebrates against microorganisms is called the immunity system. Large number of network of molecule or cells, which primarily protect the vertebrates, vertebrates, vertebrates against mono microorganisms is called immune system. Complement of immune system composed of lymphoid organs, aggregation of lymphoid tissue and non-lymphoid organs, lymphocyte of blood and lymph. Define immunity. Human body has the ability to resist almost all the organisms and toxins that tend to damage the tissue and organ. This capability of human body immune system is known as immunity. It is the inherent ability of living bodies to resist almost all type of living organ agents. Types of immunity: innate natural immunity and acquired adaptive immunity. Innate immunity is the resistance that exists in prior to exposure to microbes or antigen. It is immediate non-specific response develops to general process of direct solely against the specific disease organisms. It induces host defenses such as innate skin and mucous membrane, certain cells, HCl and digestive enzymes, certain proteins. Includes host defenses. Major functions of innate immunity: killing invading microbes, activating adaptive immune process. Acquired immunity. Acquired adaptive immune occurs after exposure to antigen, improves upon repeated exposure, and is specific, is mediated by antibody produced by B lymphocytes and two types of T lymphocytes, namely helper T cell and cytotoxin T cell. Helper T cell and cytotoxin T cell. Acquired immunity. Helper T cell and cytotoxin T cell. It is acquired immunity after exposure to agent improves upon related response and specific. It is mediated by antibody produced by B lymphocytes and by two types of T lymphocytes, B lymphocytes and two types of T lymphocytes, namely helper T cell and cytotoxin T cell. Helper T cell, cytotoxin T cell. Two basic types of acquired immunity occurs in the body. Humoral immunity, cell-mediated. Humoral immunity is due to circulating antibodies and is a major defense against bacterial infections. Cell-mediated immunity, it is due to immunological components of T lymphocytes and is responsible for a variety of important cell-mediated immunological process such as defense against viral infection, cell allograft projection, defense against cancer cell. Immunity, natural, acquired. Acquired humoral immunity, cell-mediated immunity. Acquired. Different types of T lymphocytes and in the body. Cells, lymphocytes, neutrophil and lymphocytes. Large, rounded, pale blue, peripheral, medial. And then types of lymphocytes, T and B. T lymphocytes. 70 to 75% of total lymphocyte population develops in bone marrow, mature thymus, and it derives into blood groups. Helper T cell. They help in immune system by forming a series of protein, mediator, and lymphokinase. Stimulate growth and proliferate cytotoxin and suppressor T cell. Stimulate growth and proliferate B lymphocytes. Helper T cell. Cytotoxin T cell and helper T cell. Cytotoxin T cell directly attacks cell capable of killing virus infected cells, engulf cells and tumor cells. some of the body's own cells in autoimmune disease. These cells are also called killer cells. Suppressor T cell suppresses the function of cytotoxin T cell. Granule T cell ability to system attack and immune tolerant. Memory T cell function persists for long period, responsible for accelerated response and second exposed to the same antigen. B lymphocytes. 25 to 25% of all lymphocytes function can differentiate plasma and produce antibody and thus responsible for humoral immunity. Short note, helper T cell, they are called so because they help in function of immune systems in many ways. They are more than 75% among the T cells. They are inactivated and destroyed by HIV and AIDS function. They help in immune system and formation of series lymphokinase. It stimulates growth and proliferation of cytotoxin and presence of T cells. It stimulates growth and proliferation of B cells, plasma and antibodies. B lymphocytes. Origin stem called bone marrow maturation in the bone marrow. Nomenclature. B cells were the first discovered to birds where the maturation B cells occurs in a bruise of fibrosa. Nomenclature. B cells were first discovered in birds. For this reason, the B lymphocytes disseminate on the role of the brusa. For this reason, B lymphocytes are disseminate the role of brusa. Receptor B cells having hemoglobin M, hemoglobin D functions. B cells are responsible for humoral immunity. B cells differentiate memory B cells and B cells act as antigen presenting cells. Antigen presenting cells APC. What is the antigen presenting cell? Antigen present the immunocompetent cell which antigens helper T cells are called immunocompetent. The immunocompetent cell which presents antigen is called helper T cell are called antigen presenting cell. Macrophages, B lymphocytes and dendritic cells. Innate, acquired, inborn, non-specific immune absence, present macrophages. And then what are the types of immune response? Primary immune response caused formerly by antibody or first exposure to specific region. Secondary immune response, antibody from second exposure to the same reagent. Potency during is more in secondary response. It is attributed to presence of antigen specific memory cells after first contact. How cell mediated immunity is produced in our body. CMI has two distinct components, helper T cell and macrophages are all our main defense against intracellular bacteria such as Mycobacterium and tuberculosum and fungi. If an offending agent has a bacteria enters the body, it is ingested by macrophages. The bacterium is broken down into fragments and called antigen appears on the surface of the macrophages which is present a helper T cell. This is called AG presentation. And then duration of AG during AG and then IL-1 and helper 1 cell and then virus and tumor cells allograft activation of T cell and AL2 and then activated cells viral antigens cytotoxin T cells then kill virus infecting cells. As a result they are killed. How is antibody mediated immunity produced in our body? During AG presentation macrophages secrete AL1 which helps in T cell. The activated helper T cells secretes IL4 and IL5. Plasma cells and 5 causes proliferation and antigen specific T cell difference B cells to plasma cells and memory B cells. The plasma cells produce and secrete large amount of antibody specific antigens. The memory B cells remain in blood for a loose period. Due to presence of plasma cells, HB appears rapidly in secondary immune response. Short note. Define antigen. Antigen is the substance, usually protein or polysaccharide, substance, protein or polysaccharide. Protein or polysaccharide in nature, which when introduced in a suitable animal through a suitable route processes antibody with which it reacts specifically. Molecular weight 1600,000, 160,000 to 970,000. Mechanism of antibody and what is immunoglobulins? The antigens are gamma globulins called immunoglobulins. They are usually constitute 80% of plasma proteins. Albumin, globulin, albinogen, and prothrombin. And classification in 5 classes: hemoglobin G, A, M, E, D. Hemoglobin G opsonizes bacteria. Immunoglobulin A attacks bacteria and virus. Immunoglobulin M complement system. Immunoglobulin E immediate hypersensitivity. Immunoglobulin B functions is uncertain. And then inflammation. Inflammation is the complex reaction to injurious agents such as microbes and damaged, usually necrotic. Inflammation is a complex reaction to injurious agents such as microbes and damaged, usually necrotic cells that consist of vascular spasm, regulation and activation of leukocytes, and systemic reactions. Vascular response, migration and vascular leukocytes, and systemic reactions. Characteristics of inflammation: vasodilation, and then increased permeability capillaries, opening of clot, and migration of large number of granulocytes, swelling of tissue. Types of inflammation: acute inflammation, chronic inflammation. Sign of inflammation: rudor, tumor, pallor, dolor, and then functional pizza. Signs of inflammation are redness, rudor, swelling, tumor, heat, calor, rudor, tremor, tumor, calor, dolor is pain, and then functionalisa, loss of function, functionalisa, dolor, calor, tumor, rubor. And the first line of defense: tissue macrophage. Second line of tissue macrophage, neutrophil. Third line of defense: second macrophage inflammation area. Fourth line of defense: increased production of granulocytes and monocytes in bone marrow. State the role of neutrophil and acute inflammation. Mainly the neutrophils that attack and destroy invading bacteria, virus, and other injurious agents. The neutrophil can attack and destroy bacteria even in circulating blood. Short note: triple response. The three response: red reaction, the wheal or flare of the skin of an injury is called triple reaction. Three reactions: the red reaction, the wheal and flare, the skin red reaction, and the initial redness due to capillary dilation is direct response to the capillary pressure. The swelling, the wheal, and the local edema produced by permeability. Swelling, wheal, capillary compression, the redness from injury, and the redness from injury. Flare, 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 and the flare. So the red reaction, wheal and flare. Red reaction, wheal and flare. Red reaction. The skin is stroked more firmly with potent instruments, and reddish outside appearance for 10 seconds. Right? And then allergy and hypersensitivity. Hypersensitivity may be defined as inappropriate or excessive immune response to an antigen stimulating in a pre-synthesized host leading to tissue damage. May be defined as an inappropriate or excessive immune response to an antigen stimulus in an pre-synthesized host leading to tissue damage. Combs or gel classification on basis of onset action on basis of mechanism. Give examples of hypersensitivity reactions. Immediate hypersensitivity, antibody mediated hypersensitivity, for example, asthma, anaphylaxis, and then HBO incompatibility antibody mediated, immediate, and then immune complex mediated, serum sickness, arthritis, and then cell mediated, tuberculosis, contract damage, multiple sclerosis, type 1 diabetes. Define atopy. Atopy is a genetically determined state of type 1 hypersensitivity to common environmental agents mediated by immunoglobulin G. Asthma is a genetically determined state of type 1 hypersensitivity. Type 1 hypersensitivity to common environmental agents. Graft. Autograft classification of graft is a transplant from one area to another area of the same individual. Transfer of skin of some other individual. Isograft, a graft between two genetically identical individuals as identical twins. Transfer of kidney and other organs. Autograft. Allograft, the graft between two genetically dissimilar individuals but same species, one person another. Allografts. And then xanthografts are heterografts between a donor and a recipient from different species, like a transplant heart from monkey to human. Platelets. Thrombocyte, normal count of platelets. Platelet thrombocyte, normal count platelet. Fifteen 150,000 to 400,000, 150,000 to 400,000 microliter of blood, 150,000 to 300,000 millimeter cube of blood. Cell size 2 to 4 micrometer diameter. Cell shape spherical, oval shape, rod shape. Nucleus absent. Lifespan 8 to 12 days. Colorless. Thromboxane A. Chemical characteristics. Thromboxane A. Serotonin ADP. Thromboxane A2. Thromboxane A2 serotonin ADP. Blood clotting factors. Platelet derived growth factor PDGF. Platelet activating factor PAF. Function of platelet: hemostasis, blood coagulation, phagocytosis, and storage of transport substance. Write down the substance secreted on platelets and mention their importance. Substrate secreted from platelet and their importance. Thromboxane A2 is a highly potent platelet aggregating agents of a constrictor parietal muscle. And then statocognin. It causes vasoconstriction. Thromboxane a highly protein platelet aggregation. Thromboxane A2 and then vasoconstriction. Sarcotonin 5-HT. Sarcotonin ADP. Agglutination of platelets. Blood clotting factor 145. Platelet derived growth factor causes vascular endothelial cells. Platelet activating factor promotes aggregation of platelets. Inflammatory active. Cytoplasm of platelets. The cytoplasm of platelets contain actin, myosin, and glycine lysosomes and two types of granules. The dense granules which contain calcium and ATP, and then serotonin 5-HT. Serotonin dense granules. So the dense granules which contain calcium, ADP, serotonin that are second response to platelet activation. Dense granules: calcium, ADP, serotonin. Calcium ADP, serotonin, and that are secreted in response to platelet activation. Alpha granules which contain von Willebrand factor, PDGF, platelet derived growth factor, and fibrinogen. What is thrombocytopenia is defined as a reduction in peripheral blood platelet count below the lower limit of 1.5 lakhs millimeter cube of blood. Classification or cause of thrombocytopenia acquired. More common causes: idiopathic thrombocytopenia. Classification of thrombocytopenia acquired. Less common causes, rare cases, and neutral or congenital. Acquired. More common causes: idiopathic thrombocytopenia. Drugs and chemicals: leukemia, plasmatic anemia, both bone marrow inflammation and secondary hypersplenism. So idiopathic thrombocytopenic purpura, leukemia, aplastic anemia, hypersplenism. Less common causes: infections including HPV, megaloblastic macrocytic anemia, liver disease, alcoholism. Rare causes: thrombotic thrombocytopenic purpura, postpartum thrombocytopenia, food allergy, neurological immune infections, drug adhesions, and congenital, hereditary sex limited autosomal, congenital leukemia, and giant hemangioma. Hemostasis means spontaneous arrest of bleeding under physiological process when minor blood vessels are ruptured. Hemostasis means spontaneous arrest of bleeding under physiological process under minor blood vessels ruptured. Mechanism: vascular spasm, formation of blood coagulation, and growth factor, growth of fibrous tissue. Vasospasm, local myogenic contraction, nerve reflex, local humoral factors. Formation of platelet plaque, become sticky, adhere to subendothelial collagen factors. Endothelial tissue acts as glue. Platelet adhesion, and then secrete large quantities of ADP and thromboxane, A2, DP, and AX-A2. Thromboxane A2, thromboxane A2. Thromboxane A2. Vascular spasm, formation of platelet plaque coagulation. A2. Platelet activation, loose platelet plaque, blood coagulation. Secondary hemostatic plaque, prothrombin activator, thrombin and prothrombin into thrombin, fibrinogen into fibrin, growth of fibrous tissue, formation of permanent plaque by fibrin polymerization cross-linking, clot reaction and removal. State the important plaque formation. Importance of platelet plaque is critical in early stage of hemostasis to stop bleeding rapidly and size of vascular injury, thus preventing excessive blood loss and contributing to overall maintenance of vascular rigidity. What is von Willebrand factor? Von Willebrand factor. One factor articulating protein involves hemostasis synthesis of endothelial alpha granules of platelets. Laboratory findings: bleeding time increased due to platelet defect and APTT. Active peripheral thromboxane time due to factor 8 deficiency. Laboratory findings: what are the source of blood clotting factors? Liver, platelets 15145, endothelium, and macrophages. Name the primary blood clotting factors: fibrinogen, prothrombin, tissue factor, and then calcium. 1, 2, 3, 4. Name the calcidependent factors: factor 2, 7, 9, 10. 2, 7, 9, 10. Why is blood clotting abnormal in patients with vitamin K deficiency? Vitamin K essential for gamma carboxylation transition modification of blood factors 2, 7, 9, 10. 2, 7, 9, 10. So in patients with vitamin K deficiency, there is reduction in activating factor 2, 7, 9, 10. 2, 7, 9, 10. Therefore, blood clotting becomes abnormal. That's why vitamin K deficiency leads to bleeding tendency. What are the increased vitamin K deficiency? Cause of vitamin K deficiency: fat malabsorption due to pancreatic dysfunction, biliary disease, destruction of bacteria and colony-forming destruction of large quantity of antibody, defined coagulation. The process by which soluble fibrinogen is converted into insoluble fibrin. Soluble fibrinogen is converted into insoluble fibrin by the help of thrombin, and a clot is formed. It's known as coagulation. A clot is formed. It's known as coagulation. Basic steps of blood coagulation: extrinsic pathway and intrinsic pathway. Conversion of path prothrombin to fibrin by prothrombin activator. Conversion of soluble fibrin to insoluble fibrin by means of fibrin and a formation of clot. Prothrombin, intrinsic pathway, prothrombin. Extrinsic pathway, intrinsic pathway, prothrombin. Intrinsic pathway, prothrombin. Prothrombin activator, intrinsic pathway, prothrombin activator, prothrombin, thrombin. Fibrinogen and fibrin monomer, fibrin polymer. Prothrombin into factor 8, factor 8 activator, factor 8 and factor 13, and then 13 activator. How does coagulation occur after blood vessel rupture? After vessel rupture, clotting actually occurs by both ways simultaneously. Tissue factor 3 initiates intrinsic pathway. Factor 12 initiates intrinsic pathway. Factor 3 initiates extrinsic pathway. Factor 3 initiates factor 7 initiates intrinsic pathway. 12 intrinsic pathway. Proteolysis, polymerization, conversion of fibrinogen into 3 ways: proteolysis, polymerization, clotting. Fibrin monomer, fibrin polymer, and insoluble clotting NH. State the extrinsic mechanism of blood coagulation. Extrinsic mechanism of blood coagulation. The extrinsic pathway for initiating the formation of prothrombin activity begins in traumatized vascular wall and traumatized extravascular tissue that the same content of the blood leads to the following steps: raise of tissue factor, traumatized tissue release, a complex several factors, tissue factors and tissue thromboplastin, composed especially of tissue phospholipids plus a lipoprotein complex, and then activator factor X, the lipoprotein complex of tissue factor, calcium, factor 8. Factors and ZA, prothrombin activator, factor 10A. Blood coagulation intrinsic pathway. The intrinsic pathway for initiating formation of prothrombin activator. The intrinsic pathway of initiating formation of prothrombin activator and therefore initiating clotting begins at trauma in the blood itself or exposure to blood or to subendothelial collagen in traumatized vascular wall. Then the process continues through the series of cascades of reactions shown in figure below. The intrinsic pathway for initiating formation of prothrombin activator. The intrinsic pathway for initiating formation of prothrombin activator and therefore for initiating clotting begins the trauma of blood cell. Activation of factor 9, activation of factor 10, action of activated factor, prothrombin factor, prothrombin. Enumerate the stages of formation of prothrombin activator for coagulation. Short note: prothrombin activator formation. Prothrombin activator is formed by both extrinsic and intrinsic. Extrinsic, intrinsic. Factor 10, factor 12, factor 8, factor 9, factor 8. Factor 9 factor 11, factor 9, factor 8. And a common pathway, factor 7. Then factor 10, activated factor 10A, and then factor 5 plus factor 5 equal to prothrombin, thrombin, and fibrinogen fibrinogen plot. Describe the detailed mechanism of clotting factor test tube. Clotting factors 12 platelet test tube activates in intrinsic pathway. So test tube intrinsic pathway, describe intrinsic pathway. Intrinsic pathway, tissue trauma, tissue factor, factor 7, factor 7A, and then the same intrinsic pathway. In extrinsic action, smart Soch power factor, thrombin factor 8, factor 8A, factor 9, factor 9A. Compare extrinsic pathway. Clotting factor 1 to 5, 1 to 5, 8, 9, 10, 11, 12, 1 to 5, 10, 7, 10. Number of reactions, more, number of reactions, less. Intrinsic. Trauma blood itself, transmuscular wall. Rapid process, slow process, rapid process. Factor 12, factor 7. State of role of platelet hemostasis. Platelet formation hemostasis, vasoconstriction hemostasis, platelet plaque formation, blood coagulation, and clot removal. A clot reactor. Vasoconstriction and helps clot formation, helps in clot removal by forming plasminogen. Platelet plaque adhesion and aggregation. Role of calcium coagulation. Ionized calcium primarily clotting factor except for two steps intrinsic pathway. What is retraction? In a few minutes, or a clot form begins to contract and loses most of its fluid within 20 to 30 minutes and reduces size to become tighter, more solid, elastic. This is clot reaction. 20 minutes. Fluid that comes out of the reaction is called serum, which is straw colored serum. Mechanism: attached to platelets with different fibrins to bind different fibers together. Attachment of platelets with different fibrins bind different fibers together. Release of procoagulant substances such as fibrin stabilizing factor causes more or cross-linking bonds between adjacent fibrins. Platelet directly contributing maturation of contractile proteins. Thrombin, calcium plus. A strong contractile platelets and then composition of blood framework. Clot reaction. Importance of clot reaction. As the clot reaction, the edge of the broken blood vessels are pulled together in efficient damage. Coagulation reaction. Discuss the mechanism of the fibrolysis and importance. Dissociation of the black clot by reaction of plasma or fibrinolysis is called fibrinolysis. Blood dissociation of blood clot by the action of plasma and fibrinolysis. Mechanism: fibrinolysis is due to proteolytic enzyme called fibrinolysin or plasmin. The fibrinolytic stem, there is no free plasmin, but blood plasma contains inactive precursor plasminogen. Plasminogen is converted into plasmin by means of plasminogen activator or any intrinsic or extrinsic. Physiological significance: clearing of blood vessels, reopening blood vessels, removal of blood minute clots from peripheral vessels and preventing the coagulation. Why blood does not clot intravascularly? Endothelial factor, smoothness endothelium, presence of monomer layer, speed of flow, presence and plasmin. Anticoagulant, antithrombin, heparin, antithrombin 3, protein C, protein S, alpha 2 macroglobulins, plasmin. For role of thrombin and protein: it binds with thrombin and clotting. Role of protein C: it binds with thrombin and then thrombin C vectors. Natural heparin, antithrombin 3, protein C, protein S, fibrin, heparin sulfate, thrombodulin and prostacyclin, alpha 2 microglobules. Common Paul-Heller's mixture. Paul-Heller's Haller's mixture. Potassium oxalate, 3.8% sodium citrate or oxalate. Hepat 3.8% sodium citrate or oxalate. Normal saline. EDTA, warfarin and dichromol. 3 common useful anticoagulants in important. EDTA, ethylene diamine tetraacid. Sodium citrate, heparin. Ethylene directed tetraacid. Write down the mechanism of action anticoagulant. Heparin acts by binding with antithrombin, inhibits reaction of several clotting factors. EDTA. Sodium citrate. Clotting is prevented. Free calcium and calcium from blood agents. Removing free calcium. Oxalate compounds. Double oxalate. Paul Haller's mixture. And then chemical mechanism. Will you prefer, because oxalate is toxic to the body, citrate or oxalate anticoagulant? Citrate, because oxalate is toxic to the body. Warfarin and then warfarin and then dacromol. So thromboxane and antithrombin 3, EDTA, heparin and then double oxalate and then sodium citrate and warfarin and dichromol. Warfarin, vitamin K exposure, inhibits vitamin K, gamma anticoagulant factors, decarboxylation, prothrombin, hydroxon and vitamin K exposure to reductase. Mechanism, importance of hematological changes in stored blood. Define clot. Clot is a network of fibrin that is running in all directions to intervening blood cells, platelets and plasma. Heparin was the first isolated from liver and it is called important intravascular anticoagulant. What is ACD solution? It is a solution which helps prevent blood into in vitro. This solution contains 102% sodium hydrates and 20 milli in 15% glucose solution. It is ACD, acid citrate dextrose, a solution which helps in prevent blood vitro. The solution contains 100 milli of 2% disodium hydrogen citrate and 200 milli of 15% glucose solution. Bleeding disorders are a group of disorders which is characterized by an abnormal tendency to bleed due to defective or in the mechanism of homeostasis. Bleeding disorders, vascular defect, purpura, thrombocytopenic purpura and non-thrombocytopenic purpura. Bleeding disorders due to platelet abnormalities, thrombocytopenia. Platelet count is less than 150,000 due to platelet abnormalities, primary, idiopathic and secondary. And then bleeding disorders due to coagulation defects, congenital hemophilia A, hemophilia B, Christmas disease, von Willebrand disease and other factors. Acquired vitamin K deficiency, liver deficiency. What is hemophilia? Hemophilia is an inherited sex-linked recessive coagulation disorder. Hemophilia is an inherited sex-linked recessive coagulation disorder where bleeding time is normal but clotting time is prolonged. Bleeding time is normal but clotting time is prolonged due to factor 8 and 9. Types and causes. Hemophilia A. Hemophilia B. Inherited hemophilia has linked recessive female as carrier, male as sufferer. So clinical features: wound bleeding, tissue bleeding, spontaneous bleeding, mouth and nose bleeding, and adenoma. Child with hemophilia. Screening tests. Clotting time. Tests are clotting time, bleeding time, and then complete blood count, CBC, complete blood count. That is not here, but platelet count, and then activated partial thromboplastin time. Activated partial thromboplastin time. So activated partial thromboplastin time. Activated partial thromboplastin time, bleeding time, clotting time, thrombin time, prothrombin time. Activated partial thromboplastin time, and then factor 8, factor 9. Factor 8 SI, factor 9 FA. Hard hemoglobin A. Infusion of hemoglobin 9. Nice to know. What is purpura? Purpura is a clinical condition characterized by the tendency of spontaneous bleeding beneath the skin, mucous membrane, and internal organs. Characterized by the tendency of spontaneous bleeding. Thrombocytopenic purpura. Decreased platelet count is called thrombocytopenic purpura. It may be primary purpura in case and secondary. Thrombocytopenic purpura, bone marrow depression, leukemia, acute septicemia. Bleeding is common in thrombocytopenic, can occur in count less than 50,000 cubic millimeter of blood, millimeter cube of blood. Non-thrombocytopenic purpura, infections, deficiency of vitamin C, and then allergic purpura and connective tissue disorders. Sites of bleeding: skin, mucous membrane, and internal organs. Bleeding time prolonged. Prothom bleeding time normal. Clotting time normal. Give the difference between hemophilia and purpura. Hemophilia, septic disorder, clinical condition characterized by a tendency, male to female interest in disease, spontaneous bleeding in the skin, organs, male to female who manifest the disease. Coagulation is prolonged, bleeding time is normal. Here coagulation time is normal, but bleeding time is prolonged. It is due to deficiency of vitamin A, factor 8 and 9. Capillary factor, due to platelet deficiency of capillary factor. It's inherited, only show in male. It is not inherited, maintains both male and female. Cause of bleeding: hemophilia A, excessive bleeding. Activated peripheral thromboplastin. Activated peripheral thromboplastin. Prothom bleeding time, bleeding time, factor 8, genetic settings. Prothome diagnosis: purpura and idiopathic thrombocytopenic purpura, most common. Screening test: bleeding time, clotting time, prothom bleeding time, bleeding time, clotting time, prothom bleeding time, activated partial thromboplastin time, prothom bleeding time, platelet count. Prothom bleeding, activated partial thromboplastin time, prothom bleeding time, and prothom bleeding time. Discuss coagulation tests. Bleeding time: normal value less than 6 minutes. The time required for bleeding. Importance: platelet response to avascular injury. And then the clotting time. Clotting time is normal value 6 to 12. For coagulation, clotting time. Cause prolonged hemophilia, factor 8 deficiency. Bismuth, factor 9 deficiency. DIC. Consumption of clotting factors. DIC. Prothom bleeding time acquired to take place in extrinsic pathway. Prothom bleeding time. Deficiency factor 1 to 5, 7, 10. 1 to 5, 7, 10. 1 to 5, 7, 10. And as BT, CT, PT, APTT. And Wolfram therapy. Activated partial thromboplastin required for clotting takes intrinsic pathway. Activated partial thromboplastin. So prothom bleeding time takes 9 to 12 seconds. Time required for clotting takes place in extrinsic pathway. And activated partial thromboplastin 32 to 40 seconds. So 6 minutes and above, 6 minutes and less, and then 9 to 12 seconds and 32 to 40 seconds. Thumb bleeding time 10 to 14 seconds. Fibrin assay. TIC, liver disease, platelet count 1.5 to 4 lakhs and Q. Define thrombary or emboli. An emboli, clot that develops in blood vessel. Emboli: develop blood, continue blood flow, breaks away from attachment, and blood clotting agency. Blood groups. Classification of blood according to the presence or absence of different antigens are called plasma membrane. Basic blood grouping: RH blood group, and MNN blood group. Louis Cal Daffy Diego Kidd. Louis Daffy Diego Kidd. Aim of blood grouping: main to identify individuals ABO and RH type, safe population of pregnancy and pregnancy management. So blood group and anti-A serum, anti-B serum, positive, negative, positive, anti-A serum, anti-B serum. Name the agglutinogens and agglutinins of ABO in RH group system. Give the basis of principal blood grouping: ABO grouping, RH grouping. RH is negative. What's Landsteiner's law? The law states that reciprocal relationship between agglutinogens on the RBC and the agglutinins of the serum. First part. It is an agglutinogen present in red cells of the blood. Corresponding agglutinin must be present in the first part and second part. Landsteiner law. First part, second part. It is an agglutinogen is present on the second cell of the blood. If an agglutinogen is present on the rest of the blood, the corresponding agglutinin will be absent from plasma. If an agglutinogen is absent in the blood, the corresponding agglutinin will be present in the plasma. Kernicterus is a rare neurological condition that occurs in the same newborn with severe jaundice. Probable diagnosis: hemolytic disease of the newborn, RH incapability, erythroblastosis fetalis, and hydrops fetalis, hyperbilirubinemia, and kernicterus. Blood transfusion: person into a circle another individual is called blood transfusion. Compatibility, fresh frozen plasma, transfusion of blood, whole blood, red cell concentrate, white cell concentrate, platelet concentrate, fibrinogen clotting factors, and frozen plasma, fresh frozen plasma, white cell, red blood cell, platelet, fibrinogen, and fresh blood plasma. Compatibility screening criteria: mechanism of renal shutdown following transmission, renal vasoconstriction, circular shock, renal tubular blockage. Individual blood transfusion must be placed for compensation of decreased RBC count, for compensation of blood volume. What is autologous blood transfusion? Autologous transfusion is called reinforcing the patient's own blood into the blood component. Collection, transfusion collection, and reinfusing. How inclusions differ from cell organelles. And cytoskeleton. The cytoskeleton is a series of intracellular proteins that help in cell with shape, support, and movements. The cytoskeleton consisting of a network of tiny protein filaments and tubules that extends towards the cytoplasm. It serves as an important structural framework for the cells. The cytoskeleton has three main structural components: microtubules, microfilaments, and then intermediate filaments. Function of each component of a cytoskeleton. Microtubules, they provide support and shape the cell. Microtubules participate in intracellular transport and cell organelles, assist in cellular movement. Cilia, flagella, centrioles from a spindle which moves, and cytochrome mitosis. Microfilaments participate in muscular contraction. Microfilaments. So microtubules help in forming the cells, helping in containing the cell shape, supporting the cell shape, and also in transportation of other organelles, also assist in cellular movement. Cilia, flagella, and centrioles, and participate in intracellular transport and other organelles. Intercellular transport and other organelles, and participate in intracellular transport and other organelles. And microfilament in particularly in muscular contraction. Intermediate filaments from leukocytes. Neutrofilaments help in intermediate filaments link cells to others. Intermediate filaments link cells to others. Neurofilaments help to maintain cylindrical shape of axons. Loss of water and heat, smooth muscle. So microtubules help in supporting the cell shape and also transportation of cellular material and organelles. Microfibrils, microtubules, microfilaments help in muscle contraction, participate in moving and shifting of cytoplasm, help in endocytosis, phagocytosis, movement of flagella, etc. And then intermediate cells help in linking the cells. Intermediate filaments: epithelial tissue, cytokeratin; connective tissue, venentrin; cytokeratin, venentrin, destin; neurofilaments, GFA; soluble acid protein. Crystals are composed of microtubules. Sequence of events: smooth muscle contraction, depolarization of cell membrane, opens voltage gated channels. First, depolarization happens due to opening of voltage gated calcium channels. Calcium flows into the cells into the electrochemical gradient, increasing in intracellular hormones and neurotransmitters may open in ligand gated channels and other membranes. Calcium enters in the cell release hormones and neurotransmitters also directly release calcium from SR 145 triphosphate channels. 145 triphosphate IP3 channels. So depolarization happens due to opening of voltage gated channels. Intracellular calcium releases. Intracellular calcium increases. Intracellular calcium increases. Depolarization of cell membranes opens voltage gated calcium channels and calcium flows into the cell down to electrochemical gradient. And calcium flows into the cell down to electrochemical gradient. Cell binds with calmodulin. The calcium calmodulin complex binds to active myosin chain kinase. Decrease and relaxation. Decrease intracellular produces relaxation. Calcium binds with calmodulin actin chain, and then decrease intracellular calcium produces relaxation. On the granulocyte, they have cytoplasm. On the basis of staining of the nucleus and the size of the granules, they are of three types: neutrophil with granules taking both acidic and basic stains, eosinophil with granules taking acidic stain, basophil with granules taking basic stain. Neutrophil, eosinophil, basophil. Granulocytes, they have no granulocytes in the cytoplasm. There are two types: leukocytes and monocytes. Lymphocytes and monocytes. Lymphocytes, large lymphocytes, small lymphocytes. Classified WBC with their normal values in peripheral blood. Total count of TC or WBC. It is the total number of white blood cells per unit volume of blood, cubic millimeter. TC or WBC, 4,000 to 11,000 centimeter of blood. Different count of blood, different count of white blood. Differential count of W cell is the percentage wise distribution of individual cells per 100 WBC. Absolute count of WBC, it is the total number of individual white blood cells per unit volume of blood, cubic millimeter. Count of WBC, differential count, absolute count. Neutrophil 50 to 70%, neutrophil count 20 to 40%, monocyte count 2 to 8%, eosinophil count 1 to 4%, basophil count 1%, 3,000 to 6,000, 1,500 to 2,700, 300 to 600, 150 to 300, basophil 0 to 100. Morphology of RBC. neutrophil 12 থেকে 15 মাইক্রোমিটার, ইউসিনোফিল 12 থেকে 17 মাইক্রোমিটার, বেসোফিল 12 থেকে 15 মাইক্রোমিটার, আর মনোসাইটস 12 থেকে 20 মাইক্রোমিটার। লিম্ফোসাইটস, লার্জ লিম্ফোসাইটস 12 থেকে 16 মাইক্রোমিটার, স্মল লিম্ফোসাইট 6 থেকে 8 মাইক্রোমিটার। Cytoplasm: pink, pale blue, pink, pale blue, bluish gray, pale blue. Multinuclear, multi-lobe, 2 to 5 lobes, usually 3. Bilobed, large, curved, horseshoe-shaped, kidney-shaped, round, occupies most of the cell, living in the rim of the cytoplasm. Granules: purple, orange, red, blue, black, absent and present, absent. Function of WBC: phagocytosis by macrophages, eosinophils, neutrophils. Provide immunity by T cell, B cell and macrophages, etc. Antibody formation by B cells. Antibody formation by B cells. Provides immunity to T cells, B cells and macrophages. Secretion of heparin by basophil and thus prevents intravascular blood coagulation. Secretion of heparin by basophil and thus prevents intravascular blood coagulation. Eosinophil kills parasites. Antihistamine action. Eosinophil acts as a scavenger. Write down the functions of WBC. neutrophils. It has an important function in neutrophil is the phagocytosis. The most important function of the neutrophil is the phagocytosis, the synthesis and release chemotactic factors for macrophages, thromboxinase and platelet aggregating agents, leukotrienes and phago and prostaglandin that exert a moderate inflammatory effect. Neutrophil synthesize chromatic factors, chromatotactic factors, thromboxane and platelet aggregating agents, leukocytes and prostaglandin. Monocytes enter the tissue from blood and become tissue macrophages. Macrophages perform the following functions: phagocytosis, antigen presentation, production of cytokinase. Phagocytosis lymphocytes, helper T cells, activates cytotoxin T cells, B cells, macrophages and other antigen specific helper T cells. Cytotoxin T cells kills virus infections, tumor cells and allograft cells. B cells produce antibodies. At first, B cells are converted to plasma cells which produce antibody. Eosinophils, they kill parasites by releasing hydrolytic enzymes, major basic proteins and reactive O2 intermediates. They reduce allergic reactions by release of histamine that degrades histamine by releasing histaminase that degrades histamine. Eosinophils are weak phagocytes and not sufficient to protect against pathogenic bacterial infection. In neutrophilic eosinophils are weak phagocytes are not sufficient to protect against eugenic bacterial infections. In neutropenic infections, basophil, they secrete heparin that prevents intravascular blood coagulation. They play important role in some types of allergic reactions by releasing histamine, bradykinin, serotonin and heparin. How do eosinophils kill parasites? Most of the parasites are too large to be phagocytes by eosinophils or by any phagocytes. Eosinophils attack the surface of the larva or parasites and kill them by releasing hydrolytic enzymes, major basic proteins, and probably receive O2 intermediates. Properties of WBC: diapedesis, amoeboid movement, chemotaxis, phagocytosis. Diapedesis is the process by which leukocytes come out of the lumen of the blood vessel into the tissue space is called diapedesis. Mechanism: after firm adhesion, leukocytes insert pseudopods into the junctions between the endothelial cells, squeeze through endothelial junctions. Eventually, they pass along the basement membrane that escape into the extravascular space. Neutrophil, monocytes, lymphocytes, eosinophil and basophil all use the same pathway to migrate from the blood vessels into tissues. Amoeboid movement: both neutrophil and macrophages move through the tissue by amoeboid movement. Maximum rate of movement is 40 micrometer per minute. Chemotaxis: after extravasation, leukocytes emerge in tissue towards the site of injury by a process called chemotaxis. Defined most simply as locomotion oriented along the chemical gradient, leukocytes are attached to the site of injury by chemotaxis agents, which include exogenous chemotactic agents, bacterial or viral toxins, endogenous chemotactic agents, degenerative products of the inflamed tissue, complement 5CA, leukotriene B4, cytokinase, products of blood clotting. Phagocytosis is the process. Phagocytosis definition: it is the process by which WBC neutrophil and phagocytosis engulf the foreign bodies and damage tissues. Steps of phagocytosis has 3 steps: recognition and attachment. Phagocytic cells recognize microbes by their surface receptors. Phagocytosis is greatly enhanced by microbes and opsonized by specific proteins called opsonins. The major proteins are hemoglobin G, 3CB, and MBL. Engulfment: after attachment, phagocytic projects pseudopods around the foreign bodies and completely enclose it. This creates an enclosed membrane bound cytoplasmic vessel called phagosome. Phagosome fuses with lysosome to form phagolysosome. Killing and degradation: the microbes and the dead cells are killed by oxygen-dependent micromechanisms. Phagocytosis stimulates increased O2 uptake, glycogenesis, increased glucooxidation via the HMP shunt respiratory burst, and production of relatively O2 intermediate such as superoxide, hydrogen peroxide, glycochlorine, HOCl, which kills microbes, oxygen independent micromechanisms. Bacterial permeability increases protein. These proteins degrade phospholipids and increase permeability of other membrane bacteria. Lysosome: hydrolysis bacteria cells walls. Lactoferrin chalates iron. Major basic proteins. Cytotoxins to many parasites. Defenses: cytotoxin to microbes. After killing, acid hydrolysis, which are normally stored in the lysosomes, degrade the microbes within the phagolysosomes. Figure: phagocytosis of pathogens such as bacteria by a phagocytic cell such as macrophage. Antibodies coat the bacteria, making them more susceptible to phagocytosis by the morph that engulfs the bacterium, forming a phagosome. Lysosomes then attach to the phagosomes to form a phagolysosome, which digests the individual invading pathogens, which phagocytic cells then release the digested product by exocytosis. Define the following terms. Erythropoiesis: the process of production of WBC under physiological condition. Leukopoiesis: the process of production of WBC under physiological condition. Granulopoiesis: the process of production of granulocytes under physiological condition. The process of production of lymphocytes under physiological condition. Thrombopoiesis: the process of production of thrombocytes, platelets under physiological condition. The process of production of thrombocytes, platelets under physiological condition. Leukocytosis: increase in number of WBC above the upper normal limit of 11,000 per cubic millimeter of blood. Increase in number of neutrophil above the upper normal limit of 6,000 cubic millimeter of blood. Neutropenia: the decreased number of neutrophil below the lower number of 3,000 cubic millimeter of blood. What is leukopenia? Decrease in number of WBC below its lower limit of 4,000 cubic millimeter of blood. Leukocytosis. Definition: when RBC count is more than 11,000 millimeter cube of blood, it is called leukocytosis. Cause: neutrophilic leukocytosis, infection, trauma, infraction, inflammation, malignancy, myeloproliferative disease, and physiological exercise, pregnancy. Neutrophil: myeloproliferative disease, polycythemia, chronic myeloid leukemia. Malignancy: solid tumor and Hodgkin lymphoma. Inflammation: gout, venous arthritis, Crohn's disease. Infraction: myocardial infarction, pulmonary embolism. Trauma: surgery, burns. Infections: bacteria and fungal. eosinophilic leukocytosis. Allergy, hay fever, asthma, eczema, infections, parasitic, drug hypersensitivity, gold sulfonamides, skin disease, connective tissue disorders, polyarteritis nodosa, malignancy, solid tumors, lymphomas, primary lobe marrow disorders, myeloproliferative disorders, hypereosinophilic syndrome, acute myeloid leukemia, infections, bacterial, fungal, trauma, surgery, burns, infractions, myocardial infarction, pulmonary embolism, sickle cell crisis, inflammations, gout, Raynaud's arthritis, ulcerative colitis, and Crohn's disease, malignancy, solid tumors, Hodgkin lymphoma, myeloproliferative disease, polycythemia, chronic myeloid leukemia, physiological exercise, pregnancy, allergy, hay fever, asthma, and schisma. Drug hypersensitivity, gold sulfonamide, skin disease, eosinophilic leukocytes, connective tissue disorders, malignancy, primary bone marrow disorders, basophilic leukocytes, myeloproliferative disease, polycythemia, chronic myeloid leukemia, inflammation, acute hypersensitivity, ultrasensitive colitis, Crohn's disease, iron deficiency, monocytes, chronic infections, bacterial endocarditis, rickettsiosis, malaria, collagen vascular disease, inflammatory bowel disease, malignancy, solid tumors, pregnancy, inflammation, viral, bacterial, lymphoproliferative disease, chronic lymphatic leukemia, lymphoma, post-splenectomy, accompanies monocytic many disorders associated with chronic immunological stimulations, viral infections, hepatitis A, cytomegalovirus, Epstein-Barr virus. Nice to know. Cause of neutropenia, infection, drugs, autoimmune, alcohol, bone marrow infiltrations, and congenital Costman syndrome. Cause of lymphopenia, inflammation, lymphoma, renal failure, sarcoidosis, drugs, congenital. Right short note, macrophages. Macrophages are one of the components of mononuclear phagocytic system. Diameter 10 to 30 micrometer. Nucleus, oval or kidney shaped origin. Macrophages arise from the blood monocytes. From the blood, monocyte migrates into the various tissues and transfer into macrophages. The process of monocyte to macrophage transformation results in the increase in protein synthesis and cell size, bone marrow, blood, and tissue. Lifespan several months. Activation. Macrophages can become converted into active macrophages, epithelioid cells, and giant cells. Distribution of macrophages: skin, Langerhans cells, liver, Kupffer cells, brain, microglia, lungs, alveolar macrophages, bones, osteoclast, kidney, mesangial cells, lymph nodes, dendritic cells, spleen, splanchnic macrophages, and red pulp macrophages. Function of macrophages: phagocytosis. Macrophages phagocytes microorganisms damaged in dead cells and cellular debris, digest ingest material. Engine presentation: production of cytokines such as IL-1, IL-8, TNF. Macrophages are the central figure for chronic inflammation because macrophages can produce a great number of substances required for chronic inflammation. Short note: mononuclear phagocytic system. Mononuclear phagocytic system, formerly called reticuloendothelial system (RES), is the network of immune cells that play a crucial role in phagocytosis, antigen presentation, in immune regulation. Components: monocytes, macrophages, and dendritic cells. Origin: mononuclear phagocytic cells arise from a common precursor in the bone marrow which give rise to the blood monocytes. From the blood, monocytes migrate to various tissue and differentiate into macrophages. Functions. Phagocytosis engulfs and destructs the phagogens, dead cells and debris. Antigen presentation. Present of antigen T cells, intermediate adaptive immunity. Cytokinase secretions, release of inflammatory and anti-inflammatory mediators. Tissue repair, aid in wound healing and tissue remodeling. Functions. Antigen presentation, cytokinase secretion and tissue repair. The MPS is essential for both innate and adaptive immunity, maintaining homeostasis and defending against infections. Clinical significance. Dysfunction can lead to chronic inflammation, autoimmune disease, or immunodeficiency. White blood cells, total different of count. Steps of phagocytosis. First line of defense against infection. The first line of defense against infection is provided by certain cells that act as part of the body's innate immune system. The key cells innervated in this initial defense muscle: macrophages, neutrophils, dendritic cells, mast cells, natural killer cells. White blood cells classification. Function of white blood cell. Properties of white blood cell phagocytosis. Immunity, allergies and inflammation. Immunity. The human body has the ability to resist almost all types of organisms or toxins that tend to damage the tissue and organs. This capability is called immunity. Immunity definition. The human body has the ability to resist almost all types of organisms or toxins that tend to damage the tissue and organs, and the capability to and is called immunity. Definition. Immunity is the process of recognition and elimination of antigen from body. The outcome of the process may be beneficial of the human or harmful to the host. Beneficial or harmful to the host. Classification of immunity. Immunity: innate immunity, acquired immunity. Acquired immunity: active immunity, passive immunity. Natural immunity, artificial immunity. Natural artificial immunity. Natural artificial also has cell-mediated immunity and humoral immunity. Initiate immunity. Innate immunity, also called natural or native immunity, refer to defense mechanisms that are present even before exposure to antigen. It is non-specific. Acquired or adaptive immunity. The required immunity, also called adaptive or specific immunity, refer to the defense mechanism that develop after exposure to antigen, improves upon repeated exposure, and it is a specific. Active immunity. This is the type of acquired immunity where immunocompetent cells of the body participate actively in. For two types: natural immunity. Active immunity, which develops after clinical and subclinical infections, is called natural active immunity. Artificial immunity. The active immunity which develops after vaccination is called artificial active immunity. DPT. Vaccination produces immunity against diphtheria, pertussis, and tetanus. According to types of cell involved, active immunity is divided into two types: cell-mediated immunity. T cell participates in CMI. Humoral immunity. B cell participates in HI and produces antibody. Passive immunity. This type of immunity, where preformed antibody is produced into a host, is produced specific immunity in two types. Natural immunity. The immunity caused by passing of immunoglobulin G antibody from mother to the fetus during pregnancy through the placental route is immunoglobulin A from the mother. Newborn breastfeeding through the breast milk is an example of natural passive immunity. These antibodies offer immunity for 6 months after birth. Artificial immunity. Administration of antibodies performed in other hosts produces artificial passive immunity. For example, ATS, anti-tissue serum, ATC, anti-diphtheria serum, and AGT, tetanus hemoglobin, produce artificial passive immunity. What is innate? Non-specific immunity. Innate immunity, also called the natural native immunity, refers to the first line of defense mechanism that exists before exposure to antigen. Non-specific. Innate immunity. Innate immunity refers to the first line of defense mechanism that exists before exposed to antigen. Non-specific. Cardinal features of innate immunity. Innate immunity exists prior to exposure of microbes. It is non-specific. It includes hosts defenses such as barrier to infectious agents, certain cells and certain proteins that involve processes such as phagocytosis and inflammation. It does not improve after exposure to the mechanism. It has no memory. Functions of innate immunity. Two major functions. Innate immunity is the initial response to microbe that prevents, controls, and eliminates infections of the host. Stimulates adaptive immune response. Two major functions. Innate immunity is the initial response to microbes that prevent, controls, or eliminate functions of the host. Stimulates adaptive immune response. Lists the component of innate immunity. Mechanical, physical factors. Certain layers of intact skin, intact mucosal lining of the different organs, washing action of the tears, saliva and urine, respiratory cilia, muscular layer of the prevent over the inner surface, coughing and sneezing reflexes, chemical factors, fatty acids of the skin, inhibit growth microbes, acid and pH, sweat and sebaceous cells inhibit the microorganisms, lysosome in tears, nasal secretion and saliva degrades bacteriological and bacterial cell wall, spermine and zinc in the serum are antibacterial and antibactericidal. Lactoperoxidase in milk has bactericidal action. Low pH of the stomach gives and vagina inhibits the growth of many microbes. Biological factors, cellular factors, normal bacterial flora presence in throat, colon and vagina inhibits colonization by other pathogens, bacterial interference, phagocytosis in necrophils are neutrophils and macrophages. Natural killer cells are involved in extracellular killing of the target. Eosinophils involved in killing of large parasites. Surface phagocytosis, for example, alveolar macrophages, Kupffer cells, liver, etc., are involved in second line of defense. Soluble factors. Interferon secreted by various virus infected cells and other immunocomplement cells inhibit viral replication. Incomplete mediate cell lysis and inflammatory reaction. Acute phase proteins, C reactive proteins, alpha 1 antitrypsin, alpha 2 microglobulin, fibrinogen, C9 factor B, all acts either on the limit on the spread of infection agents to the stimuli of the host response. Genetic factors. Individuals with deficiency with glucose 6 phosphate dehydrogenase are resistant to Plasmodium falciparum. Genetic factors. Plasmodium falciparum resistant. Miscellaneous factors. Elevated body temperature. Many microbes, for example, Glucococcini and tamponins, are inhibited over 40 degrees Celsius. Transfer of hemoglobin of other fetus through placenta. Transfer of hemoglobin A from mother to infant by breast milk. Transferrin and lactoferrin. Subsequent iron required for bacterial growth. Fever. Elevated temperature retards bacterial growth. Inflammation response limits spread of microbes. Causes hypermutation in retroviral DNA and mRNA. Name the immune cells. Illustrated functional importance of the cell body. Cellular response to innate immunity. Natural killer cells, neutrophils, macrophages and dendritic cells. Cellular components of acquired immunity. Antigen presenting cell, antigen recognizing cells, effector cells, regulatory cells, macrophages and dendritic cells, viral infected cells and tumor cells, activated B cells, T cells, B cells, B cells, up regulation by CD4 T cell, down regulation by CD8 suppressor cells. Adaptive immunity. Acquired immunity is also adaptive immunity, refers to the defense mechanism that develops exposure to the antigen. improves upon adaptive exposure and is specific. This mediated by antibody production of B cells are by two types: T lymphocytes and T helper cells. T lymphocytes and T helper T cells and cytotoxin T cells. Write down the cardinal special unique features of acquired immunity. Cardinal special unique features of acquired immunity. All humoral and cell-mediated immune response to foreign antibodies have a number of fundamental properties. Specificity ensures that distinct antigen elicit specific responses. Reasons. Diversity enables immune system to respond to large amount of antibodies. Memory leads to enhanced response to repeated exposure of the same antigens. Clonal exposure increases number of antigen-specific lymphocytes to keep pace with microbes. Specialized contraction and hemostasis and non-reactive to self. Different and between active and passive immunity. Briefly discuss the function of T cells. Macrophages, maturation types, and according to cell types, according to the function. And shorter T helper T cells, B lymphocytes. Give the difference between B and T lymphocytes antigens. Nice to know. Haptin, MHC proteins, antigen presenting cells, class 1 MCH. What is antibody? Structure of immunoglobulin antibody. Define and classify immunoglobulin. State their important functions. Immunoglobulin classify substance of immunoglobulin. Functions of immunoglobulin G, immunoglobulin M, immunoglobulin D, immunoglobulin E. And how cell-mediated immunity produced in the body. Helper T cell and cytotoxin T cell. How is antibody-mediated immunity? And outline of the humoral and cellular immunity. Complements definition, number of complements, pathway of complement activation, biological function of complement system, inflammation, and role of neutrophil inflammation.